Bom dia a todos. Professor Canuto, Scientific Provost of University of São Paulo. Jennifer Hurley, Deputy Vice President of the International Affairs Direction of Institut Pasteur. Professor Raul Machado, former President of the National and International Agency of USP for Academic Cooperation. Mirdad Kazanji, my copain, General Director of the Institut Pasteur de la Guyane and Regional Director of the American Network of Institut Pasteur. I'm sorry, I'm a little bit emotional, so I am going to tremble a bit. Dear colleagues, today we are here at the University of São Paulo, initiating the Regional American meeting of the International Network of Institut Pasteur. Mirdad and myself have jointly proposed to organize this event here in Brazil because Institut Pasteur consecrates a large importance to the partnership with the academic institutions, research centers, and University of the Southern Cone, and additionally, an historical collaboration exists between Brazilian institutes and France. We have a common culture, common roots, and this alliance is reciprocally beneficial. We would like to thank our University of São Paulo friends to host this initiative that we are delighted that became true, and all the partners of the American region, Institut Pasteur de la Guadeloupe, Institut Pasteur de Montevideo, Laval, Fiocruz, and Institut Pasteur de la Guyane Française, that helped us to build a very interesting scientific program. Please, you can sit. You have a place there. With the support of the French Embassy, the General Consulate of France in São Paulo, and the regional French delegation, we were able to associate to this meeting scientists from several South and Central American countries, such as Bolivia, Uruguay, Chile, Peru, Argentina, Costa Rica, and Colombia, that are not in the RIIP, actually, that will be enriching the future partnership we can develop in the region. Special thanks to Alexia, Perini, and Samantha from the French Embassy. Finally, we will have later on the possibility to share with all of you the inauguration of the first platform issued from the tripartite agreement between Pasteur, Fiocruz, and USP, the scientific platform Pasteur-USP. I would like to publicly express my gratitude and friendship to all the colleagues from this new platform and the ones from USP that allowed us to organize this event. I would particularly cite all the students and group leaders of the Pasteur-USP for all the efforts they are deploying to set up the scientific platform and to move forward to our common goals. Thanks to our colleagues from Inova USP, Stella, Peter, Terezinha, Lick, colleagues from other that work in the Inova USP, Maria Alice and Zancu, our colleagues from Alcani, Jonatas, Sonia, Nicholas, and Paulo, and a special recognition to our partners of Thermo Fisher and the Society Sotelab, Nicholas, Senhor Fernando, Carlos, and Viviane, to our colleagues from CEF, MDP, and Biosafe, Luciana, Larissa, Professor Nakao, Pedro, and Marcelo. And I cannot forget to express our gratitude to the security agents, protocol, and press from the university campus, the Biomedical Science Institutes, here represented by my colleague and partner, Luis Carlos Souza Ferreira, to FAPESP and the Pasteur International Network Association. Finally, we thank all the speakers and participants, and I wish you an excellent meeting. I pass the microphone to our Professor Canuto to welcome you all, the Scientific Provost of the University of São Paulo. Thank you very much.
Thank you, Paula. I have to say I'm nervous too, because it's a very important occasion. Good morning to everyone. It's a great pleasure, really indeed, to be here today to address you a few words on this opening. So let me first salute and thank my colleagues at this table here. I have to salute Professor Jennifer Hurley. I have also to mention my colleague Raul Machado, who represents the international cooperation. Professor Mirdad Kazanji, my dear colleague Paula Minoprio, and Dr. Kathleen Victoire. It is, as I said, a very important occasion, and I take this opportunity to thank the organizers also, in special Dr. Paula Minoprio, and I have also to thank Professor Luis Carlos Ferreira from the Institute of Biomedical Science for taking this journey along. And on his behalf, I thank all the colleagues that worked to make this happen. I should also mention the name of my colleague, Professor Catalani of the Innova USP, for participating in this and for hosting this partnership. So I have to thank also the outstanding group of scientists for attending this meeting. It's a great pleasure having you all here at the University of São Paulo, and I should say it's a great honor to host you. I think one of the aims, or perhaps the purpose, or perhaps one of the aspects of this scientific meeting is to give a testimony of the quality of the research that is coming in the coming years. So this partnership with the Institut Pasteur is a long-time aspiration, possible now, thanks to the effort of many of our colleagues that are here present. And again, I have to thank them on behalf of the University of São Paulo. This is a very important partnership for carrying high-level scientific research. Today, and perhaps I should mention officially tomorrow, we have finally come to this historic date where the Institute of Pasteur and the University of São Paulo give an important step in the development of common projects to attack present and emerging diseases. So briefly, these are my words that I want to address you, but before I finish, I should say that on behalf of the University of São Paulo, and in special on behalf of our rector and president, Professor Vahan Agopian, I would like to say that I'm very proud to welcome you and wish you a very productive meeting. Thank you very much.
I would like to ask Professor Raul Machado to say 2 words.
I'm supposed to say 2 words. Yes. Thank you. Good morning. I just would like to express my very I'm very pleased with this event. This consolidate one very special partnership that is starting 2014, and I'm very honored to be a person that could contribute a little bit to construct this partnership and to consolidate the presence of Pasteur at the University of São Paulo. So I wish you all a fantastic meeting, and tomorrow a great celebration. Thank you for being here.
Now I'd like to ask Jennifer Hurley, Deputy Vice President of the International Affairs of Pasteur Institute, that started the Projet Pasteur with us in 2014. And I'm very happy that you are here with us today, because we followed every step of this project together with Luis and Jennifer. And I am very happy that you are here. And please.
Thank you very much. Thank you very much, Paola, and thanks to all for being here today. I will say a few words, of course, for the Institut Pasteur of Paris, but also in my own name, as Paola just said, because I'm following this partnership, very important partnership, since the very beginning in 2014. So I have lots of emotion today also. I would like to say a few words to Paola and to our guests from the University of São Paulo, because they have made this very ambitious meeting happen. Very ambitious meeting, which has been made possible also because Mirdad Kasanji here, the Pasteur International Network Association, and all the institutes of the network have accepted that for the first time in the history of the Institut Pasteur International Network, such a meeting, a regional meeting from the association, is taking place in an institution which is not currently member of the international network. Not currently, not yet member. So it's something which, in my opinion, puts the inauguration of the Pasteur USP scientific platform tomorrow under the best auspices, because it's blessed by the network. And I'm very happy to say that. So now I wish you a very fruitful meeting, a very fruitful days, and I leave the floor to Cathy and Victoire.
Thank you very much for passing me the word. So on behalf of the Pasteur International Network Association, I'm very proud to be here, and I'm very proud to be in Sao Paulo, in the university, to start these partnerships with the members of the network of the Pasteur Institutes. I think this meeting is an important meeting because, as Mirda said me yesterday, it has been since 2014 that we didn't have a regional meeting. The Americas is for us an important region. There's lots of things going on, and for that I think it's really great that our other institutes which are represented at this meeting from the region and who are wanting to work with our network. And I thank you very much for joining us at this meeting, and I thank very much Paola for having taken the lead on that. It's very important, and for the next steps towards how we are going to work in this region, what are the activities that we are going to develop with the region of the Americas and the other regions in the network. This should be an important starting point. And I talk about starting point because I think it's important also to have strong follow-up on these meetings to see how we can integrate all the actions we are going to talk about. Thank you very much.
Thank you, Katie. Now I would like to ask Mirdad, that is our partner to the organization of this meeting. So,
thank you, Paula, dear friends, dear colleague. It's a great pleasure for me today to be here. And as this morning we said with my friend Luis Carlos Ferreira, that the dream became reality. And I would first congratulate Paula for this great job that you made. And it's really, it's your œuvre. Je sais pas comment dire œuvre en anglais. And believe me, it's not so easy. With Luis, I started this project in 2014. And when I saw the building for the first time, I said, oh, this will be the Pasteur Institute in the future, or the platform. And I visited the labs 2 days ago, and it's really a wonderful place, great place. And congratulations all of the team that participate to that. Then this dream became reality, and during this day we'll inaugurate this platform, and this is something very important for Pasteur Institute and also for the network. Just a couple of words of our network is 32 institutes across the world, and it's divided to different regions. And we have the American region here, and the American region is 5 institutes, including the Fiocruz. And I would like to take a couple of words just to thank the Fiocruz, because they deeply participate to this project, and I'm very happy to see all the team from Fiocruz coming today and join us to this meeting, that since 2014 we didn't get any American region, that it was the opportunity today to join the inauguration of the platform and the American meeting, including all the institute and other institute, which is, you know, the network with other countries, Biociências network. Then this is a great opportunity for all of us to discuss science, to think how we can collaborate together, what ideas, what project we can build together. And I really believe that these 3 days will be very great opportunity for all of us to discuss and to think together how we go forward with this platform, with Fiocruz, with other an institute in the American region. Then these 2 days will be divided first to scientific meeting with we try to participate all the institute on one big thematic that all of you know, that since many years now, the arboviruses was a great problem for all of us. Zika, chikungunya, dengue is still a big problem, yellow fever. And this is why we thought that that would be very good to think first on these emerging viruses in the region and in the American region. And here I was really impressed in Sao Paulo, as well as in the Ficus. There's great teams working in this area. Paulo Zanotto, Edmond, and other friends. They work very great teams working on that. And I really believe in the future we will get more and more collaboration together. The second part of the meeting will be consecrated to brainstorming between us. And I really believe that this is the opportunity also to see how and can we work together in the future and build a great collaboration. Then thank you again for all of you. Very happy to see my friends here that we collaborate during last years, and to see today that in a couple of hours we'll integrate this new baby to the network. Thank you again, thank you, and I hope really that we'll enjoy the meeting and enjoy the discussion. Thank you again.
Just to inform you, we have more than 200 subscriptions, how do you say that? Yeah, to the meeting, and I think that subscribe. So people are arriving. We have some minutes, 10 minutes to start, but as the program is very condensed, I would just ask Mirdad if we can start with the session, and I acknowledge everybody participating in this table, and I leave the stage to Mirdad. I don't know if Eduardo Massad is here. Eduardo Massad, that was supposed to share the session with Mirdad. May I invite someone else? Edson Durigon, can you share with Mirdad this session?
Edson.
Thank you very much. Thank you all for that.
Thank you very much, Paola.
Okay.
Thank you very much.
Okay. Then Paulo Zanotto is here. I saw him this morning. Yeah. Then we will start the scientific meeting, and today is mainly consecrated to research on arboviruses in America. And the first talk will be given by my friend Paulo Zanotto. Where is Paulo? Paulo's there. Okay. And he is going to give a talk on new challenges of emerging diseases in the America. He's virologist and working for the evolution of the viruses. More virus evolution, yeah. Okay. Go on, Paulo.
Good morning. I'm very happy to be here. I'd like to thank you all for being here. As a matter of fact, most of the people here know each other. So it was like a family reunion, but it's a very interesting situation because now we're in a different position. We're a family with a house. So for that regard, I'd like to thank you all that got so involved in having us this house. Savino, Paula, Luis Carlos, Edson. I mean, I can't say the names because you're all here, so you know what it means to get a house. And from the deep of my heart, I thank you all for that, for this big, 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 impressive effort. And it is a big honor to be here receiving you people. And I hope I don't make a big mess in this house. I promise to be well behaved. So now I would like to just position this situation in which we have this kind of serious problem of outbreaks. A friend of ours, Amadou Al-Fassal, is not here because he's in Congo now. He was supposed to be here. Part of his team is here, but most of them are in Congo working in the Ebola outbreak. So it's very usual to have this situation. These people that will live in this house is always going to be involved in very messy situations. So what keeps us together is this notion that we're doing something good. And I really like to be part of this effort. So one of the first things I would like to do is like a few concepts of emergence that basically the emergence of disease depends mostly on anthropogenic activities, on man shifting, encroaching niches, causing anthropogenic degradation in environment, and human overpopulation, urbanization being made on completely, let's say, unreasonable ways. And so basically, we are living in a situation today on planet Earth that we have to think seriously what we're doing, what we're going to be doing, and how we can help in reorganizing this relationship with planet Earth. So our house is part of this effort. And there are several other important efforts here at the University of São Paulo in this direction, like the Big Cities project by Marcos Bucherich, things done by the Institute of Advanced Studies at the University of São Paulo. So I think it's a very nice place to be at because we're going to be associating with several independent activities towards that. So like a 101 on emerging infectious disease would be the fact that we have humans. Well, the color changed a little bit. But anyways, we have humans here. And basically, this is a representation of the fact that we have transitioned from animals, from the zoonotic pool to peridomestic animals, and eventually to humans. And then the thing gets really bad once you have trafficking from one species to another of a disease, and eventually this thing reaches humans and may establish itself on the web of transmission of humans, and then you have what we call emergence. This happened with yellow fever, probably in the 16th century, with dengue a little bit after that, and so on, until recently when we were following with our friends in Africa, Zika virus, and I was bored working with that virus because it was like a poor cousin of dengue, and people in Africa said, no, you should keep working with it, and we did, and eventually, you know the rest of it, because it just went ballistic here in Latin America, and the Americas in general. So this idea that things have a zoonotic cycle basis, and then eventually they gradually seep into human transmission, is crucial for us to understand emergence. And based on this notion here, we have this concept of One Health, meaning that in order for us to see which viruses are going to arrive in humans, we have to consider the entire scenario, from the zoonotic host in the wild to transition among species. Eventually, this thing is going to end up in humans. So it's crucial that we are proactive. We have to look before things become a problem. So Zika taught us a big lesson, because we actually had the potential, sorry to say that, but if 20 years ago we had all things ready for Zika, diagnostics, vaccine, so on and so forth, we probably would have dealt with this situation in a very different way. I think it's a big lesson for us. We had this lesson with SARS, when people in Germany had dismantled coronavirus research, and all of a sudden SARS pops up, and the government in Germany comes to the German side and said, what the hell, are you not doing nothing with corona? So Martin Pfeiffer takes this big list of projects that were canceled by the government because they were dealing with a sneeze virus, coronavirus, because like something very mild. So that's the message to take home. We have to be ahead of the game. So now I'm going to be talking a little bit about several issues here. This is a very nice figure that I got from Gustavo, who works with Edson Durigon. And you see several bats, and several of these bats are, this is a heat map, number of pathogens. And you can see that some species of bats are thick in terms of condensed, and being like basically carrying several viruses. So it's very important to do this research that Edson is doing here, looking at bats and everybody else. And it's very interesting that if we look at the map of global hotspots for disease, Brazil is almost central on planet Earth. And mainly, these things here are bats. So the largest diversity of bats on planet Earth more or less follows this graph. And the least known viruses in bats are exactly in the area where you have greater diversity of bats. But this is just an example for you guys to see how this thing is a problem. How if we think forward, we have to start taking considerations some kind of long-ranging plans of actually looking at things we don't know yet what they are. So, very interestingly, if you see the map here, those are flaviviruses and density of activity, and filoviruses, basically in Africa, you don't see nothing in America. But you see, those are bats. So we would imagine, and Tatiana is here, she can tell you, I hope you can talk with her, that eventually if we look very closely to these bats, we're going to find things that would make us really scared today. So it's very interesting to realize that we still do not have a good balance between our understanding of distribution of species, zoonotic species, and what they are carrying. We know that, but what else are we missing then? That's the basic notion. So this is a very interesting map of Brazil. I thank Gustavo again for that map. And it's very interesting to show that agriculture land, meaning regions where we have anthropogenic degradation in Brazil, are very much intermingling with forest. And as a matter of fact, if you look at the rainforest here in São Paulo, it's very interesting because it basically, it's recolonizing environment. Now, so you're getting a tighter connection between rainforest, forest, and crazy, unsound urbanization. So the thing is becoming like a pressure pen, if you will. Interestingly, if we see the global hotspots, areas where emergence of diseases are more likely, exactly the place where we have our house today is in the center of one of those, which is exactly here in this region of Brazil, which is very interesting. So you have to think of the fact that we're putting our house also in the middle of the largest conurbation south of the equator. So we have a few miles around us, we have almost 23 million people living packed together near forested areas, near animals that we don't know what they are carrying, and so on and so forth, which makes our task momentous. So an interesting thing is, if we look at now, we jump from bats now to the mosquitoes. Okay, let's go very fast with that. And you see this is Aedes aegypti. I don't know, this thing is not working. Well, thanks. Aedes aegypti, it's taking the midfield. It's like playing soccer when your opponent takes, hand of the mid. It's a very tough match. Because the most densely populated areas in the planet as well are the ones that are all infested with vectors for flaviviruses. Meaning that this makes our mission daunting. So don't think of anything else but a very tough match ahead. And this is our friend Aedes. But we have to think of the fact that Aedes gets infected from humans. We are the infecting agents on mosquitoes. So that's crucial that we understand. Demography, human density, urbanization, basic health, social justice in the end of the day. If it's not done, we pay with disease. So again, this is the distribution of Zika, overlays precisely the distribution of this is just an example in the islands of the Pacific, and also is overlaying exactly the distribution of vectors. And several other viruses will come on top of it. So let's look at some of the viruses that we have here in Brazil. These are probably going to be the ones that we have to look in a proactive way. So these risks like Busuquara, Cacicaporé, Rocio, Iguape, Ilhéus, Laranjal, those are viruses that are here in Brazil, and that they are related to Japanese encephalitis. They're basically neurotropic viruses, and we know nothing about these guys. And probably they are circulating among our populations, and we need to know what they're going to be doing eventually. West Nile is very well known. West Nile is making its way to Brazil. There is an increasing trend through time of the virus spreading through the Americas. And the reason why it didn't come here early on, maybe almost 18 years ago, is probably due to the fact that we have so many other flavivirus here that may cross-react with these guys, that some kind of herd immunity could be playing a role. We don't understand that. But then dengue, we know all of these guys. Yellow fever is being established here since the time of the Portuguese, when the Portuguese arrived here in the 16th century. But then this thing goes into the forests and living there. You're going to see in a while what a mess it's been causing, and what happened in the siege of the city of São Paulo last year, the ongoing siege, if you will. And the Zika virus. Those are very interesting because this group is related in terms of genetic phylogeny, and those are related to Japanese encephalitis, and those are related to yellow fever and dengue and Zika. Those are all cousins. That causes a problem because they cross-react in terms of serology, making a daunting challenge as well for diagnostics. But now we have to think of further and desired newcomers. Usuto virus, I'm going to talk a little bit about Usuto and what we've been doing with our fellows in Dakar, in the Institut Pasteur in Dakar. Rift Valley fever virus. Anyone from the Pasteur Institute remembers Michel Boulard in Paris working with this virus, and how problematic this virus is and may become if it moves out, because it moved out of Africa from the Rift Valley, went to the east, to the west of Africa, and then moved to Saudi Arabia. We're going to talk about that in a while. Sponduini is another virus that we should think about. Cotango, Barkeji, you see, these viruses, Sponduini is a problem. It's a Zika-related sibling that has sufficient diversity that eventually may cause some kind of enhancement if associated with Zika, if it comes in a superposition of circulation. And also West Nile, we're going to be talking, which is a serious problem because West Nile lineage 1 moved out, boom, caused a massive problem in the Americas. But I'm going to show you what's going on with 8 other lineages a little bit. So that's the scenario. This thing becomes, the last lesson on our 101 in EID, is basically these things circulate in animals. Eventually, it spills over to the inner core of peridomestic interaction with humans, and eventually jumps into humans and causes a massive problem. So we have to talk about control operations. If within the One Health concept, if we interact at certain stages here, this thing is, if we are able to do mitigation efforts, we may reduce the amount of impact in humans because we cut the transmission, okay? So when like 20 years ago here in São Paulo, and I think this has a lot to do with what we're doing today, we thought, well, let's do something about it, shall we? That was the basic idea of the very minute community of virologists in São Paulo. So then we create this VGDN, this network that was funded by FAPESP. And it's very interesting because I'm very sorry that I make a comparison with Rio de Janeiro. Rio de Janeiro has way more tradition in virology than São Paulo, basic virology. The biophysics department in Rio de Janeiro had fantastic virology there, and it still has. But this is more or less the rate of publications. And this is what happened when FAPESP started funding the VGDN. It was a massive enhancement. Edson knows because Edson was involved in making maybe 7 to 10 P3 labs around the state of São Paulo. And all of a sudden we had 25 laboratories doing virology in São Paulo. So it's like, and you can see immediately by scientific production. This is something that Eduardo Massad, unfortunately, is not here. He made this very nice graph, and we sent it for publication, because this is clear impact. So now let's see what's going to happen once we put another slice here and we go, house. Paula's monopoly effort, what's going to cause? It's probably this thing is going to go ballistic, I hope. has to go as ballistic as dengue, okay? And so during the time that VGDN was like, giving us conditions to do things, what we've actually done, for instance, this is a very interesting, one of the coordinated tasks of VGDN. Others were like HIV, hantavirus, 2,500 square kilometers in Brazil. We sampled and we looked at this virus and we made the movement of this virus. And this was published, very nice work. And extremely important collaboration from Adolfo Lutz and from people from Fiocruz as well. And then in RSV, we made this beautiful work. Edison was collecting samples since 1990 something, sorry. And we see the oscillation, the changes on different clades of RSV and how they respond to the herd immunity in humans and how after a while they come back, the old lineage comes back. So it kind of oscillates in time. And it's very nice findings we made that. And but this is something we did with dengue as well, because we can do, once we have people, have money, have computers, start working. This is for dengue 1, and we see 3 genotypes of dengue 1, and we can do the migration movement around the planet Earth, and we see that Latin America receives, but it's like sinks, okay? But the sources are in Asia. This is well known, but now we can dimension, we can measure, we can look at the rates of migration and how these things work, and then we can immediately think of the between the centrality of some nodes as the place to interfere with. So this idea that by following viruses, we know where they are going, what they are doing, therefore we can think about how to interfere is very well shown in this study here with hepatitis C, which we see different genotypes of hepatitis C in time having different behaviors. And we see then all of a sudden, very recently, one of the genotypes start to pop up, goes ballistic. So what's going on? We measure based on sequences that we have sampled from people at known dates. We can actually measure those different populations. This is basically a density of time, estimate of time of entry of different viruses in the population. But the funny thing is that if we ask people, do you do sex? Obviously they say yes. But then you say, how many? How much? And then when we measure that, it's fantastic because all we see here is that this is the connectivity due to a number of partners. And you can see, that's very interesting, the difference between the 1B, that was the virus that infected, the generation before ours. These people were infected mainly by transfusion. Oh, gosh. By transfusion. Whereas the new virus, the ones that are blowing up recently, are the people that do a lot of sex, tattoos, go to prison, use drugs, the bad boys. So interestingly enough, once we published that, it caused a massive problem. People went crazy over that because Hep C is taking, people understand Hep C as something benign, being transmitted by other means. And all of a sudden we have this evidence that there's one genotype of Hep C that basically has an association with behavior, social structure, and so on and so forth. The CDC made, 1 year after we published that, made in the yearly date of Hep C, they put exactly an individual that fits all these kind of things as a subject of a message. Meaning that once we know about what's underlying an event, we may do an interference and it may work. So it's very important to know that there is sexual transmission Hep C. This paper was crucial in kind of establishing this reality because here is a study done with almost 1,500 patients of all ages in 20-something cities in Brazil, in the state of São Paulo. And there is a very strong association, behavioral association with different genotypes, and this may explain the temporal variation of these things. Now, I'm moving very fast towards, I have a few more minutes. This is dengue in São Paulo. These are notified cases. If we look this up and down thing in time, it's like government saying, I did well, oops, I did well, oops, I did well, oops, okay? But if you fit a very simple model, that's what we got. And interestingly enough, this is the predicted, but this is the actual number of people that were infected by dengue, 3.5 million. in Brazil, in São Paulo, let's say, in 2014. And now the thing is coming back. But I'll show you why I don't believe this thing is going to go up. First of all, this is work we've done in São José do Rio Preto, a city that you have like 2 different lineages spreading through city, and we know through time how they spread. Very interestingly, they were associated, the spread of it, with a river that crosses the city at the same time where the shanty towns are. Again, showing a very serious implication of social and basic health issues on the spread of diseases. Interestingly enough, we are still looking at dengue in São Paulo, and now we have something very interesting. We go in the city of the conurbation of São Paulo, go Jundiaí, Guarujá, Santos, so we find the 4 serotypes of dengue co-circulating in São Paulo, which is kind of an amazing story. The government got really upset that we were looking at dengue 3 here, but dengue 3 is probably reintroduced in 2000, and from a Chinese origin. So you ask someone like Jeffrey Powell from Yale, which kind of mosquito you're having here in São Paulo? He's going to say, in Santos, he's going to say Singapore. And these things come crowned with eggs. They're all infested. These containers come with dried eggs everywhere. Once these containers arrive in Santos, they start getting rain, because rainforest, and all of a sudden these eggs say, oh, second, third rain, there I go. Then you have mosquitoes coming, and eventually these guys can vertically transmit virus like the ones we found. So it's very interesting to look at this scenario here. So now I'm going to tell you a story which is very nice. This is Guarujá. It's very near the harbor here. It's across the harbor. Is that landmass you see across the channel. And it's a place everybody goes in the summer. From the state of São Paulo, they move in this area for the summer, for the beaches. Very nice. I don't have a picture, but believe me, it's very, very cool. You should go there. But eventually, we have this very strange scenario here where we are following people from the 33 communities around this island, 175,000 people living in 33 communities attended by 22 units of basic health. And interestingly, Julian was a student with his PhD. He comes around and he says, to Odimara, which is the vector control agent here, said, well, we are measuring NS1 in people, and NS1 also indicates when the guy has the virus in the blood. And what happened is that immediately she sees that some areas have more cases than the others. She starts putting insecticides there, and we move from 200 cases down to 10 cases a week report. I mean, the thing collapsed. It was very interesting to know this is concerted operations being applied, and immediately the thing collapses, as you can see. So if you go back and see the people that did not have dengue but were showing disease, we found underneath it, a few years ago, we go in the freezers and we find an outbreak of RSV. And several of these patients were diagnosed as dengue. So it's very interesting how we should actually pay attention. So now I'm going to move into Zika very fast. These are our friends from Dakar coming here to assist us during the thick of the operation, who had no access to anything here in São Paulo. So they came and they gave us samples, they gave us serology, they gave us technology. We trained 32 scientists in the state of São Paulo. Here's Amadou Afassal in São José do Rio Preto, talking with Maurício Nogueira. And here is his team training scientists here. This is Marielton sitting looking at something, probably. And this moved us into a very fast, boom, operation. This is a very nice work done by Patricia and Jean-Pierre. And it's very interesting because we can see immediately that they were able to show that Zika virus could, as a matter of fact, cause alterations in mice. And at the same time, the first, not the first, but one of the good descriptions of what was going on in terms of reduction of animals in the brain and impact in the immunology due to the infection. So this is a very nice picture that we have, like, those are Patricia mini brains, and we were calculating the multiplicity. This was like in the middle of the night, we are calculating these infections, and that's few weeks, a few days before this paper was submitted for publication in Nature. It was very interesting because we end up with a very good, let's say, titer of infection that allowed us to see very good things in terms of explaining what the virus was causing in these mini brains. Interestingly enough, then Jean-Pierre had the idea of collaborating with his friend Rafael Larocca from Dan Baruch's lab in Harvard. This immediately jumped into the group in Harvard. They had a proof of concept. It just worked very nicely. So now, just a few more seconds. We see the movement of West Nile in Africa, so the different lineages, we have 8 lineages. One escaped out, went to Europe, but the very important thing is to see the lineage 2 now is moving to Europe as well. So even if lineage 1 went to the Americas and caused a massive problem there, now we're waiting for other lineages to move in. That's very important. Rift Valley again moved, this is the west of Africa, the movement of West Nile that we've been actually following with our friends in Dakar. And it's very interesting that the thing moved, and we also made the movement of a virus in Africa. And it's very interesting that it just got noticed because we were publishing this thing, but the other thing goes to the line. Why? Because this thing goes into Saudi Arabia and eventually becomes a serious problem. And also we find reassortment in this virus, meaning that the virus is a shapeshifter. It may cause, like influenza, very strong alteration. And the last thing I'd like to talk with you is the Lesotho virus that we are following with our friends in Dakar as well. And it goes urban. This is a city near Koudou, and you see the virus being, it's being sampled from mice, from rodents in the city. We are now following this virus in terms of seeing all its changes. The virus actually made to Bologna in Italy, in Vienna, and eventually Lesotho Savino is going to come into Brazil. So if I were in Fiocruz, I would be strongly working with this virus because it's probably going to be our novel boy and a new kid on the block. So yellow fever, the bugs, distribution in Africa and Brazil. The main difference being that this is caused urban cycles and here sylvatic cycles. And possibly to the fact that the Aedes aegypti you have now in Brazil, it's from Asian origin, was replaced old African Aedes aegypti, and this guy is no good for transmission between humans. So this is basically the first wave of Zika coming, of yellow fever coming through monkeys in São Paulo from the west. As you can see, this thing is not affecting the central area of São Paulo. We know now, Marialto knows now, that eventually what happened later on in the city was due to another introduction. And people in Fiocruz know already they have different lineages spreading through Rio, coming down to here. This is the number of cases. During this time, Shahab and Marialto were working with Amaro. There in the school, they actually got almost 90 samples from people who died of vaccine, and they're working on it. And they find virus everywhere in the blood in the body of these people, and the entire description of disease is changing here at the medical school because now they are following the virus everywhere. And we also are following the virus is spreading to Brazil in São Paulo, and you see that it's accumulating changes. And those are very important, Paola, because now we have to make infectious clones, change those positions, and see what they do with the virus. And finally, we see the city of São Paulo, number of cases. This map shows more or less, this is the siege of São Paulo. This is the area of São Paulo. These are monkey cases in white and human cases. And the very interesting thing is that the distance between humans is larger than the distance between men and monkeys, meaning that eventually we could be understood as a proof that this thing is not being transmitted through men. So there is no human-to-human transmission of yellow fever in the city of São Paulo. If we see the data that Marialto has now, it's almost like the city works as the deflector. The virus arrives in the city, goes around, and goes to the south and spreads out. So I would like to, I'm on time. I would like to make this statement again. I would like to thank you all, great science and human beings. I can't read that, but I know that's basically what I said in the beginning. You could read for me. And it's an enormous pleasure to be here, and I thank you again for all that you've done, allowing this thing to happen. Bye-bye.
Thank you. Thank you. Thank you, Paulo. Thank you for this very interesting talk. I will take one or two questions if there's any question. Okay, you didn't mention any other arboviruses which is endemic, for example, in South America, like Mayaro, like Orupuch, like.
Yeah.
What do you think about the emergence of these viruses?
Well,
in the future?
Mayaro is becoming a serious issue. We are growing the virus in the lab. Edson is following, we are following it. We're having cases everywhere. We're receiving samples from other localities in Brazil that we are dealing with the virus. We are growing it in cell lines. We're getting ready. Now Orupuch is the same thing. Rocio is the same thing. There's some very important evidence that arenaviruses are still around. We had an 87 outbreak, one case of Sabiá virus that caused a massive problem in the EAO. The virus actually caused a problem in the lab. You all remember that. And the EAO's arbovirus unit was moved to Texas because of it. It was a virus that was circulating here, maybe less than 8 kilometers from where we're sitting now. But what we have is an evidence that these viruses are still around. Rocio, massive outbreak, more than 1,200 cases of encephalitis in 75. Roughly 96 people died. But if you go in the 80s and you sample, you do serological surveying in the population, they're still there. People that were born after that outbreak have IgG. So it's for Rocio, which means that the virus is still circulating. So as I told you before, the nice thing about having this house here in the middle of São Paulo is because we are sitting in the middle of a very important area where anthropogenic degradation took, São Paulo, the temperature in the city, around the city, increased 4 degrees since I was born. So if we're talking about climatic change, okay, whatever it may mean, we see that São Paulo is an area that is post-warmist fear, because we are warmer here. And the environmental conditions here change so much that we are near a scenario where probably if things will take place, we're going to be around. And we have to be very attentive and aware of that. So all these viruses, we're considering them, and we're growing them. And the idea would be to make infectious clones, sample from the field, and move on into trying to understand what they do and what we can do to developing monoclonal antibodies, snap tags, diagnostics, and so on. That's our plan, is to study these things. But mainly because you guys told us to concentrate on neuropathology, we're going to, isn't it, Paula? We're going to focus mainly on neuroinvasive agents here, because this thing is important.
Okay, if there is no question, I think we can move to the next presentation. No question? Okay, thank you, Paulo.
Thank you very much.
Thank you. I will present Anubis, and after that, I will present. Okay, the next talk, the next speaker is Anubis Vega Rua. Anubis, she works in Pasteur Institute in Guadeloupe. She is an entomologist, and I am very happy to announce that Anubis became a permanent position, get a permanent position at Pasteur Institute a few weeks ago. Then we are very happy now to get her for a long time in the American region. And today she will give a talk on current gaps and challenges regarding vector control in the America. Thank you.
Good morning, bon dia. So before I start, I want to thank the organizers. Paola, thank you very much for giving me the opportunity to be here today to discuss with you about the current challenges and gaps of the vector control in the Americas. So as you know, vector-borne disease are a global and major public health problem. They account for 70% of the estimated burden of the infectious disease, with more than 700,000 deaths per year, and they also put a high number of population at risk of contracting this. As you can see here, 80% of the population is at risk, and when you look at the distribution of the vector-borne disease, you will notice that in fact they are mainly distributed in the tropical and subtropical areas, and we are really concerned by this disease in the Americas. So, as you can see, in the Americas, we are really concerned by parasitic disease and also arboviral disease that are transmitted by a variety of vectors. And you can see here that all these diseases, they really have important burdens associated. They put millions at risk. They are widespread in many countries, sometimes in more than 20 countries. And they are transmitted by mosquitoes, phlebotominescent flies, kissing bugs, and so on. But as you can see, most of them are transmitted by mosquitoes. So during this talk, I will talk about vector control, but I will focus mainly on mosquito species because of their importance for the region. And as the common point of all this disease is the vector, very rapidly we realized that if we control the vectors, we could reduce simultaneously the burden associated to this disease. And what we are doing in many regions is to implement integrated vector management programs. This is a very old definition that was already used by William Crawford Gorgas in the beginning of the 20th century. And the idea is to have an optimal use of the resources for vector control to overcome the challenges that are experienced with single intervention approaches. Like that, we can increase the efficacy, the cost effectiveness, the ecological soundness, and the sustainability of the implementation of this. And this is how an integrated vector management program looks like. I took that of the Mosquito Vector Control Association of California. So mainly you have several axes. We have a very nice introductory talk about the surveillance and the importance of the surveillance of the vectors and the disease. We have also the vector control methods that are purely applied. We have research axis in order to improve the vector control methods we have and also to do a quality control of the interventions. And of course, we cannot do this alone, so we need really to build collaboration and to work with the communities to enhance the positive outcome of the results. And in the past, these integrated vector management strategies resulted in several successes, and some of them took place in the Americas. We have, for instance, the eradication of yellow fever in Cuba in 1903, also from Panama during the construction of the Panama Canal in 1904. In Brazil, also, we have the decline in the populations of Anopheles gambiae. In Latin America, in general, we have a decrease on the infestation rates, but the kissing bugs in many regions. But despite these successes, we're still being challenged by this vector-borne disease. And we are still seeing a contemporary increase of certain vector-borne disease in the Americas, as you can see here with the example of dengue. Here you have the number of cases and the death caused by the disease, and you will see that in all the regions of the Americas, especially in the southern cone, the incidence is increasing. So why this is happening if we are doing integrated vector control programs? The thing is that, as it was mentioned before, the challenges for IVM strategies are greater today. We are not in the same context that at the beginning of the 20th century. We have a commercial and touristic air travel network that is more developed. Also, we have a growing urbanization along with the population. And these are factors that contribute to the spread of the pathogens. And we have the clear example of Zika. Here you have a study that modeling all the travel network that were departing from Brazil. And you can see how the virus used these networks to travel and to spread in many regions. We have also the global trade, which is a problem, a major problem, because it contributes to the propagation of the vectors. And we have the example of Aedes albopictus mosquitoes that was introduced in many regions via the trade of the used tires and the lucky bamboo, for instance. And here you have the map of the Americas with the colonization of albopictus with different dates. And this was after the eradication program that was conducted to eliminate yellow fever in the 60s and the 70s. Also, we have climate change, and there are several studies that show that the changes that can be, that can happen in the future years can modify the distribution of the vectors. And here you have a study that was conducted in Ecuador that shows that the suitability areas for distribution of several vector species will change by 2030 or 50. So the distribution will be different than the current one. And what it is very complex is that these species have different ecologies, so the changes in the distribution will not happen in the same manner. You have some vector populations that will shift, that will decrease, others will increase, so this adds a layer of complexity to the challenges. So overall, all these global challenges increase the cost and the implementation difficulties for the integrated vector management programs. But besides these challenges, we have particular challenge of the use of the vector control that we have historically done. So broadly, the vector control methods can be divided in physical, also called environmental vector control, chemical and biological. And the physical vector control was the oldest that was implemented, as you can see in this picture where the people were filling or draining. I don't see very well, but this water body. And the idea of this is to modify the environment to reduce, especially the immature stages of the vectors. So you have some measures like the mechanical elimination of the breeding sites, the source reduction, and improvement of the water supply and water storage, and waste management also. And this was the first measures that were taking place at the beginning of the 20th century. But then, at the beginning of the 21st century, people started to interest in the improvement of the housing as an alternative to reduce simultaneously several vector-borne diseases. Because when you are changing the housing, you can change parameters such as the humidity, the temperature, the accumulation of the CO2 in the houses, and you can even reduce the vector entry. So for those that are biting indoors, and this will probably change and modify the contact between the vector and the humans. And because of this idea, many programs have been, well, some programs have been implemented, and you have here the housing improvement program for Chagas disease. Here you have the house before the program, and then, so you have a new house, and when they record it and they follow up the visits of the kissing box, they realize that you don't have kissing box here. But you can imagine that all this is very costly. But the good thing is that now we have made some technological processes in the way of constructing houses, and by doing minimal changes, you can reduce dramatically the contact between the vectors. And it was demonstrated in this trial in Gambia, where for this house prototype, by only changing the doors and using fitted doors, they reduced the entry of Anopheles gambiae mosquitoes by 96%. So at this can be promising. However, where are we so far? So this is a study that was conducted that reviewed all the housing improvements since the 80s until 2017. And when you look at the quality checking and the workflow of the authors, you realize that first, they found only 82 studies in all these years that attempt to do house improvement and to see the results in the vector community. But when you look into the detail, 50 of these studies didn't have appropriate controls or quality checking of the intervention, which means that at the end, only 32 studies have been retained. So 32 studies since 1980. So this is a clear gap because we have few trials and with inappropriate efficacy assessment. So as I said before, the improvement of the housing could be a physical vector control method that could be very interesting because you can simultaneously, in principle, reduce many vector-borne disease. So I think that this deserves further exploration, and by increasing the number of trials, but with a nice protocol that allows to standardize and to measure accordingly the efficiency of the intervention. Of course, this is not easy, I'm aware of that. This is costly. You need strong community involvement, political and economical support, which makes it's very difficult to make implementation in large scales. And because of all these problems with the environmental vector control methods, the chemical methods, so they have seen the daylight. So at the beginning, to make a little bit of history, the very toxic insecticides like the Paris green were used for the vector control. Then you have all these well-known DDT campaigns, but in the years that followed, the people realized that the use of these insecticides was hampered by the lack of the specificity of these insecticides and also by the negative effects that these had on the environment and on human health. And also they found out that they select for resistance and the mosquitoes and other vectors could resist to their use. So because of this, many countries started to change molecules. And here I'm giving you the example of Guadeloupe that I know very well, but it's the same for many regions. At the beginning of the 50s, we used DDT, then malathion, temefos, then deltamethrin, and as we are in France, so since 2009 and 10, we cannot use all these insecticides, which makes that now we are just using deltamethrin, an insecticide molecule that has been used for more than 30 years. So what happens when you have such policy of vector control? This is what happens. Your mosquitoes are like this, multiple resistance to insecticides. As you can see in this table, these are measures that we have taken in the lab. So these are our field populations, and here you have the resistance ratios for temefos, malathion, and deltamethrin. When these ratios are superior to 1 or 2, this means that populations are resistant when compared to the susceptible strain that was used here. So the situation is complex. And when you look further, the mechanism associated with this resistance, you find that in fact you have target site mutations that reduce the affinity of the insecticide by the target, and this confers to your mosquito a resistance to the knockdown effect that is normally provoked by a neurotoxic insecticide. So here you have the susceptible genotype without the mutation in the susceptible strain Bora Bora, but when you can see here are our populations, you don't have homozygotic susceptible genotype, you have homozygotic resistance in blue and heterozygotic. So the mutations are almost fixed in the population. And when you look at other mechanisms, like the detoxification enzymes, that they degrade the insecticide, which will be less toxic for the mosquito and can survive, you see that you have several genes coding for these enzymes that are overexpressed in our field populations. Here you have the baseline of the expression in a susceptible mosquito, or these genes, and here you see that many of them are overexpressed. So you have a combination of mechanisms. And this problem in Guadeloupe is also seen in other islands in the Caribbean. But today we don't know which are the mechanisms associated with these resistant phenotypes. And when you go further into the Americas, ta-da, exactly the same. So you have here the frequency of mosquitoes that are dying when they are exposed to a diagnostic dose of deltamethrin, which is supposed to kill the susceptible mosquito. And you see that everything is in red, which means that the mortality with this dose is almost 0. When you look at another molecule, the temefos, you have similar results. You have very high resistance ratios when compared to the susceptible strain. And regarding the mechanism, we have the same problem that in Guadeloupe. There are several mutations that have been reported for the Americas that confer target site resistance, and also many of genes coding that are overexpressing, and they code for detoxification enzymes. So this is a big problem. And so far, I'm just talking about the insecticide resistance linked to Aedes aegypti. But what about Aedes albopictus, which is a mosquito widespread in the Americas, and that is able to transmit almost all the Aedes aegypti-borne viruses? When you look at the availability of the data regarding insecticide tests on pyrethroids and also organophosphates, you see that you have data on Aegypti, but there is almost anything for Aedes albopictus. So this means that you have a serious knowledge gap, and this is just an example of all the gaps we have for insecticide resistance. Because when you think and you look backwards and you ask the question, what have we learned? You realize that some questions like, was the strategy we use for chemical vector control appropriate? Which have been the results? Did we collect all the needed data regarding the vector populations before the intervention? Do we know which strategy have worked the best and where? Because maybe I'm doing a specific vector control strategy in my island, but in the neighboring island they are doing something better than me. So perhaps I can get inspired by the good practices. So this is very important to move forward. And here, information sharing is really lacking. And also we have many questions that when you look at them, the answer is that we don't know because we have not looked enough backwards. So what is the context today? We have, of course, a diversity that is very good because we need diversity in this world. But also these different legislations that we have, they result in different vector control strategies. And sometimes the legislation is not adapted to the local needs. There are molecules that are forbidden. There are other molecules that are so promoted, but you know that these molecules are useless. You know also you have a problem of lack of standardized resistance assessment and surveillance. You have isolation, you don't have enough information sharing to have feedback on the good practices, and you need to fill these gaps, and a way would be to make a transfer of competence and capacity building, because if we want the different countries and regions to make a surveillance and assessment of the resistance, they need these capacities. And building networks like the WIN could be a step that could improve this problem. But of course, networks that really work and that they keep doing their work correctly. And beyond resistance, the insecticide resistance can also modify all other mosquito phenotype traits like the behavior and the competence for pathogens. We have seen, for instance, with Anopheles mosquitoes that after the implementation and the massive distribution of insecticide-treated mosquito nets, the mosquitoes changed their activity. They were resistant, so somehow they learned that if they were biting earlier or later in the night, when the people were not under the mosquito nets, they can be successful to achieve their blood meal. And also they started to bite outdoors, while these mosquitoes usually bite indoors. We have shown experimentally that these mosquitoes have different behavior and perception when faced to human odors. And regarding the competence, you have here this study. These are these are mosquitoes that have the same genetic background, and the only difference will be the mutation that conferred the target site resistance, and the prevalence of the sporocyte will be higher in the resistant when compared to the susceptible one. So again, this shows that our populations of vectors are evolving, so our IVM strategies should also evolve along with the vectors. And because of all these problems with the chemical use of insecticides, the biological vector control methods saw the daylight, and you have many approaches that have been proposed for targeting the immature stages. We have, for instance, the use of larvicides and oviposition deterrents, or we have also the predators of immature mosquito stages. So regarding these larvicides and oviposition deterrents, we have many knowledge gaps regarding the plant extracts that can be used as larvicides, and regarding the oviposition deterrents also. We don't know that much regarding their characterization, their specificity, and today the only, let's say, biological insecticide that is really being used in many countries will be the BTI. That is a toxin released by this bacteria, Bacillus thuringiensis. But the problem of this is not also, even if these BTI have different toxins, which is good because it will have a less selective pressure for resistance, the remanence of the BTI is low, so their use in large breeding size, for instance, will be challenging, and in regular breeding size, you will need to put over and over again. So we have logistical issues. And regarding the use of predators, the first thing is that the three of them, they are omnivorous. So there is a specificity problem because, of course, they eat the larvae we want, but they can also eat other organisms that are present in the breeding sites. And so this effect in the non-targeted fauna will not be very beneficial for the environment. However, the use of these larvivorous fish have been used in 60 countries with very good results, but in big breeding sites. In small breeding sites, their use is more complicated. You can imagine to put a fish in some millimeters of water, that won't be easy. The other approach that I found really interesting will be the use of Toxarachnites mosquitoes. So these are very friendly mosquitoes because they do not bite, they are not hematophagous at all, but the larvae, they eat the larvae of the vector. So this will be a nice approach in the countries that have this mosquito. But, of course, for this has a logistical cost because you will need to make a mass breeding of the Toxarachnites and of the mosquito vector because you need to feed them. Regarding the copepods, the mass rearing will be easier, but you have the suitability of habitats that do not always match with the habitat of some mosquito species. However, there have been successful trials in Vietnam. Sorry about the dates. It was between 1993 and 2000 that have a very good result in the reduction of Aedes aegypti mosquitoes. Also, all these approaches that targeted immature stages work if we found the breeding site, but you have many breeding sites that you don't see. So you will always have a population that will be developing there. And other approaches also of biological vector control have been proposed to targeting adults. You have, for instance, the use of entomopathogenic fungi. So this is very interesting because they are toxic for many insect species, which allow you to implement an approach One Health, because they can be toxic for insect pests, also for vectors of animal pathogens, and also vectors for human pathogens. And they have several toxins like the BTI, so you have a selective pressure that will be less important than if you use a chemical insecticide. However, we still have knowledge gaps regarding the delivery methods to increase the specificity of all these entomopathogenic fungi, and also they have different variations in their virulence and so on, and this should be further characterized. And finally, we have the methods that the principle is to release modified mosquitoes in order to reduce the populations, and I'm just going to talk about the three principles of them. The first one will be the release of sterile males that have been irradiated. The idea is that these sterile males would mate with wild-type females, and you won't have an offspring, so your population will reduce. And it's the same principle of the Riedel approach, where you are using genetic modified mosquitoes that are being released, and when they mate with the wild-type female, the mosquitoes, the offspring will die while developing in the breeding site and will never reach the adult stage. And finally, you have the Wolbachia approach that will be slightly different. You transfect here the mosquitoes with the bacteria that is present in other insects, and the phenotype that results from this transfection will be resistance and refractoriness for the arboviral infection. So in principle, these will be mosquitoes that are not able to transmit the arboviruses, and when they mate with the wild-type mosquitoes, the bacteria will be vertically transmitted to the offspring, and you will have a replacement of the population. And all these three approaches have a laboratory proof of concept, so they work. They have some trials that have been conducted. However, when you look at the large-scale implementation, the only one that has been the object of several trials, it is the Wolbachia approach. And why we don't have enough trials with this? Well, the first thing is the legislation, because in many countries you cannot release GMO mosquitoes, and the Wolbachia transfecting mosquitoes can also be considered like mosquitoes modified. Also, when you do this intervention, you need to characterize very well your population before and after the release, and this monitoring and efficiency assessment is costly, and of course you need the mass breeding of a lot of mosquitoes. The cheaper of all these techniques will be the Wolbachia, because you don't have to release mosquitoes every time, and you can rear less quantities of individuals. However, as all these techniques rely on the mating success of modified mosquitoes that should compete with the wild-type mosquitoes, we have an important gap in the behavioral knowledge of these modified mosquitoes, and this could be really a very nice tool to enhance the mosquito control programs, because we can, in principle, generate high-quality mosquitoes that can be mass released, and like that you can reduce the cost of rearing. So to sum up, I hope that I convince you that the IVM strategies have led to punctual successes in the past, but the challenges for their implementation are greater today, which makes that they are costly. We also have a modification of populations because of the use of the different vector control methods historically. They are resistant now, they have changed in behavior and in competence, but the good thing is that we have an increased knowledge on all these aspects. We have more tools, and we have identified the major gaps. So I think that if we fill these research gaps, we can perhaps use in a more intelligent manner the vector control methods, because all the vector control methods have advantage and inconvenience, so we can do a nice compilation of them. And of course, we need for this to make efforts in the quality control of the interventions, and this is an important gap, but I think we can do it, and this is important to see if we need to adapt locally the strategy, or if we can apply the strategy in different contexts. This is important, and for this, of course, we need information sharing. And today, I'm just talking about the effect of vector control measures on resistance, but we know that, in fact, agricultural use of pesticides can also impact the resistance in the vector population. So we really need to go across different disciplines and different sectors. And this is what it is proposed in the Global Vector Control Response framework that is proposed by WHO for the upcoming years. So I think it's not easy, but now we know much more things, so I think it is possible. But we need to coordinate our efforts to reduce the burden of all these diseases, because we are all concerned by them. And with this, agradecimentos para sua atenção, and thank you for your attention.
Thank you. Thank you very much, Anubis.
I was on time?
Yeah,
Okay.
just I would like first to welcome with us the Vice President of Pasteur Institute, Monsieur François Romanex, just arrived from France, and Dr. Vincent Richard, who is the Executive Director of the Association of Pasteur Network Association, and my colleague and friend Antoine Talarmin, who is the Director of Pasteur Institute of Guadeloupe. Thank you for being with us this morning. Then any question for Anubis? Comment, question? Yes? Yes, please? There's a microphone? Or?
Examples that worked in the past, you mean? You have many punctual examples, but the thing is that, as I said, we have an isolation, so we are not looking enough, I guess, of what we are doing next to us. So you have a lot of trials regarding this housing improvement, but this is very localized, and also all of this is very costly. So that's why perhaps there are many countries that want to start to do the approach, but they don't have the political will or the funds to do it, and that's what is a problem. But the physical measures in the past, yes, for the eradication, these examples I gave you for the eradication of yellow fever, they were a combination of physical measures and isolation of the patients that have fever. You have also house visits to reduce mechanically the breathing sites. So in urban context, these approaches worked, but it was a different context. You don't have the same level of urbanization that you have today. And I mean, in that time, it was even military interventions. You have full delegations that have the authority, and the people say, okay, I do this. Now it's different. We are axing our efforts in the communication, but sometimes I see this in the field in the Caribbean. Many often, the people, they look at you and they are not completely convinced that it will be important to reduce the breeding site or to cover it to avoid that the mosquitoes lay down. So yes, you have punctual examples of the modifications of the environment, but this, a good example is in Singapore, for instance. But Singapore, they have a means to do a really good strategy on integrated vector control program. So they contain the emergencies and all this, and they implement all these measures. And also they have a nice authority. So that's why you need to have everything to make your integrated vector management program work. Did I answer to your question?
Transmission indirecte pour tout le monde, dans le monde entier. Il y a tout simplement que les gens des autres pays, donc ils peuvent informer leur pays, PTV.
Okay, thank you. Another question? Yes, Antoine? Microphone, please, if you can give the microphone.
Sorry, can you repeat the question? The vector competence for what?
Direct transmission to all the world.
For dengue and not for West Nile, and do we know why this mechanism happens? And
so, it is true that the internal mosquito barriers to the infection by a given arboviruses are not well described. There are many works that have been worked with the RNAi response of the mosquito, the immunity system, but at the end, we see when you do the vector competence with different viruses that sometimes they do not overcome the mid-gut barrier. So it is not well explored. Even so I think we should improve knowledge on that. And also in the mosquito salivary gland compartment, that is an important barrier for chikungunya, for instance, in many cases. You have the virus that overcomes the midgut, but then it's blocked in the hemocell. So we're making progress by understanding the immunity. That is a very important component for the vector competence, but we don't know much about the interactions between the receptors. We still don't have very well characterized the receptor in the midgut for the binding of these viruses. So these are major gaps in the mechanistic of the understanding of the vector competence.
Okay, thank you.
Thank you.
Last question? Okay, if there is no question, thank you again, anyways.
you.
Thank you very much. Yeah, Paula told me that I should tell you that our meeting is transmitted directly by TV, the IPTV, to all of the world. Then if you want to click on YouTube, direct transmission, then then you can see our meeting in all of the world. Everywhere. Thank you. That's great. Thank you. Then we will move to next presentation, and the next presentation is 10-minute presentation with 5-minute discussion or question. And I will leave my colleague Edson to present the next speakers.
Thank you very much. Our next presentation, I have a pleasure to hear Dr. Simval from Fiocruz, Recife, to make this presentation, please. And Dr. Celina Torsi. Dr. Simval.
Good morning. On behalf of Instituto Agil Magalhães Regional Unity of Fundação Oswaldo Cruz in Recife, Pernambuco, we are very grateful for the invitation to participate of this interesting meeting and say a few words before Celina make the presentation of our work to attend the expectative of the regional community of Pernambuco to the many new challenges made by epidemics of Zika in 2015. Our unity made many articulations in this period to integrate a research group named Microcephaly Epidemic Research Group, and made many projects developed and since then to understand the epidemics, and Celina goes to presentation these efforts. Thank you.
Good morning. I want to start thanking the organizing committee and also Simval, who gave me this opportunity to come to this family meeting. So I'm very pleased to be here today, and I'm going to say some words about our research group and about how Fiocruz started or worked together in the epidemics. So what I want to say that in general, epidemics are a time of high visibility for infectious disease experts and for health authorities. Research and policymakers, we have to make decisions in time of uncertainties and high social pressure. Now I want to start showing our core team, that we were at the front line of the unprecedented microcephaly epidemic. And it was an outbreak of a known cause in the early 2015. And since the detection of the cluster of microcephaly cases in September, we understood immediately that this was a global crisis and required a global collaboration. So our Brazilian microcephaly epidemic research team, MERG, was the first network created during this public health crisis related to the cluster of microcephaly. MERG is based at Fiocruz, Pernambuco, Instituto Ageu Magalhães, that we familiarly call that Ageu, okay, which is a facility that has many labs setting and a lot of epidemiologists and social community workers. So since the beginning, we had many partners, and we had some public health institutions. I think the main public health institutions from Brazil, Europe, I would like to name London School of Hygiene and Tropical Medicine, and one of our colleagues, Dr. Laura Rodrigues. He was a collaborator since the beginning, and many people from US joined the team. So what I want to say that here I just put the pictures of the senior researchers. We mean the ones that came to work together very close in the beginning. So we want to say that during our career trajectories, we had collaborated previously in previous projects, building up a high level of trust. I think nobody tells about trust, they say about connection, but trust is very important if you have the network. We trusted each other as a scientist, as human beings. And we are, and still are, very motivated in sharing work and responsibilities up to now. So if we define research collaboration as the working together of researchers to achieve a common goal, producing new scientific knowledge for the population, I would say that MERG is indeed a research collaborating group which was built up during the emergency and continued to work hard, I would think so, after the epidemic scenario. So what I would say that's very important for us, that if I can show you, we have here some governmental institution here, like you say, the Brazilian Ministry of Health, PAHO, and the Secretary of State. So I think very early in December 2015, Fiocruz Pernambuco signed a collaboration agreement to deal with research during epidemic. And I thought this was fundamental because we had what we call the managers of the system working together with research. So I think that the Brazilian Ministry of Health and PAHO worked closely with the research teams, and they had the mission, to deal with control strategy, that's their mission, but they also had the mission to support research during this time. So if I want to put a very brief timeline, I would say if I want to ask people to raise hands who had studied Zika in Brazil before 2014, I don't think that I see Zanotto, that's a virus hunting, okay? So he knew something about virus, but I would say that for most of the scientists and for most of us in the public health sector, it's unlikely that we had ever heard about the disease, not about the virus, but about the Zika disease or about microcephaly related to the Zika disease before 2014. So in the 2014 here, there is a very interesting epidemiological finding. That's the first time that Zika becomes to be known as an epidemic period of Zika, because previous to this time, in the 70 years before, it was thought like a zoonosis with a very few spillovers to the human population, very few cases, very few interest from the publishing literature, at least we call about applied science. So if you think about the French Polynesia, that was a very important mark. It was well published by the European surveillance system. We say that in the French Polynesian, we think about 30,000 reported cases, but this is about 70% of a population of a very tiny island in the Pacific. So we still think about this epidemic. Zika disease is still a mild disease, with a lot of asymptomatic infection, but the first time there was a warning system, a warning about the Guillain-Barré syndrome following the Zika outbreak. That was an important alert sign, and we can see this alert sign in the literature. But however, in 2014, the French Polynesia outbreak did not show any evidence that Zika infection was associated with microcephaly. So up to 2014, we had no clue that Zika virus wasn't important to adverse outcomes of pregnancy. So if you think about 2015, now we think that the time when a virus hit the continent with large urban population, that was talking about the population density and the importance and the different outcomes of these outbreaks when you're thinking about large, huge populated, concentrated in areas. So that's what happened in Recife and in other capitals. They're on the coastal area of Brazil, and then we had the cluster of microcephaly. So what we can say now is that in 2015, there was this warning, there was this cluster of microcephaly. The Brazilian Ministry of Health declared the public health emergency. It was still a time that there was a lot of unbelief or kind of a friendly fire that the microcephaly was not from Zika, it was from cytomegalovirus, that people in the northeast of Brazil were not counting the case properly. There was a lot of friendly fire from everywhere, and the outbreak was even so important that with the important pieces of information, the Brazilian Ministry of Health declared this public health emergency that was followed by the WHO declaration of public health emergency with the recommendations of increasing research. So if you think about WHO declaration of public health emergency, I think the definition for a public health emergency means that the event was serious, unexpected or unusual, carries implication for public health beyond the borders, and it requires immediate international action. So I think Zika epidemics and the microcephaly epidemics fulfill all these criteria for definition as far as I'm concerned. So I thought that this picture about the former director of WHO, Dr. Margaret Chan, and followed, with Dr. Marcelo Castro, that was the Brazilian Minister of Health of the time, and Dr. Paulo Gadelha at Fiocruz, is really emblematic to show what happened at that time and the media interest. You see all the microphones on the table, so you see the huge amount of media interest because of the threat of the human reproductive population from the Americas at that time. It was well seen. For me, the interesting thing here is that for the first time, an arbovirus was considered an important risk factor for adverse pregnancy outcomes. I may be wrong, but that was the first time in my life that I saw an arboviral disease, not just with the mosquito cross like this, like to combat a mosquito, but with a pregnant woman, and warning in major airports all over the world saying that if you come or visit a country with Zika circulations, please be aware about your status as a pregnant woman, or if you want to become pregnant in the following months. So I would say that much of the fear and uncertainty around Zika epidemics comes from the potential association between Zika virus complication on infected pregnant women and risk of their babies being born with microcephaly because of the adverse effect of this virus during pregnancy that Paulo Zanotto showed, and it was proved in animal models and non-human primates, and in very interesting cohort studies in Rio de Janeiro and other parts. In fact, I think countries like Brazil, Colombia, Honduras, Ecuador, El Salvador, and Jamaica have recommended delay of pregnancy during the Zika virus outbreak. That's so important and so striking it was at that moment. Tourists were recommended to avoid areas where Zika virus circulated and even are. And now, these control measures about delaying pregnancies and now and avoiding these areas, we know that they cannot be sustained long, and they're really very transient strategies during the epidemics. Well, I want to go back because I think that we know a lot about the epidemic and the post-epidemic, but we think very little about what happens before the epidemic, before the epidemics become called an epidemic. And that's, I think, that's a very brief story that I would like to tell you, and what researchers have to cope doing in this short period here. So if we think about, in our point of view, the narrative would be, we start working very hard in August 2015, when two neurologists, they were pediatricians, the Van der Linde family, working in public health hospitals, they reported an increased number of neonates with microcephaly in the city of Recife. Does it ring a bell? I mean, it's always like this. I mean, the ones who have warning about the epidemics or from confoundings are doctors that working hard in real big attendance or in hospitals are at the front line attending people. So microcephaly had been rarely reported before the Zika epidemics. Like, it's important to say that in Recife, the capital of Pernambuco state, there is around 2 million population. It's a very densely, one of the most 5 densely populated areas in Brazil. So we were at the center of this epidemic. What we think now, and it has been described in several reports, that Zika virus found a large pool of susceptible individuals. We had abundant presence Alpha Aedes aegypti. We had adequate environmental condition. And what's very important, it was very different from French Polynesia. I think the French Polynesia population would be just one neighborhood of Recife. So at that time, it was possible, under this condition, this population condition, in this urban environment, it was possible to have a possible clinical outcomes to become apparent, very clustered. So we had a cluster of Guillain-Barré syndrome very close in time with the outbreak, and people had announced it. And 6 or 7 months later, we had a cluster, a severe cluster, a space-time cluster of microcephaly. So if I can tell you, from the epidemiological point of view, there was this obvious space-time, because we're seeing 40 cases of microcephaly, newborns in 1 week in Recife. So the past history was around 8 cases annually in a very stable during this period. So it's not, if we would say, it's such a very strong scientific evidence. So I think for us, the declaration of national public health emergency was really a turning point. It's a point that we had no more to shout that this is important. I think, I don't know what happened, I think they cut my time. Did I complete my time?
Not me.
No.
Yeah.
Something happened.
So, can I continue?
Okay, that's back.
Thank you. Thank you. Okay. So, I think that just to complete that, everywhere I go, they think what the declaration, national declaration meant for research. I think it meant a lot. For us who were the frontline, it meant that now we had a voice, we have a channel. There was a fast track for money. There was a fast track for everything. So this is so important to have it understood. That's the important part of the epidemic. It's not the epidemic. The time of the epidemic, we lost the battle. Before the epidemic is the important part to have it controlled and to have it, the knowledge to have it prevented and avoid spreading. So I'm going to just briefly say some about our first publication, because we aim...
Sorry, can you conclude, please? Because, I mean, it was 10-minute presentation and 5-minute discussion, but if you
Okay, if you want, I can stop now. I just want to say...
Just conclude it, it's possible.
1 minute,
Okay,
yeah.
so if I have 2 minutes... No, I may stop now. I just thought that I would like to say, because they said to me, I had 15-minute presentation, and I even wrote it down, because every time I talk about it, I get very carried out. But I'll be happy to stop now, or either to continue 2 or 3 slides, if you allow me.
For 2 minutes.
For 2 minutes. Okay, so if I want to just skip, I'll just say very briefly, these were the case series studies what we did. We collaborate with the genetic group. We had out a very aim to say about the piriproxyfen. There was the larvae side that we had to explore because there was a great media attention about piriproxyfen. The case control studies that the one we prove causality, and causality is very, very important when you think about epidemiological. The big challenge at that point was about the laboratory, but we were very happy to have the people from lab and the hard science with us. Lavici was our main partner. We had the Fiocruz Pernambuco holding the first meeting that was called A to Z from Zika. 3 years after, we still have a lot of attention trying to figure out what was going and showing and disseminating the ongoing projects and the consortium. Now, we have the research collaboration with Zika, that's the national consortium, with the ZIP, now it's ZIP2, that's National Institute of Health, and the Zika plan, that's a consortium with very many things. Just showing the last slide, we say that science was made very quickly. There was like an epidemic of articles in 2015, and the Latin American, the red line, just represents 80% of the publication. And here I would like to say that the top institutions that published during this period was Fiocruz, USP, and Institut Pasteur. They were among the 5 institutions. And here we have the connections, this really broad, the thick lines, showing the connections between the groups. So you have a strong Brazilian and American connection and a very strong Brazilian and European connection. I tend to say that these connections were because, first, above the equator line, we are vector-borne countries, disease countries, and from the upper part of the equator, there is the route, that the transmission route about sexual partner and travelers are very interested. So we had this very good connections made in a very short time. I won't go to the public questions, but I just want to leave you with these two images. That's why Zika was so important. That was the beginning of the pandemic. And here I want to point the gender imbalance, like say all females taking care of the babies. And even as a specialist in public health sector, here is the new image 3 years after babies from this epidemic that have been followed up. And I'll just leave this message that was for Ebola and for every virus. Thank you very much for your attention, and I apologize for being a little longer. Thank you.
Thank you very much, Dr. Celina, for this exciting work in the epicenter of Zika in Brazil. This is very, very important thoughts. Unfortunately, our time is running, and we still have 10 minutes in the end of this section for questions. Okay, thank you very much.
Thank you, merci.
And I'd like to introduce Dr. Jaime Castellan from Colombia, that will talk about the epidemic, Zika virus epidemic in Colombia, and bring out the data for us.
Hi, everyone. Firstly, I want to thank to organizing committee for this invitation. I am Jaime Castellanos from Bogotá, Colombia, from Universidad del Bosque. In the program says Universidad de Bogotá. It's not right, it's wrong. It's Universidad del Bosque. It's a private university in Bogotá. It's a small school of medicine. I am the head of the virology lab in that university. And I would sorry, I'm in Rome. I would like to present some results from my lab and some results of Ministry of Health and National Institute of Health in Colombia about the Zika epidemic and after some sequelas in that in children after that epidemic. And some activities we developed with the Ministry of Health, to help them to control the epidemic. So, you know, you don't see this map. It's an American map, and this is Colombia. This is in the northwest corner of South America. We have two oceans, contact with the two oceans, Caribbean Sea and Pacific Oceans. And also, for that reason, in that Caribbean coast and Pacific coast, there is a lot of municipalities with ecological conditions to grow, to breed the Aedes aegypti. And we have more than 1,000 municipalities, and 62% are below of 2,000 meters above sea level. And you can see there the municipalities where the Ministry of Health detect the Aedes aegypti in almost 60% of municipalities. And also, recently, also, I have been detected Aedes albopictus in 41 municipalities near the Pacific coast, in the Cauca and Valle municipalities, provinces, sorry. And regularly, we have between 100 and 200,000 dengue cases, and between the 100 and 200 fatalities each year for dengue. And this is a big concern in our country. The diagnostic activities and the surveillance activities in the country. After, we don't see. After the starting of Zika epidemics in Brazil in 2015, rapidly the virus was detected in the north coast in Colombia because we have a very, very large foreigners, Brazilian mostly, tourists in the Caribbean coast in Colombia. So we detected very early in October 2015 the Zika virus in the north coast in Colombia. This is the map after. 3 months later, all the country was with cases, confirmed cases, and after, you know, as you saw previously, in the Central America countries and Caribbean countries, and all the countries in the Central America and Caribbean, even other countries in the world. This is the map after. 15 months later, the Zika outbreak started. And very early, thanks to the warning made by the WHO, the National Institute of Health, and we collaborate with them to describe some activities of Zika virus disease in Colombia, and detected in that 15 months 100 cases and 18,000 cases in pregnant women. This is a picture showing the characteristic of that epidemics by week from October 2015 to 2017, where the cases rise until 6,000 by week cases. Some of them were detected in pregnant women, and that women were followed very, very close by the health systems. And for the same reason, the country detects, the Ministry of Health and National Institute of Health in Colombia detects very, very early the first cases of microcephaly associated with Zika virus infections in the mothers. And after, some of that mothers were confirmed by laboratory with our help. Because we also make the settings to detect by molecular biology the Zika virus and other arboviruses. It's for that reason we had at that moment very strong project detecting and of detection, diagnosis, and surveillance of dengue virus with febrile patients, and we started to detect Zika virus, and we culture the virus and titrate the virus. We are using that virus now to different studies in our lab. And also in different patients, we detect also, even in this case is very interesting because this is a nursing mother. We detected the virus, Zika virus, in serum samples, also in the breast milk, and even during 3 months after the infections. Fortunately, the children is well, and doesn't appear any infection in that children. After we described this technique, I have to be very, very extensive in this picture, because we described this easy and inexpensive molecular detection of 3 viruses in the same sample using a triplex nested PCR, a conventional PCR, and we detect in different serum samples chikungunya, for example, in this sample 01, and the Zika virus, and different kinds of virus, even different dengue serotypes, to make the following of that patient. And that patient is a pregnant woman with chikungunya, dengue, and Zika virus simultaneously. That woman was pregnant, but also the children was well after the birth. Using this strategy, we are also making some studies in dengue studies, collaborating with the Ministry of Health to establish a very strong protocol to diagnose the arbovirus, because the Ministry of Health and the National Institute of Health are focused in the surveillance, but patients and clinics and hospitals doesn't have support to different labs to get the right diagnostics. In this project is ongoing. We have 152 patients, and it's a dengue diagnostics project, but it's very, very recently patients, enrolled patients, and that patients are diagnosed as febrile syndrome or dengue, but most of them were confirmed as chikungunya positive by IgM or PCR, showing the PCR more sensitive than IgM ELISA, that PCR I showed you. And even we compared our PCR with the classic PCR, qPCR, with Lanciotti primers, and we detected that that protocol is less sensitive than our PCR to detect viruses. We don't, or almost don't detect dengue or Zika virus in that patients, in that children. So, for that reason, it's just a small slide to show you another project we performed with the Institut Pasteur Paris, with the group of Anavash Shakuntabe, where we use lymphocytes T from Zika donors with previous infection with Zika or previous infection with Zika or dengue, to detect what kind of peptides of Zika were detected with different lymphocytes, just to know the response to next vaccine Institut Pasteur in Paris are developing. And finally, I want to show you the 3 studies the Ministry of Health are doing in this moment. It's because after the Brazilian Ministry of Health awareness in the world about the microcephaly associated with Zika, the National Institute of Health in Colombia established a very, very strong surveillance protocol for microcephaly and other congenital diseases, central nervous diseases in patients. And they detected 110 microcephaly cases in that year, but 466 cases, microcephaly cases, during the 2016 year, a really increased rate of microcephaly and other congenital diseases. For that, after that surveillance in Colombia during 2 years, this active surveillance, they established a very, very well designed also with our help, a clinical form, and developed also the torch and Zika test, and also for all of them, that cases, developed the karyotype, and classified the microcephaly or nervous defects as infectious, generics, or multifactorial, and establish a following for that children. And these results are very interesting because after that following, 858 cases were included because they have full information, and they can also establish 500 cases as other etiology and 356 cases of nervous congenital defects, Zika virus associated. It means 41% of that number. It's the final number of Zika associated cases in Colombia. And in the same activities, in the same way, they established a second project. Is the best study, Vigilancia en embarazadas con Zika. It's from that mothers with showing symptoms of Zika virus during the pregnancy, were studied molecular and serology studies, and they detected 68 cases of microcephaly. It means 5.5% of all the cases. It is very interesting because they, for all of that babies, they make following by echography, laboratory test, placenta examinations, recording of the outcome of the pregnancy, and an examination during 2 years after. That data I show you, it's what the results they obtain about that 2-year following of that babies born of infected mothers. And we know the number of congenital syndrome. It's without battery. That doesn't work. And
Dr. Ram, just 1 second.
Okay, so I finished.
Thank you.
In the congenital Zika virus syndrome, they detected, obviously, microcephaly, calcification in brain, corpus callosum anomaly, ventriculomegaly, seizures, sensory hypoacusis, abnormal swallowing, and these kind of defects are very, very well known in that patients with microcephaly associated with Zika virus infection. But in a large number of Zika virus exposed children, but with that children without congenital syndrome, they detected 2 years later, 40% of that babies have ear hearing or speech development anomalies. 4.6% of hemiparesis, a very, very large number of cases of convulsive syndrome and hypoacusis. It's very interesting because it's the first data about a very close following of that children without congenital syndrome and born from confirmed mother, Zika exposed mothers. So, in conclusion, we saw a reduction of viral cases after Zika epidemic because the communication media and different activities of Ministry of Health for vector control. And now we are very concerned because chikungunya virus is circulating with a wide distribution in Colombia, and we are thinking if that virus is replacing endemic dengue virus infections, and the clinical syndrome for this arbovirus are identical, and the diagnostic approaches are still limited to detect the virus involved in each infection. Asymptomatic Zika virus infection in pregnant women could lead to neurological damage in children. This is a hypothesis. Is being confirmed. And Zika virus now is low, the transmission is low, but we expect an epidemic rebound. Thank you very much.
Thank you, Dr. Castellano, for this exciting work in Colombia and Brazil with Zika. It's almost the same epidemiology. But unfortunately, we are running out of time, and all the questions will be to the coffee break time, okay? Thank you very much.
Okay, we will move to the next speaker, and the next speaker is Claude Flamand. Claude is an epidemiologist at Pasteur Institute in French Guiana, and he is going to give a talk on assessing the burden of arboviral disease using a multiplexed serological survey in French Guiana.
It's okay? Ah, yes. First of all, I want to thank Paola Minopuyo and the whole organizing committee for giving me the opportunity to be here to talk about a multiplex serological survey conducted in French Guiana in order to understand the transmission of priority arboviral disease. So Aedes aegypti and Aedes albopictus have a history of global expansion associated with human movement and international trade, and the increasing risk of transmission of arbovirus has evidently been a consequence of increasing global expansion of Aedes mosquitoes. During the last decades, a growing number of major outbreaks of dengue associated with the co-circulation of all 4 serotypes was observed in much global, tropical, and subtropical regions. And more recently, large outbreaks, we just talked about it, of chikungunya and Zika occurred in the Caribbean and the Americas, and this was rapidly followed by yellow fever outbreaks in Brazil and Angola in 2016 and 2017. So despite this public health burden, surprisingly, a great deal remains to be done in the field of spread and maintenance of arboviruses, and frequent clinical misdiagnosis, substantial asymptomatic infection, changing in epidemiology, and serological cross-reaction between viruses from same families are very common features of these pathogens that result in a poor understanding of transmission dynamics and difficulties to accurately estimate the true burden of disease. Outbreak occurrence prediction will remain also difficult, and vector control activities are extremely difficult to target. So finally, serosurvey suffer from low specificity in the context of co-circulation of closely related pathogens. So French Guiana is a French overseas department located in the Amazonian forest complex in South America between Brazil and Suriname. And the territory is composed of 2 main inhabited geographical regions with coastal urbanized strip, regrouping 90% of population, and a most remote and river area along the Surinamese and Brazilian frontiers. So the territory is confronted to very high level of immigration, and the population is composed of various ethnic groups. And the total fertility rate has remained at 3.5 trillion per woman. That represents the highest fertility rate in the Americas. So, Aedes aegypti has been responsible of regular major dengue outbreaks in French Guiana from 91 to 2013, and we can see the different epidemics with the different circulation of the 4 serotypes, and also responsible for recent outbreaks of chikungunya and Zika virus. However, no seroprevalence data was available before these studies to evaluate the impact of past epidemics and estimate future transmission risk, investigate the surveillance system coverage, or assess the impact of prevention strategies. So we decided to implement this household survey in order to estimate the true burden of arboviruses and to study spatial distribution of viruses. So we implement a general population household survey in order to include about 2,500 residents from French Guiana between 2 years and 70 years old, according to a stratified 2-stage sample design based on the size of municipality. And data was collected according to a standardized questionnaire, including socio-demographic data, environmental information, type of housing, and also knowledge, attitude, and human behavior. So each serum sample was tested for dengue, chikungunya, Zika virus, Mayao virus, yellow fever, and West Nile, using a multiplexed microsphere-based IgG immunoassay that returns a relative fluorescence intensity. And we use also a seroneutralization test in a subsample of individuals to confirm the serological results. And finally, we applied an age-stratified modeling approach developed by our colleagues from the modeling unit, from the modeling of infectious disease unit of Institut Pasteur of Paris, sorry. And the modeling approach was used to reconstruct the history of circulation and to evaluate the extent of cross-reactivity. So we can see here the spatial distribution of including subjects. We have a total of 2,697 cases, and the number of individuals per municipality varied from about 450 individuals for the biggest municipality of Cayenne in the coastal area, and 11 cases in Saint-Élie, which is a very deep Amazonian complex municipality. So the multiplex approach allowed us to explore serological cross-reaction, and as we can see here on this graph, it was alpha virus were clearly discriminated from flaviviruses, including dengue, Zika, yellow fever, and West Nile, suggesting the existence of cross-reaction between the viruses from same families. So the multiplex approach allowed also to investigate antibodies titer levels in order to define serological status. And if we take, for example, the alpha viruses for chikungunya and Mayao virus, we investigate serological status, and if we see on the graph, negative and positive cases are here also were clearly identified for chikungunya, but we see also that Mayao response strongly impact the chikungunya response. So after that, we investigate the differences of antibodies response according to age and location data in order to estimate location-specific forces of infection modulated by pathogen-associated risk factors, socioeconomic, environmental factors. So after that, we fitted a model-based classification of the infection status in order to determine the infection status for both Mayaro and Zika virus from the serological response, and we can see the different cases for infection status for Mayaro and chikungunya. And the model helped us to understand how an infection boosts the serological response for an assay and cross-react with another assay. And it helped us to reconstruct the history of circulation using the risk factors that I mentioned. So finally, we validate the model using seroneutralization results, comparing the model-based classification with the simple cutoff classification. And we can see that, particularly for Mayaro virus, the model-based classification performs substantially better than the simple cutoff classification. And for example, the specificity was at 0.66 for Mayaro with the simple cutoff classification, and it was at 0.96 for the model-based classification. And it was also better. It was also better for chikungunya, but the difference was not so important. So after that, we could estimate the true burden of arboviruses, and for dengue seroprevalence, it was estimated at 70%, with small variation in the areas exposed to Aedes aegypti. For Zika and chikungunya virus, the seroprevalence was about 20 and 25%, with a large transmission in the most important municipality located in the coastal area, but also in the northwestern part of the territory. And so that was interesting to see that in French Guiana, the majority of the population is not immune after the outbreaks of chikungunya and Zika virus. And for Mayaro virus, the seroprevalence was estimated at about 3%, and the model was supporting a sylvatic constant transmission in the inland and Amazonian forest area, with higher infection probability for males male under 20 years old. So in conclusion, this study was interesting to determine the level of circulation of priority arboviruses at a country level in South America. And it shows the interest and the importance of the contribution of modeling techniques to assess viral circulation from detailed age-stratified seroprevalence to deal with cross-reactivity issues that are a very important issue in serosurvey in this context. And next challenges will help to investigate dengue virus serotype specific and yellow fever seroprevalence using the same approach, and to elucidate spatial differences in risk, exploring more determinants such as population mobility, entomological risk, data about insecticide resistance, etc. And the international network and in the region of Américas and the different partners that we can have here today is a very good development opportunity to elaborate new research projects and to establish partnership in order to improve the understanding of transmission dynamics in South America. So thank you for your attention, and I want to thank, of course, all the institutions that participate to our study. I mean the Institut Pasteur of French Guiana, the Mathematical Modeling Unit of Institut Pasteur of Paris, the National Reference Center of Arboviruses in Institut Pasteur in French Guiana, and all the financial partners. Thank you for your attention.
Thank you. Thank you very much, Claude, this very clear presentation. Unfortunately, there is no possible to get any question but I hope that maybe we will get some minutes to few question and discussion at the end of the section. Yeah.
Okay. Now I'd like to introduce Dr. Marcelo Lopes Lassa from Catholic University of Chile, that will talk about the dengue and the impact of mRNA translation. Please, Doctor.
Okay, so first of all, I want to thank the organizers for selecting this talk, and I want to especially thank the delegate, the Embassy of France and Brasilia that allowed me to be here. So first, I want to apologize for the title. In fact, the title is not as neat as I wanted it to be, but that's what my student came up with, and I didn't have time to check it. But anyway, so you can understand what I'm going to try to show you today. First of all, these are my students. This is Leandro. The work I'm going to be presenting is his PhD thesis. So it's work that's been recently submitted. So if any reviewer is here, please, any question, I'm willing to take. And the funding, as I said, is really important. So my lab works on translation. One of the things we do is work on translation. So we're interested in protein synthesis. And we're interested in understanding how viral messenger RNA actually takes over host protein synthesis. Because we believe, and I'm going to point this out at the beginning, at the end, that we can develop new antiviral therapies by specifically targeting viral protein synthesis. So, just to give you an overview, we study initiation, because initiation is the most regulated step in translation. So, normally a cell, a messenger RNA, will recruit the ribosomal subunit based on its 5' end, the cap. It recruits based on a complex which is formed and brings in the 40S ribosomal subunit. Yet alternatively, we have a mechanism that relies on RNA structures that is able to capture the 40S ribosomal subunit, and it's independent from the cap and normally independent from the 3' UTR and the poly A tail. So what about dengue? Is actually translation important for dengue? Yes, it is. First of all, when dengue infects cells, it modifies translation factors. And on the other hand, it's been shown that the efficiency of translation of dengue too is associated to its ability to infect and to cause disease. So actually, translation seems to be important. We were not really interested in this because we thought that everything had been said on translation. So if we look at the messenger RNA, it's a positive strand virus, so it can directly be translated. It has a cap in its 5', it lacks a poly A tail in its 3'. But anyway, it has structures in the 3' UTR and structures in the 5' UTR. So old work from Deborah Harry's lab actually showed that translation can occur in a cap-dependent fashion, that's canonical as cellular messenger RNAs, but also can occur in a cap-independent fashion. So she shows that when she inhibits cap-dependent translation, actually dengue translation stays the same while the controls are affected. So this was really interesting, and she proposed this mechanism by which she involved the 3' UTR in the recruitment of the initiation factors and the complex. Most recently, however, this is the beginning of this year, the Eckerd's lab, they published that actually there was an IRES in the Wimmer's lab published that there was an IRES, internal ribosomal entry site, that is an RNA structure that recruits the ribosome, both in dengue and in Zika. So then we said, okay, now this is our field. Now we understand, now we want to understand this process. So the first thing my student did was actually build these constructs where we have the 5' and the 3' of dengue, or we alter them with a poly A tail, with a cap or with an A cap. Now, what's important about this A cap is that this A cap is not recognized by the initiation factor complex. Therefore, it cannot recruit the 40S ribosomal subunit. And then we tested these constructs as RNA in a whole bunch of cell lines to see what came out. And I'm going to summarize this simply in the next figure, saying that, in fact, we did get activity from the A cap. However, only in some cell types. It doesn't work in insect cells, for example. So basically what this is telling us is that translation of this RNA in insect cells seems to be different of the translation of this RNA in other cell types, including human and dog and other cell types that we've tested. So then we wanted to characterize this just to see how it actually works. So to do that, we constructed a bicistronic system, including only the 5' UTR. No 3' UTR, only the 5' UTR. So we can isolate the fact that we're looking only at translation from this region and not from the cap. As a control for the cap, we use renal luciferase. As a control for IRES or internal entry, we use firefly luciferase. We made our constructs and we test this in reticulocyte, in vitro. So this is easy to control. And actually what we get activity as high as hepatitis C. So this means that actually in vitro it works. Having this system working in vitro, we asked how is the ribosome recruited? It means, is it recruited as hepatitis C directly over the initiation codon, or is it recruited upstream of the initiation codon and then it scans downstream until the AUG? So we have, like in the case of polio. So when you do these experiments with a drug called edein, which what it does, it inhibits the ability of a scanning ribosome to recognize the AUG. In the case of hepatitis C, nothing happens because it doesn't require scanning. In the case of polio, both translation of the cap-dependent and IRES-dependent go down because it requires scanning. When we do this with dengue, both go down. So basically what this is telling us is that dengue the RNA is recruiting the 40S ribosomal subunit upstream of the AUG, and then it's scanning down. So then we wanted to test this again in cell lines, but now only looking at the IRES and not the 5' UTR. And in this scenario, again, we get cell type dependency. And I just want to point out, look at these cells, HEC293T cells, which are human kidney cells. They actually do not allow IRES-dependent translation initiation. When we compare this in a cell line that does, the best one, it's a dog cell line, we can compare it with the activity of other IRESs that are well characterized. This is our control with no IRES activity. So we see that dengue has an IRES, but its activity is rather low. So then the point is that we know that translation is regulated by proteins, cell proteins. It's important because gene expression is dependent on cell proteins. So a good hint we had was PTB. Why? Because PTB had been implicated in dengue virus replication, and PTB is a known modulator of a translation. So we looked at PTB. What's the problem of PTB? That PTB is nuclear from principle. So it's a nuclear protein. How can a nuclear protein participate in translation if it's in the nucleus? So the first thing we did, here we see PTB in the nucleus of the cell. When we infect with dengue, PTB is delocalized to the cytoplasm. So PTB turned out to be a really good candidate. When we do siRNA over PTB and we look at only translation, this is in HEC293T cells, so we're not looking at IRES-dependent translation, but we're looking at cap-dependent translation. And what we see here is that translation goes down. So then what we did, PTB has 3 isoforms. So what we did, we made these isoforms, and we made them in order for them to express in the cytoplasm. So this is just a control that shows that our constructs are actually being expressed. When overexpressed, they're cytoplasmic, because we're looking at translation. And what we see with the 3 isoforms is none of them, when overexpressed, affect cell viability, nor do they affect overall global translation. However, when we look at what happens with dengue virus translation, cap-dependent translation, PTB1 tends to enhance it, PTB2 reduces it, and PTB4 also reduces it. This is important to know because cell distribution of PTB1, 2, and 3 vary, and they also vary according to different stresses. So different stresses can switch from one PTB to another PTB. So then we make a competition, and what we can see is that PTB1, that is the enhancer, overcomes PTB2, PTB4 and PTB2, when overexpressed, they inhibit, and PTB1 and PTB4, PTB1 again wins PTB4. So that means that if PTB1 is present in the system, dengue virus will translate. If PTB2 and 4 are present in the system, then it will not translate. So then we're facing this big problem, because what we're seeing is that dengue virus RNA has different ways of translating. And they're depending on RNA structures. So we asked the question, is there a riboswitch implied in this? Why? Because dengue virus actually requires ER stress. And ER stress is known to reduce cap-dependent translation. So it makes no sense that during ER stress, translation will work because it will be shut down. So why would dengue induce ER stress? So we said, okay, if there is a switch or a riboswitch, we should be able to turn the IRES on in a system where it doesn't work, that it's HEC293 cells. So Leandro, what he did was plan this experiment where we use a protease from rhinovirus that cleaves 4G. This is the initiation complex. 4E is the cap-binding protein. So in the presence of 2A, 4G is cleaved, therefore cap-dependent translation is inhibited. We see this in our first cistron, cap-dependent translation is inhibited in each case in the presence of a wild-type 2A. When we express a mutant 2A, however, and this mutant 2A lacks the enzymatic, it has a mutation in the enzymatic site, so the protein is there, but it does not cleave. What we see is no effect on translation. But what happens with the dengue virus IRES now, or the dengue virus RNA? It doesn't work in 293 cells in a cap-dependent fashion. It does, but it doesn't as an IRES. When we shut down cap-dependent translation, we turn it on. So apparently there is a riboswitch that is regulating dengue virus translation. So these are basically what I've shown you today. So these are main conclusions, but I don't want to stop there. So you can look at them just for a while. And this is the real message I want to give. So basically what we've been doing for other retroviruses and flaviviruses and Bunja viruses is try to define or identify the cellular proteins that participate in translation. Because normally these cellular proteins do not play a role in cellular translation, only in viral translation. So, and what is interesting, most of them have post-translational modifications. And we've been able to show that by modifying post-translational modifications, we're able to specifically inhibit viral mRNA translation. So in this paper, we show that we're able to inhibit HIV, HTLV, and MMTV translation from these RNAs by affecting a modifier, and AIDS, lymphoma, cancer, mammary tumors. So we show that by inhibiting HIV IRES, by using a compound that inhibits the enzyme that will modify the protein, we reduce IRES activity, but what we also do is reduce viral expression or virus replication. So this is an infectious clone, and we're able to reduce viral replication. So basically what I'm saying is that by knowing how these cellular proteins are modifying or assisting on dengue virus mRNA translation, we hope that we'll be able to do the same thing with dengue and other viruses such as Zika, or as we've done with hepatitis C, to specifically inhibit translation. So thank you very much for your talk, for your patience. I hope I didn't go that far. And this work was done in Chile, so I finished with this. So you can see Chile if you want to visit it. We just had an eclipse yesterday, so it was really nice. So did I make it? I did. Thank you.
Thank you very much, Dr. Marcelo.
I also thought I had 15 minutes.
Yes, you are just in time.
Wow.
Yeah, that's good. Great. Well, we have 6 minutes for questions, and now is open for all of our speakers. Please take our time in making the question. Where is the microphone?
Any question for the last 3 speakers? Claude, to the last 4 speakers. Yeah, there is one question there. Anubis, please, yeah.
Yes, I have a question for Jaime. What do you think about the possible role of Albopictus in Zika transmission in Colombia, in the place where this species is settled? What do you think about that?
We don't know yet because we detected a mosquito, but we have
Yeah. Can you please speak on microphone or come to the here? Yeah.
Okay. Yes, we don't know about if now Aedes albopictus is transmitting dengue or Zika or chikungunya yet. We have a very large work with different mosquitoes. We are detecting the RNA for the 3 viruses, but we don't found yet the viruses in that mosquitoes.
And do you have any going on with the local populations of albopictus and Zika?
No. We don't have the animal facility to get the mosquitoes. It's just experiments with mosquitoes caught in the wild, mosquitoes wild.
Thank you.
Okay, thank you. Another question? Yes, please. Up there, there is a question. Yeah, up there.
Well, good morning. I have a question for Claude, right?
Claude, yeah.
Because what we have seen in Brazil and also in Colombia is that the frequency of detection of Zika virus infection sharply decreased after a couple of years of circulation. And that is usually interpreted as most of naive population was infected, right? And we should expect some years so new susceptible hosts to be available for Zika virus establish a new outbreak. But you show your estimate that about 20% of the population in French Guiana was infected during the first year. So you have still 80% of the population susceptible, in theory. You have seen a similar decline in the frequency of Zika virus infection, or in French Guiana, Zika virus continue to circulate at a high frequency?
Yes, we have seen a very important decline, and now, since I think more than 1 year, we don't have any confirmation of Zika virus cases.
So you have any hypothesis of why this is happening if you have a high % of the population that is still susceptible?
Tell me again?
Yeah, do you have some hypothesis of why this declining happened if you still have 80% of the population susceptible?
Okay, I think it can have a lot of factors that can play a role on the decline after only 20%, but in French Guiana, we think that the distance between population area are very, the distance are very important, and I think that it limits the dynamic of transmission comparing to, for example, outbreak in island context, for example, where the density is quite homogeneous. In French Guiana is more complicated, and with chikungunya and Zika virus, we have the same kind of seroprevalence rate. But before this study, we don't have any idea about the impact of an emergent outbreaks. We have about, for all outbreaks, dengue, chikungunya, Zika, we have about between 2 and 5% of clinical cases, but it's just now that we have an idea of the attack rate of an outbreak, and probably is more, it's less important that in over island area. And in Suriname, they have also make just an evaluation in emergency department, and it's quite coherent. Also, they have about 30% in emergency. It's not general population, but it's quite similar with, and probably is the repartition of population in the territory with very deep Amazonian forest in an important part of the country that probably limits the transmission. But we have questions that we will continue to explore because we didn't expect this level of circulation.
Thanks.
Okay, we will take the last question here. After that, we have a coffee break. Then during the coffee break, you can discuss all together and exchange information, question, and discussion on. Yes, please.
Just a very brief comment about your data from French Guiana. See if I understood it correctly. When you place 70% of prevalence for dengue, you're talking about the past exposure of this population.
Yes.
That may be many, many years.
Yes.
When you think about 20% of prevalence in a population for Zika, maybe you'll be talking about 3 or 4 years after the Zika introduction. So it makes the whole difference when you're thinking about prevalence. You're thinking the time of viral exposure in that previous population. But that's my comment. But only I was wondering about the test. I mean, how are you coping with the Zika and dengue-related laboratory tests, and how you're making to disentangle the Zika and dengue exposure?
Okay, thank you for this question. So we used seroneutralization test to evaluate the cross-correlation between Zika and dengue virus. But we have made a lot of seroneutralization for Zika virus, but not for dengue for the moment. And we think that we under-evaluated the seroprevalence of dengue. But we have different, the epidemiology of dengue has completely evolved since the introduction of chikungunya. And since 2013, we didn't have a confirmation and important transmission of dengue. Except now we have just 2 very limited clusters, but it's very exceptional now for the situation of dengue. And with the important fertility rate, too, there are a lot of young individuals that are not already exposed. That can explain the level, but we think that it's maybe under-evaluated, and we will apply over things.
I am very sorry to say that. Our meeting is very solid. There is a program that is, we have to be in time. We have a coffee break that is very short, in 15 minutes. We have to come back for the next session. So I just have one thing to say, un hommage à une personne très importante de l'Institut Pasteur. The coffee break is served. She is called Nicole Bachala.
Okay, thank you for coming back. We are going to begin, but first there is an announcement for a virological meeting, and maybe you can...
So, our colleagues from Universidad del Bosque from Bogotá, Colombia, they asked us to announce the Latin American meeting in virology. They are still receiving abstracts and submission till July 15. The meeting will be held from the 3rd to the 5th of October this year, next year, sorry.
And as you can see, we'll call the first speaker.
Yeah, so I would like to, first of all, to congratulate the organizers, Paola, well done, excellent meeting. And I would like to introduce Dr. Carlos Robelo. Carlos Robelo is a very old-time friend of us from Uruguay. He's a biochemist. He did a PhD in the University of Granada in Spain, and then he returned to Uruguay, fully supporting the recovery of Uruguay from a science point of view. He has a long tradition and a long work on Trypanosoma cruzi, and he is welcome to talk about the biology of this parasite.
Okay, thank you very much. First, I am very pleased to be here, and congratulations because of this new USP Pasteur platform. And thank you very much for the invitation. Congratulations, Paola. What I doing here, talking about Trypanosoma cruzi, when most of the people was talking about virus. An important point is that in any case, T. cruzi is a re-emergent disease. As you can see here, due to globalization, migration, etc., the distribution of this parasite is not in America, but also in North America, Europe, Asia, etc., and also Africa. Okay? So, in the definition of emerging infectious diseases, it can be considered as a re-emerging disease. And I want to talk about the importance of genomic approaches in studies to reanalyze some of these complex diseases provoked by a parasite that is a protozoan parasite. So it is very complex. It interacts through different proteins, surface proteins, etc. The year 2005 was a landmark in the study of biology of trypanosomiasis because of the publication of the 3 trips. The 3 trips are Trypanosoma brucei, the African trypanosome, Trypanosoma cruzi, the American trypanosome, and Leishmania. But in the case of T. cruzi, the genome is highly repetitive, chromosome pairs are different, and some strains are hybrids. So the less complete genome in this number of science was the genome of T. cruzi. This is a view of the complexity of the parasite. There are some groups, different groups. Some of them are heterozygous hybrids, like TC6, and the C.L. Brenner strain was sequenced here. So in that paper, authors showed that the genome of T. cruzi was expanded because of the multi-gene families, transialidase, MASP, mucins, etc. As you can see, there are more than 1,000 copies of these genes in these parasites. That means that was defined as a genome in expansion because of the multi-gene families. Most of them, if not all, are surface proteins, code for surface proteins, so are related directly with interaction with host. I will come back with this in a few minutes. But authors of this paper in Science said the copy number of these genes is likely underestimated because of the collapse of multiple tandem repeats into fewer copies during assembly, as evidenced by regions of locally high sequence coverage. This is because the repetitive nature of the genome will collapse by the Sanger method of sequencing. What we did a few years ago was to use the long-read technology PacBio, and I will comment some results of this paper that we entitled Expanding an Expanding Genome. Expanding because through PacBio sequencing, we could see the real dimension of that expansion because of the technology in comparison with Sanger. What we selected, we chose two groups of strains, TC1 and TC6. Since TC6 C.L. Brenner is hybrid and was sequenced, we used another strain that it is almost identical to C.L. Brenner. Here you can see the distribution of reads. Remember that when you sequence by Sanger, the classic sequencing method, your reads are of 600, 800. Here you can see that this is 10,000, 20,000, etc. So we could pass through the repetitive elements, pass through the multi-gene families, and for example, we could determine that the genome that was estimated in 50 megabases, now we can say that is almost 86 megabases. This is a consequence of the long read sequencing. Here there is a chromosome of the C.L. Brenner sequenced in 2005, and here is one of our chromosomes, yes, sequenced by PacBio. So, for example, these nucleotides were named N because could not be assigned, and now by this method we could assign them. This is a collapsed region. Here this region is, I don't remember, but imagine 10 kb, and now we can see that there are several tandem repeats, yes, and that is the expansion that you could make by PacBio. Here is another example. So long read sequencing allowed massive improvement of genome assembly. Also, we could separate haplotypes. That is important because you can compare now homologous chromosomes. So this is another important point. And when we mapped Illumina reads, I don't know if you can see them, but when we mapped Illumina reads on the haplotypes, you can see here that the reads are in this one and go to this to the second one, to the homologous. This means that there is an area of recombination here. So recombination can be also studied by this method. This is just to show that when we compare the sequence with itself, these are the repeats and how we can pass through the repeats. We did a web interface where you can see all of the genes. In green are the typical classical genes. In red and blue, orange, red and blue are the multi-gene families, transialidase, MASP, and mucins. And what we see is that the genome of T. cruzi is compartmentalized. We defined a core compartment with the conserved genes, this one, and a disruptive compartment with transialidase, MASP, and mucins. So genes are divided in the genome. They have differences in the GC content. They have properties, very different properties. Here you can see the core compartment and the GC content, and here the disruptive compartment and the GC content, and you can see the differences. And these members of the disruptive compartment that we are referred as subtelomeric genes previously, now we can see they are not subtelomerics, but they can occupy for an entire chromosome, or they can be in the same chromosome with the core compartment. Well, we redefined the number, the more precise number of the multi-gene families. We could detect that the only retroelement that is complete is this L1TC, and we found that it is a very useful molecular clock for evolutionary studies in T. cruzi. In this case, we found that there are 3 major clades instead of 6 or 7 as were previously classified. More recently, we analyzed the genome of another strain, that is Berenice strain, that is the first Carlos Chagas patient here in Minas Gerais. And this strain that was from the Raju Institute was sequenced by another technology that was nanopore, that is very cheap, cheaper than PacBio, and you can do it on your desk or on your lab because it's very simple. And we compared here the assembly of Illumina or Illumina plus nanopore, and you can see here the context of the combined methodologies, and you could get a very good genome of Berenice that belongs to another group. We did new phylogenetic trees with the new genomes, and we found that in fact, there were 3 groups, 3 clades, instead of 6, 7, or 8, as was previously proposed. Finally, when we analyze the maxi circle, if you go to the gene bank, you will find that this is the complete maxi circle. It is annotated as a complete maxi circle in T cruci with 22 kb, yes, in length. And we could find that there is a repeat that is longer and could be expanded by the new technologies. And this is our new representation that is about, instead of 22 kb, it is about 50 kb, and here is the big repeat, and this is a circle representation where you can see the repeats, the tandemly distributed in the maxi circle, okay? When we compare, when we compare the different strains of T cruci, we found in the maxi circle that represents the mitochondrial genome, we found that there are 3 kinds of maxi circle. Again, we have 3 kinds of T crucis, A, B, that we name A, B, and C. And the phylogenetic tree of the sequences of maxi circles show us that there are 3 maybe species of T cruci. So, final remarks. The genome of T cruci has two compartments, a core and a disruptive compartment with different architectural properties. The disruptive compartment is composed mainly with genes related with interaction with vertebrate and invertebrate hosts. So that is a source of analysis in the host pathogen or host vector interactions. Complete genome sequences of all the members of these multi-gene families could give us new elements for new developments in immunodiagnostic and immunotherapy, for example, because now we know all of the genes of the surface. The precise number of genes also allow to better study, to make knockout experiments to determine which genes are really essential by CRISPR experiments, and hence develop to define new targets for drug development. And this is a general idea for not only for T cruci, but for more complex genomes. Diversity of pathogens can now be revisited through nanopore technology that is very cheap in comparison with the other or another technologies. And an example was this T cruci classification. Maybe we can classify T cruci or rediscuss now if they should be divided in discrete typing units or in clade species A, B, and C. So probably we'll need to deconstruct some paradigms and restart with these technologies, at least in the more complex genomes, in those organisms with more complex genomes, yes? So we will need emerging paradigms. So this is our lab, the group of our lab, that is the Laboratory of Host Pathogen Interactions. Also, people from the Section Biomathematics and Microbial Genomics Laboratory worked in here, and this is the support. Okay, thank you.
Sorry, we have time for one question.
I have one. Do you always associate PacBio or nanopore with NGS because of the mistakes?
Yes. I forgot to say that, but in all of the genomic studies, we associated Illumina with PacBio or nanopore. Other way, because of the errors, yes.
Thank you, Carlos.
Okay, we now welcome a friend of mine. I've known him since well, we don't say when, a long time ago. And so he has begun in the Pasteur network in French Guiana more than 20 years ago. Then he went to Niger, I think, and then Madagascar, and for a time he left the network to go in Australia, and now he's back in French Guiana. Yes, Jean-Bernard Duchemin, we will talk about the future of vector control.
Thanks very much. So after the
15 minutes.
Sorry. After the emerging arbovirus or disease, I think we have to go to emerging vector control techniques, so that maybe, not maybe, that will be the topics of my talk today. So quickly, you know roughly this picture by the talk of Anubis. It's not the same orientation, but that's exactly the same phenomena in French Guiana as in Guadeloupe. Aedes aegypti has presented several sets of resistance according to each use of insecticides class of family. So some number of notion here. Clearly, you know there's a lack of drugs, or at least very low number for drugs and vaccine for most of vector-borne pathogens, with the exception of yellow fever. And there's some limits for chemical methods for vector control. So development of resistance, you understood it. The restriction of available products for use in public health by regulation, which could be very strong. Here in French Guiana, we are in Europe, sorry. But so we have such a heavy burden of regulation. Having said that, regulations are due to impact on health and environment, and acute toxicity or chronic toxicity, and even the potential question of endocrine disruptors. So there's a big need of exploring our new way of vector control. So one way could be to try to enhance the race and try to have a step ahead of the mosquitoes and develop new products. So we develop a system of a cellular system for differentiated neurons of mosquito, and we found some interesting. Sorry, where is it? So examples there's a Timephos and so on. We find neurophysiological signal which show toxicity of neurons here. But we all agree that's just a race, and that just going the same walk than the insecticide use. So I will present you several concepts and principles that are used in this new area for vector control. The first one is clearly the two main goals or two main targets could be either the propagation of sterility by the technique which is called SIT, sterile insect technique, or make the mosquito unable to transmit. That means pathogen interference, and as said Antoine some time ago, try to modify the vector competence. So all these techniques are all based on mosquito release, and I would say massive mosquito release. This technique could be either self-restricting or self-propagating, and that last character could be questionable. And this one use now the technique of gene drive. I will detail a little bit further. So all this technique based on mosquito release, first they are very specific, and no one of any tools have reached that level of specificity for vector control. That means that there's a reduction of collateral damage on environment and health, but that means for each vector species you can have in a transmission complex cycle has to need a specific tools for each species. The efficacy obviously depends on the competition of released mosquitoes with wild population, and if the density of the mosquitoes is low, it will be better for the technique. There are some potential problems there. Either the persistence of the released mosquitoes in the environment, the potential release of female which could bite, with or not intention and something else. And the big things too is that all this technique needs a delay. That means the massive release will need a time to act and to have an impact, and that could be several weeks or several months. So that means we have to still have technique in use in context of emergency, like insecticide, for example. And finally, there's a lot of technical and logistical constraints specific to the mass rearing and the release across a city or urban area or village, etc. So for the sterile male, once again, that technique is very specific. The mosquito female mate only once. That means that if a sterile male mate with a female, the female is for her whole life, won't get any progeny. And you remember that the female only do bite, so the released male is are not harmful. This technique has been historically very successful. So there's many examples. The best one is probably the screwworm flies, which is a pest for cattle industry. And this technique of irradiated sterile male for these flies have led to eradication of that flies in some countries of Latin America and Ireland, and it was used for U.S. agriculture to protect the invasion of these flies. Among different methods, the currently most accepted one now is irradiation, where the protocol of irradiation and procedure are relatively well defined, at least for mosquitoes, for Aedes albopictus, for example, in La Réunion Island. Another concept too is a transboundary transport. All these techniques are very, I would say, relatively technological. Need a technology which could be used at a regional level on if the countries allow the transboundary transport of such irradiated mosquitoes. So one slide about the concept of gene drive. So clearly gene drive is resumed here. It's a system where a gene will be driven through the generation, not in a Mendelian mode like that, but on a much higher efficacy for transmissions. That means that the two copies of the chromosome will be transmitted at each generation. So you see at the end, in a few number of generations, you could have a full invasion or integration of a given gene in the population. So the Wolbachia, you have heard of Wolbachia. Wolbachia is an intracellular bacteria, very frequently found in arthropods. We consider that about 60% of species do harbor Wolbachia. There's two main characteristics for that. One is called cytoplasmic incompatibility, and that means that if the two male and female are harboring bacteria, the progeny will have bacteria, that's logical. If the female only harbor bacteria then, and the male has not, the progeny will be infected by Wolbachia, and if the male only have Wolbachia and the female not, then there will be no progeny. So you can imagine that configuration is clearly the same configuration that I described for sterile male, where indeed the male are not clearly sterile, but it's sterilizing. Okay? And the second characteristic of Wolbachia infection is what's called the pathogen interference. That means there's a modification of mosquito vector competence. Here, for example, you get an example with Aedes aegypti and Zika virus, where the control show a load of virus about 10 to 5 TCID50 in the saliva, whereas the Wolbachia infected have 2 or 3 order of magnitudes lower from the control. So clearly, it's really important, and that's the big success currently of Wolbachia is that big effect. It has been found for many viruses. The big problem for that is that we don't really understand what are the mechanisms for that. It could be competitions for lipids between the Wolbachia and the pathogen, but the things are not completely well defined for that. So another example are genetically modified mosquitoes. Here there's several examples. I have taken only 2. Here, for example, it's the insertion of an antibody, which is a PAN serotype against active against the 4 serotypes of dengue virus. And then you see for the 4, you've got the control here, the same with the load in the saliva. And if you get the homozygous, that's completely, sorry. There's no virus in the saliva for homozygotes, and for heterozygotes, you've got a level which is much lower than the control too. When you see for dengue 2, for example, there have been comparison with Wolbachia, and heterozygotes, they are roughly the same level of reduction compared to the control. Another construct here with against Zika virus with a series of insertion of microRNA, which are targeted against several regions of the Zika virus genome, and you see the same effect that with the control, the homozygotes have a neat, clear cut on the virus production in the saliva, and the heterozygotes are roughly the same as Wolbachia here. So that's the idea. You understand that the major benefit of being homozygotes for a gene, for a transformed gene, is really crucial. That push through to that technology of gene drive now. Sorry. So, not going too much in detail, but roughly the big interest of a gene drive through CRISPR-Cas9 technique is that the guided RNA portion of the construct will really define a very precise region of the genome. Here, for example, you've got, it's for Anopheles for malaria vectors. You've got an antibody directed against the parasite. You've got here, it's a fluorescent marker, which is important to really tag every transformed mosquito. And then you've got the Cas9 part here, and with several promoters active either for the fluo or for the payload part of the genome. So the big question, that's a very, how to say that? That's a technology now which is really efficient in the lab, and there's many experiments in the lab which show that it works. The big question is that there's a lot of questioning upon the evaluation of such a system in the field, either for a problem of speciation between vectors, for example, or genome organization, and as it has been shown in some case of natural gene drives. The risk, the potential risk...
1 minute only.
Combien? Une?
1.
Okay. Well, you can read that. So, oh, sorry. No, I mean, so it's really, in conclusion, it's really premature to consider implementation of the gene in the field, and because of a lot of ethical issues. I encourage anyone interested in that field to read the report from the National Academy of Science and Engineering of USA. Really interesting one. You can find the website here for finding it. There's a lot of questioning for that. The main outcome of this report is says that there's a need for the researcher to be responsible, which sounds good. There's a need too of a lot of public engagement, and especially the one which are targeted for action in the site. So what could be the place of the Institut Pasteur for that? I guess the preparedness for testing on all the lab works upstream could be probably performed in some lab. And the first pre-release tests, there's probably a lot of space for Pasteur Institute and associated institute to work with. Again, the science communication is really important in that field, and the strong engagement, again, with communities, and that could be an interesting goal for our teams. Thank you.
Thank you, Jean-Bernard. Unfortunately, we don't have time for questions.
So now we would like to introduce Josine from the Institut AMSUD-RIIP. She is doing a very nice work as a master degree candidate, working with dysbiosis induction to reduce virus infection efficacy. So welcome, and you have 15 minutes.
Here we go. Thank you for this opportunity. For me to present the work I'm doing and working on for my master project back in Canada. That also use the help of Institut Pasteur of Guyane Française and of Guadeloupe. So my master project is bacterial microbiota recruitment by Aedes aegypti larvae in French Guiana. So we know that a lot of functions are provided by bacteria that we found with insects. For mosquitoes, it can play a role with blood digestion, the digestion of nutrients, and also in also in oviposition, and for what is interesting for me, larval development. We also know that for some other insects, the microbiota can help with insecticide resistance, and it's something that has not been observed yet with mosquitoes, but cannot be pushed aside. We also know that during the development of mosquitoes' life, there is some genera that are conserved from larvae to adult, even though there is a flush of the microbiota content at the pupal stage. We don't know a lot about what explains this recruitment and how this selection is done. So in my project, we are looking at the diversity and the recruitment of bacteria for the larvae in the water environment. And we have some index that we use to observe these. So we can have the alpha diversity, which is the number of species that we found in one habitat. So as example, for the water bucket, we have 3 species, and we also have only 2 for the larvae environment. So this is the example of alpha diversity, but we can also use the beta diversity to explain and compare those 2 habitats. So we can use the presence-absence. So as you can see, the pink bacteria are the only ones that are only found in the water environment. But we can also use the relative abundance of bacteria. For example, the yellow bacteria that are much abundant in the larvae environment than in the water. So these information can indicate us the bacterial recruitment, which is all the species that are acquired from the environment by the larvae. So our study hypothesized that the structures of bacterial microbiota that we found in the larvae results from the structures of bacterial content that we have in the breeding site's water. And to assess this hypothesis, we did a sample collection in French Guiana, where we choose 69 sites in which we collect water and the larvae. Also, a bunch of pictures were taken from each site to characterize the environment, so the specific era around the sites. We also create 3 categories. So the first one is the macro parameters, which involve the geolocality information, so where the sites was collected. We also have the meso parameters, which is everything that has a link with the vegetation around, the habitat, landscaping, and so on. And we also have the micro parameters, which is on one side the biotics parameters, so the microbiota that we found in larvae and also in the water content, and the physical-chemical analysis in the water. So we have a bunch of parameters that were assessed, like pH, turbidity, and nutrients in the water. So these are parts of the micro parameters. So what was done after the sample collection in Guadeloupe, the DNA extraction were done with the help of Dr. Anubis' help. Back in Quebec, the library were prepared and sequenced, and we use the ISV, which is not the OTU, but exent sequence variant. So it's 100% OTU, and we did the biostatistical analysis after. So just for a kind of example, we have here different sites and pictures that were taken, and my job here was to relate and note everything that was going on in the environment. So like the walls, there is absence of walls or clothing, or here it's really difficult to circulate, so these are some of the parameters that were characterized. So the first result that we have to show you is that among the 1,662 ISV that we found, we have some ISV that are largely distributed in the breeding sites and less in the larvae environment. So this is the case for Novosphingobium, but we also have some genera that are much observed in larvae that there is in the breeding sites, like Tabrisicola. So this can also imply that there is some structures that are different. So the beta diversity is not the same here for most of our genera that we found. We also found some genera that were exclusively found in the water. So it's about 11 genera that were only found in the water and were not found in the larvae, which can suggest a specific recruitment. And we also have 3 genera that were exclusively found in the larvae. So we hypothesize that it might be some vertical transmission, since we know that some case has been found in the literature, but it can also be some of bias, meaning that these genera were so in such a little quantity that they were not detected by the methods. So that's something we have to keep in mind. So to continue to the characterization of the difference between the water and the larvae from the same site, we use Euclidean distance. So briefly, the Euclidean distance measures the diversity and the variability between samples. And what we did here was to compare the larvae and the water from the breeding sites that come from the same site. So Euclidean distance that tends to 0 implies that the larvae and the water are the same. As we can see here, our values range from 30 to 60, which means that the structures are not the same in water and in larvae. So it's not the only thing. We cannot say that larvae's microbiota comes all along from the water's microbiota. We can also see that we have such a kind of a gradient, since some of our sites are more closer to each other and some of ours are more diverse. So to continue to explain, we use our first parameters, which was the micro parameters, to explain what's going on with our ISV found in the water. So as we can see here, it's a canonical analysis. We use the ISV that we found for the larvae, and we put them on a matrix. So we can see some of them are clustering together, which means that they have some similarities in the bacteria that were found in those larvae. Then we had the bacteria that were found in the water, and we tried to use some ISV to explain what we observed. So we can see here, for an example, that the ISV number 6 is maybe what is differentiating those larvae apart. However, the water content only explains 55% of the variability that we found in larvae, which suggests that there is some other parameters that is influencing the microbiota found in larvae. So that's why we are looking at other parameters. So the next step will be to add the physicochemical parameters to see if they have an effect on what we see as a variability in the larvae. And we will also add like the site collection, localization, so the macro and macro parameters that I was talking about at the beginning. So this was about what I have to say. So I would like to thank my labs for support and you for your attention, and I will take your questions. I was fast.
We have time for questions.
What is known of the mosquito microbiota, or the fungal microbiota, and did you consider adding that to your study?
The core microbiota?
The fungal, mushrooms, fungal.
Oh, yeah, well, we tried. It was part of the, sorry, it was part of the studies. So we also tried to add the fungal, so the ETS sequencing. However, the only way we had to manage that was to do nested PCR. So we decided to put it along and not continue with that, since there is some bias with the nested PCR. So we really focus on the bacterial communities. Yeah. Well, it's something that we will continue on. So this is my master project. But I also went to French Guiana a few months ago, and we did actually modulate the microbiota. So we used breeding sites that were natural, and we diluted to see if the dilution has an effect on the recruitment. So less bacteria, is it going to mean less bacteria in the larvae? And to see what's going to happen. And to continue with resistance of insecticide, we also use these bacteria that will grow in different concentration of water, and we expose them to deltamethrin. And we collect the 31st dead and the 30 last dead larvae to compare if there's a change when we expose to deltamethrin. So that's what is coming along, but it's not done for the moment since it was. Other question?
Well, one question. Yeah. Is there any other question?
Yeah, I've got one. Do you intend to test also the microbiota of the adult female to see if the complement of the bacteria come from the mother?
Oh, the vertical transmission?
Yeah.
This is not something I will intend to do since we are really working on larvae, but it's something that has been shown it can happen. It's not that large of an event. So vertical transmission is not that frequent.
So how do you explain the bacteria then?
They're coming from the water?
Yeah, but not all of them.
Most of them. So, well, that's some of the stuff we have to figure out. So the genera that we only found in larvae, it might be some vertical transmission, and this is something we will have to push on, but that's not something that we have done yet.
Thank you. Okay, thank you. Okay, we now welcome Milton Osorio Moraes, who is a doctor in molecular and cellular biology, and he's working at the Fiocruz, and we talk about a very important topic which changed from our viruses, antimicrobial resistance.
So, thank you. Oh, sorry. Good morning, everyone. It's a great pleasure to be here. I'd like to thank Paula for the invitation and Savino for suggesting my name to be here today. I've been talking about, for the next 10 to 15 minutes, I guess, about antimicrobial resistance. It's indeed a very important topic. And I need to tell you that I'm wearing two hats here. So I'm head of leprosy laboratory at Fiocruz, but I'm also coordinating the innovation program in the institution. So I will try to, in the beginning of the presentation, show a little bit what we are doing in the lab concerning leprosy, specifically, and some other genomic techniques. And then I'm gonna move towards what we are trying to do as an institution to actually face and understand a little better antimicrobial resistance. So this is our outpatient unit in our laboratory. We are accredited internationally by JCI. And we are a national reference center, and we are seeing patients every day and also their family members. So we are trying to do surveillance considering antimicrobial resistance for the drugs that are being used to treat leprosy. And obviously some of the drugs are the same that we are using for TB, especially rifampicin, and we have to be very careful with the circulation with those strains, the resistant strains, because it is the most microbicidal drug for the treatment, right? This is one patient that I'd like to show you. This is one patient that we had a few years ago. We actually detected the resistance in him, and thankfully, after an alternative treatment regimen, he was completely cured. We can see the swollen hands here. We can see the back, which is a lot of lesions, and now he's completely cleared. But the reason I'm showing this patient specifically is because actually we had almost 40 years, more than 40 years of treatment, of different regimens for that specifically patient. So it's important to mention that antimicrobial resistance is a multidisciplinary approach. We need primary care, we need surveillance, we need better drugs, we need vaccines, we need everything, because otherwise it's very difficult to identify and to treat. So just briefly mentioning, he started in 1971 only with dapsone monotherapy. It was a regular treatment from all of the time, and after dropped the first, abandoned the treatment in a few months after dapsone treatment, he started a few years later treatment with rifampicin, abandoned again, and it happened more 2 times, and then we actually detected the resistance in 2015. So it was more than 30 years before the first treatment until the detection of the resistance, and probably the emergence of the resistance was around in 2000, late 90s or in the beginning of 2000. So it's a gap of maybe 10 years around that. It's important to mention that we did whole genome sequencing for that specific strain to test whether there was some difference in this in the resistance, but it was basically rifampicin resistance. So this is one of the things that I would like to point out that's important, and you probably know this graphs. It was produced by a commissioned paper by the Wellcome Trust a few years ago. Jim O'Neill was the head of this, and basically what he showed was that maybe if we not care enough for new drugs and new diagnoses for antimicrobial resistance, we will probably reach 10 million deaths in 2050, which probably will be more than cancer. And the impact in GDP loss will be around $100 trillion. So it's really a huge impact, economic impact for antimicrobial resistance. And in 2015, and recently here in Brazil, we had a global action plan for WHO, and the Brazilian action plan last year was published in the end of the year, around November, December. And the most important issues are here, and I'd like to actually highlight a few of them. Mainly that obviously we need a lot of public engagement, communication, and education. We need a better development and information systems to monitor and do surveillance. We need to strengthen regulation and monitoring and for antimicrobial use, and also to improve and broaden the knowledge, fostering research projects for new antimicrobial drugs. And mechanisms, understanding better how bacteria and different, and also different pathogens actually are acquiring and developing resistance. So these are the most important issues. And as you know, I will talk later today, and I will also mention this, but it's important that here we in Fiocruz are well, Selena mentioned earlier that we are driven by response in the public health emergency. And well, actually for more than 100 years now, we are coping with different kinds of infectious diseases. And so we did for, and we have more than 12,000 people now. I don't know if you are aware of what Fiocruz are. We have 2 hospitals, around 1,200 PhDs, 3 production plants for vaccines, biologicals, and pharmaceuticals. And we have reference centers for surveillance. It's very important to mention. So what do I think that we can do for, as an initiative for antimicrobial resistance? So we have basic research, and so we need a better understanding of the mechanisms of antimicrobial resistance. We need surveillance. This is very, very important for hospitals and also for environment. We need clinical research to test new drugs and pharmaceuticals, and we need development. We need vaccines. We need diagnostic kits. So this is what we are trying to do. This here, obviously, I'm sorry I didn't mention, but it's very important that we have a cross-cutting edge here on education and communication and information that obviously bypass all of these other areas, right? So it's important to mention that we are now doing research on biocides and resistance in aquatic environments. We are doing surveillance in veterinary drugs, either in food samples and environmental samples. We are doing molecular studies, epidemiological studies on antimicrobial resistance, and so on. It's important to mention here that we're also developing new adjuvants and diagnostic kits and vaccines as well. Running few clinical trials and discussing with GARDP and some other stakeholders whether we should start new trials in these areas. And we are doing, we are prospecting using biological collections that we have in different institutes of research in Rio and other institutes throughout the country. So it's also important, and I will go a little deeper in that in the afternoon, but we just launched a program called Innovation Program in Fiocruz, and we have around 35 million reais in funding, and we approved around 250 proposals, and we have a portfolio in antimicrobial resistance that was depicted maybe to foster some of these areas that we think that is more important that we need to pursue. And these are some of the projects that are being funded so far. One in CRISPR-Cas9 system, understanding a little better the evolution of resistance to pathogenic bacteria. This is in Pernambuco in the northeastern of the country. This applicability of a high-resolution melting analysis of DNA association. This is basically for diagnostics. Genomic characterization, which is basically analysis for epidemiological surveillance, using omics, and also another computational model for understanding Pseudomonas resistant drug for multi-drug-resistant strains. So, what we are planning to do, and this is my last slide, we are actually trying to do some on-demand kind of fostering research in Fiocruz, using either all the capacity that we have built in our institution. Since we have hospitals, we have surveillance, we have to do all the analysis using different institutes. Here are the name of the institutes that are already doing this kind of research. And so we are also doing the prospection of new drugs. This is very important because we didn't have new classes of antibiotics for the past, maybe I would say, 40 years. And vaccines and adjuvants are also very important, and also alternative methods. So there are some projects that we are evaluating to fund, which is basically mainly on the area of phage therapy to also be used here. And diagnostic kits. It's very important that we have, direct resistant detection. This is being developed now. Also, the discrimination between viral and bacterial infection is quite simple and easy to perform. And one that's been discussed here just before this presentation is that we could use metagenomics to improve the detection and try to understand a little better even the surveillance. This is important to mention because then I can wear my head of laboratory hat again and tell that I'm part of the MetaSub, which is a large network to do surveillance in the mass transportation systems around the world, and we are just swabbing different surfaces and trying using metagenomics as a mechanism to early detect resistance. We are about to, send to publication a very interesting paper. More than 50 cities in the world doing this kind of surveillance. So this is maybe the next step for antimicrobial resistance. And obviously, we need a lot of monitoring, information, integration, mathematical modeling to understand a little better the distribution of those strains around the world and how we're going to manage and how we're going to depict that. And obviously, education and communication. That's it. Thank you very much.
We have time for 2 questions.
Milton, very nice talk. I was wondering, because prevention seems to be a very important player in this field, and I was wondering why we are not using more vaccines in order to prevent infection and in that way prevent antimicrobial resistance. What do you think?
Yeah, this is a very good question. Actually, the point is that, well, here in Brazil, we do have a very, very good national program for immunization, and we are using flu shots every year, especially for elderly and children. And but quite frankly, I don't know how the impact of that, because obviously we still have the circulation of those, and we still, we know that the more antibiotic that we use, the more resistant we have, right? So this is a very direct and straightforward correlation. So we have to reduce the use of antibiotics, and we know that vaccines are preventing, but here in Brazil we don't have this impact analysis yet.
But do you think that, for example, the problem that vaccines cannot be protected by intellectual property rights instead of like a new drug or a new antibiotics maybe, and because of that the pharmaceuticals are not pushing for new vaccine development?
Yeah, well, this is one of those issues that we have to discuss. But what we know is that, at least there's this correlation that's quite, again, between infection, flu infection, and bacterial infection, and then resistance, right? And but the point is, we should have better and coverage and trying to understand a little.
Okay, thank you.
So now we want to introduce Marta Molerach. Marta is a professor of microbiology at the Pharmaceutical and Biochemistry School in Buenos Aires. Marta has a long career studying antimicrobial resistance, so we are listening to learn from her.
Good morning. Thanks to the organizer for inviting me to be here today, and congratulations for this new platform for the people from the USP and the Pasteur Institute. So in my lab, we are working on antimicrobial resistance for a long time, and now it's an issue that was presented before me of great concern for the people. We are going towards the post-antibiotic era. What does it mean? That we will not have more useful antibiotics to treat infectious disease. And moreover, the cancer chemotherapy and also major surgery will be complicated if we would not have good antibiotics. So in 2015, as it was shown before, the WHO released the Global Action Plan to tackle this problem. Among the strategic objectives, there is some that, as part of the academia, as scientists, we can work a lot. We can work on education, we can work on surveillance, we can work, obviously, in research and development. So we have to remember that microorganisms and resistant bacteria are found everywhere, not only in humans, but also in environment, water, animals, food. So the WHO proposed that this problem must be treated from a One Health perspective. Additionally, it was released a global priority list for the pathogens on which we have to make focus in concerning research and development. Among this list, the methicillin-resistant Staph aureus is a pathogen considered for high priority. In that list, we also found Klebsiella pneumonia resistant to carbapenems, Pseudomonas, Acinetobacter, and all of these gram-negative bacteria. In our lab, we are working on all these pathogens, but today I will present you the result of my group, who is working in methicillin-resistant Staph aureus. So, as you know, Staph aureus is a bacteria that is present in healthy people. In 30% of us are carriers of this bacteria. But it can also cause it's the most common cause of skin and soft tissue infections, and it can also cause very severe invasive infections as endocarditis, osteomyelitis, pneumonia, and meningitis. Methicillin-resistant Staph aureus is a special kind of Staph aureus that have acquired a mobile genetic element carrying MEK-A gene. And as a result of this acquisition, this bacteria is resistant to all beta-lactam antibiotics. So since the 60s, MRSA became synonym to nosocomial pathogens. Worldwide, there was a worldwide pandemic of MRSA in every hospital, not only in undeveloped countries, but also in the central countries. But what happened? By the end of the 20th century, another type of methicillin-resistant Staph aureus emerged among healthy people without any contact with a hospital setting. So there were a lot of press releases very causing fear, and schools were closed in the United States, for example, and there was young people without risk factors, healthy people who died by infection caused by this bacteria. So at that time, the question was, which is the difference of the strains causing healthcare-associated disease and community-acquired disease? In first place, the cassette that carried MECA gene is different in both groups. Another good news is that community-acquired methicillin-resistant Staph aureus are most susceptible to other families of antibiotics, but the bad news is that these strains are particularly virulent. So in our country, when we speak about hospital-acquired clones circulating, at first we had the Brazilian clone that was a multi-resistant strain that was an epidemic in every hospital here in Brazil and also in Argentina. Then it was replaced by the Cordobés clone, a little bit more susceptible, but also resistant to more than 3 families of antibiotics. And then this clone began to replace the previous one, the Brazilian clone, and it became the prevalent one clone. After that, we begin to detect in the hospitals community clones that were circulating inside the hospitals. So the barriers between those environments began to fade. What about the community-acquired clones? Some clones have been described so far. For example, the ST8 is known as USA 300, is a very virulent clone that caused a lot of concern in the States, and also then it go to Europe. But as can you see here, there was no information about what was happening in Argentina. So the first reports we released were methicillin-resistant Staph aureus as a cause of meningitis in the community. 2 children without risk factor for MRSA acquire meningitis caused by this bacteria. Then we begin to study in a multi-center study, including the eastern and the northeastern part of our country, which are the main strains circulating, and also we screen healthy children in the last year of the kindergarten. After these studies, we conclude that the lineage ST5 carrying the cassette 4 was the main clone circulating at that moment. And also we find a small proportion of ST30. In 2009, the infectious disease specialists in our country were very worried because the evolution of patients who have skin lesions caused by MRSA have very, very bad evolution, because at that moment, the empirical treatment for furunculosis and all the skin lesions caused by by bacteria were treated by beta-lactam antibiotic as an empiric treatment. And the use of vancomycin, linezolid, and daptomycin was preserved for patients with systemic manifestations. So at that moment, the question in the guides was, well, we do not know which is the rate of methicillin-resistant Staph aureus causing skin and soft tissue infections. And if this rate is very high, it is a moment to change the treatment. So the questions at that moment was, which is the prevalence of skin and soft tissue infection caused by MRSA in Argentina? Must we change the guidelines for empirical treatment of these infections? Is ST5 clone still dominant in Argentina? So to answer these questions, we carried out 2 prospective multicenter studies. One to study the prevalence of MRSA in skin and soft tissue infections, and the other was a study of invasive infections. After this study, which included 19 institutions in our country, we determined that MRSA was the main cause of skin and soft tissue infections. So the guidelines for the empirical treatment of this pathology was modified after our study. And the other question, what about the clone that was circulating in both studies, in invasive studies, infection study, and also in skin and soft tissue infection study, we determined that ST30 is the new predominant clone causing invasive and non-invasive infections in replacement of the ST5 previously described. Well, but what does it mean, this information? Why it was important to know the clones that was circulating? When we compared these two studies, it was evident that ST30 was the predominant clone in both types of studies, but the association between ST30 and invasive infection is significant. So at that moment, a new question arises. Is the new predominant clone more virulent than the previous one? So we carried out a study to compare ST30 versus ST5. And the first step was to analyze by PCR the virulence gene profile, and there were genes that were different and can explain this different behavior. But also we carried out two different animal models to compare the behavior of those strains. And the higher virulence in the two animal models was demonstrating ST30 was the clone that is recovered in higher quantity in both animal models. So, as a conclusion, we can say that ST30, the actual clone that is circulating and causing methicillin-resistant Staph aureus in the community, is a high-risk clone. Why? Because it is very virulent and it has an extraordinary ability to spread and replace the previous one clone. So now we have new questions to be answered concerning this clone and concerning methicillin-resistant Staph aureus. We have a lot of work in progress using whole genome sequencing. For example, one of the questions we want to answer is there any genomic change in ST30 from the first time we detect it in the very first studies to now that caused the expansion of this clone? Did it acquire additional resistance or virulence determinants? And to answer all of this, we are carrying out genomic surveillance of MRSA not only in our country, but also we are trying to expand this to South America. Another area in which we are working is to study the microevolution of methicillin-resistant Staph aureus in the lung of cystic fibrosis patients. As you know, these kind of patients receive very, very complicated treatments with a lot of antibiotics of different families. So we are following these patients, the strains we recover from these patients, and we have obtained a lot of results that show that the movement of insertion sequences in the genome of this bacteria that has an extraordinary genome plasticity are in part responsible of this evolution. So, concerning the genomic surveillance, now we are engaged in a project called StaphNet South America as a network for genomic surveillance of Staph aureus in South America. We are carrying out this project with in collaboration with the Sanger Institute, with Dr. David Annensen and Silvia Argimon, and we have invited a lot of people from Brazil, from the University of São Paulo, the University of Rio de Janeiro. From Uruguay, we have people from the UDELAR and also the National Reference Center. We have people from Bolivia and also from Paraguay. We are trying to incorporate people from universities and also from national reference centers. And at this moment, well, last year we performed the first meeting that was held in our faculty at the University of Buenos Aires. And the specific objectives are to establish a genomic surveillance network of Staph aureus, to study the population structure of Staph aureus, to identify the circulating clones, and to determine the presence of virulence factors and antibiotic resistance determinants. But one of the main points is to develop regional capabilities for the sequencing and informatic analysis of full genome sequences, not only of Staph aureus, but also of other resistant pathogens. Well, thank you for your attention. This is my group, and these are the institutions who give some financial support to our work. Thank you very much for your attention.
We have time for one question.
Thank you for your presentation. I would like to know the proportion of the pentavalentine leukocidin among these both clone ST5 and ST13. The other question is the prevalence of these both clone in the hospital setting.
Sorry, would you like to repeat me?
So I say I would like to know the prevalence of the leukocidin of pentavalent, the PVL, among the clone ST5 and ST13 isolated in your country. And the second question is the proportion of the ST5 and ST13 in hospital setting.
Okay, it depends on the exactly on which region of the country we are speaking about. In the university hospital, we have 30% of their MRSA are now ST30. And, but in the south part of the country, for example, ST30 is not so prevalent compared to the ST5. So central region and north region, the prevalence of ST30 is higher. And the other part of the question, you say something about the phantom-valentine leukocidin. Well, all these strains harbor the phantom-valentine leukocidin, are positive. Did I answer your question? Thank you.
Thank you a lot, Marta. So we would like now to introduce Esteban Chávez Olarte from the Center for Research in Tropical Disease from Costa Rica.
Well, good morning. First, I would like to thank the opportunity to be here, particularly to Paula, who allowed us to give you this presentation. Thank you, Paula. First, I would like to start with a brief introduction of who we are and where we are. We are located in the University of Costa Rica, which is the main university in our country and also the major university in Central America. It's ranked as number 19 in Latin America. And our center, which is called Center for Research in Tropical Diseases, is located within the Faculty of Microbiology. It's very nice that we have a student from our university and our faculty who is here, and actually she brought me from the airport this morning, which is very nice. We are a very small center, but still very active and very enthusiastic, and we have several research interests, as Clostridium difficile, brucellosis, arboviruses, malaria, Xylella fastidiosa, which is a plant pathogen, and we have a group that is starting working on cancer. We have had a strong tradition of regional cooperation, and actually currently we are not part of the International Pasteur initiative, but we do believe that since we have this strong cooperation in the region, we can serve as a hub for interaction of Pasteur with all Central America. Thanks to the help with Javier Pizarro, who is a Costa Rican, who has worked at Pasteur for 20 years. We organized a meeting this year and trying to promote the effort that we can get an agreement together with the Institut Pasteur. Now going into science, I would like to discuss with you one of our major research interests, that is Clostridium difficile. As you may know, Clostridium difficile is a nosocomial pathogen that affects older people that who have an underlying chronic disease, debilitating disease. And when this kind of patients, they receive an antibiotic treatment, what this antibiotic treatment is going to do is that it will erase the normal flora from the gut, and this will eliminate the competition that normally keeps Clostridium difficile under control. When Clostridium difficile proliferates, then it will produce two toxins in the gut, toxins A and toxin B. And what these toxins will do is that they are cytotoxins, and they will affect the epithelial cells of the gut, and they will trigger a strong pro-inflammatory response. And this will lead to what is called antibiotic-associated diarrhea, and if it is it can go into pseudomembranous colitis, which can be a fatal disease. This bacteria is a very variable species. You can see that the core genome can be as low as 30%, and it presents a high plasticity. That means that it can exchange a lot of genetic factors. And that gives rise to a lot of strains with different virulence potential. For instance, there's a worldwide distributed strain that is called the NAP1 27 strain genotype that is globally distributed, is a hypervirulent phenotype, and it has several characteristics that makes a strain that has a very high virulence potential. For instance, it presents resistance to fluoroquinolones. It presents an additional toxin, binary toxins. It presents increased toxin production, toxin A and toxin B production. And it has also increased sporulation and presents a different kind of toxin B, which makes it more active. So we became interested in CIET on Clostridium difficile because an outbreak that was presented in a major Costa Rican hospital. You can see that we have a lot of cases at that time. And then we started collecting all the strains that were responsible for this outbreak. We did a genomic characterization of those strains, and we found that most of the strains that were responsible for this outbreak belong to this NAP1 hypervirulent phenotype. But we also discovered another circulating strain that was also very prevalent in this outbreak, and this was an autochthonous strain that we named NAPCR because this genotype has not been described before. So we started working on this emerging virulent strain. And as you can see from the clinical data, we can see that this autochthonous NAPCR strain was as virulent as the NAP1 strain in terms of severity of the disease, in terms of recurrences, and in terms of mortality. Also, when we took this strain and we tried this strain on the hamster model, we could see that this strain was as virulent as the NAP1 strain. So the message here is that we detected a new emerging strain that was as virulent as the globally distributed NAP1 strain. So we did a full genomic and phenotypic characterization of this strain, and we found some similarities with the NAP1 strain. Particularly, we found that this strain was also resistant to fluoroquinolones due to the exact same mutation as the NAP1 strain presented. But in contrast to the NAP1 strain, this new genotype did not overproduce toxins. So we landed with a new hypervirulent strain that did not have overproduction of toxins. So we formulated the hypothesis that the NAPCR strain should secrete additional components in addition to the toxins that should synergize the effect of the toxins, and that would be one of the reasons why this strain would be more virulent. So we tested this hypothesis using another animal model, and this is the ligated loop mice model. We took a loop of the intestine, and we inoculated this loop with supernatants from the different strains. And we inoculated supernatants with toxins and without toxins. And as you can see, when you inoculate the supernatant from the NAPCR1 strain and the NAP1 strain, then you get a very strong response, But when you remove the toxins, only the NAPCR strain is able to induce a strong response. When we measure the cytokine release using exactly the same model, we could see that, again, when we inoculated the loops with the NAP1 supernatants and the NAPCR supernatants, we had a strong release of cytotoxins when we used the supernatants with toxins, but when we removed the toxins, we could see that only the NAPCR strain was able to induce a very strong pro-inflammatory response. So we believe that we have described an emerging virulent strain that is resistant to fluoroquinolone, and this could be very important for the spreading of this strain. And we think that we are in front of a new or novel virulence mechanisms that give rise to emerging virulent strains in Clostridium difficile. Together with the Sanger Institute, we have sequenced a lot of Clostridium difficile strains from our collections, and then we had done another contributions in the field. For instance, we have seen that this NAPCR strain is multi-drug resistant because of the acquisition of chromosomal mutation and novel mobile genetic elements. So in addition of its hypervirulent phenotype, this strain is also multi-drug resistant. We have studied the microevolution of both the NAP1 and the NAPCR strain, and we have described new strains that escape current diagnostic tests because they produce different variants of toxins which are not detected by current techniques. I just want to finalize with a couple of additional research lines that we have at CIET. We work also very much with brucellosis, which is a zoonotic disease. As you may know, Brucella abortus infects cattle, and then it produces abortion, and the human can be infected when it consumes unpasteurized milk products. So within the genus, we have different species, and then in this genus, we have found, for instance, that Brucella seti goes into the brain of dolphins, and it produces neurobrucellosis. And we have described this as a major cause of stranded in this kind of mammals, of marine mammals. And we have also described that Brucella neotome, which was not known before, can be also a pathogen for humans. We have found it in the brain of a couple of patients. So we are also doing a contribution in emerging diseases as brucellosis. And finally, a very exciting field that we have a team working on, which is the developing of molecular and cellular sensors for the replication of flavivirus. The principle of this sensor is that if you have a GFP and then you have a quenching peptide, this GFP is not going to provide the fluorescence that it normally does. And if you introduce a sequence that is going to be a target for the protease, and this was developed for caspases, then the caspase is activated, and then this quenching peptide is released, and then the GFP is going to provide the fluorescence. So we have modified the sequence of that is recognized by the protease, and we have introduced a sequence that is recognized by the NS3 protease from flaviviruses. So what we have is that when we mix the GFP with the protease from Zika virus or dengue virus, then we have an increase in the fluorescence. We can get this increase not only in vitro, but also if we put this construct into cells using lentiviruses, you can see that when you infect cells, then the protease, you infect cells with the dengue virus, for instance, then you get the expression of the protease, and then the GFP is cleaved, and then you get a very nice signal due to this effect. So we do believe that this is a very exciting field that we can use in the future for diagnostic purposes or for research. And with that, I would like to finalize, and thank you very much.
Thank you a lot. We have time for 2 questions.
I have one question. Do you restore the microbiota in case of Clostridium difficile infection?
If we have restored the microbiota,
yeah,
we have not worked on this line of
stools,
research.
you know,
yeah, but if you restore the microbiota, then you will get the disease down. Yeah, for sure. But we are not working on that.
You don't use it in Costa Rica?
No.
No. Okay.
All right, so because we are very afraid of Paola and we are running out of time, so we invite us, everybody to go for lunch and no? Okay, one picture outside, but no time for questions.
Yes, I'm sorry because my co-chair is absent today, he's sick. And I'm very glad to introduce this session. It's the first round table with my colleagues, directors of different institutes in the region. And the aim of this session is to have a discussion and each director will have a presentation of the research themes and of the platform, and you can have some question and some interaction with research action we can share about this. That's right? Mm-hmm. Sabino, you start?
No, I start.
Sorry, I'm the chairman.
Yeah, the idea was to show you in a couple of slides what is our institute, what is the institute in the American region, and how we can interact together. If you are interested in the future doing a postdoc in Montevideo or in Fiocruz or in French Guiana, that you have an idea what exists there and what we are working on. Then the French Guiana, Pasteur Institute in the French Guiana is the, next year will be the, comment on peut mettre là? Yeah. Yeah, the next year, it's the 80th anniversary. Then the institute was built in 1940, and it's a part of the institute located in South America. And the mission of our institute is like all the Pasteur Institute across the world, which is focused in 3 missions. The first one is research, the second one is public health, and the third mission is teaching. Then you will see that in all of institutes in the world, even in Pasteur Institute in Paris. Then the Pasteur Institute is the technical hub, which is located in the north part of French Guiana, in Cayenne. This is the main city. And there is many research laboratories, medical analysis facilities for people. And there is also a big animal facilities. For many years, there was a primate center with a huge number of Saimeri monkeys. Now this center was closed, and we have animal facilities for mice. We have 3 level 3 biosecurity laboratories, one dedicated only for entomology, for vector studies, and one for virology and one for bacteriology, but essentially for TB. And there is an insectarium, molecular biology platform, and the Vectopol. This is some picture of our Vectopol. It's 500 meters of lab with many compartments, and each compartment dedicated to different kind of studies. The main research activity in our institute is focused on vector approach related to arboviruses to improve the diagnostic and care, and also to understand the transmission and dissemination of resistance of the vectors and also the viruses. Other objective of our institute is to understand the biodiversity of the virus located in this area of the world with their reservoir, as Paolo this morning showed us very well the importance of this aspect of studies. And we also have a lab for malaria research in vector and also in the diversity of the parasites. The lab, the institute is divided to many laboratories, and rapidly I will show you the different departments. This is the Department of Entomology, and directed by Jean-Bernard Duchemin, who will give us a very interesting talk this morning. And this is the main objective of this department, studying the dynamic of the resistance, insecticide discovery, and the reaction between insecticide resistance and vector competence and their staff. In this department, we have also a G5 facility. The G5 is in all the Pasteur Institute in the network. There is G4 facilities, and Paula get G4 facilities here, and it's very, very important for the lab here. And we, as French Guiana is a part of France, then we have the possibility to get a G5 facility. Then since now 2 years, we have a group for 5 years working in microbiota and insect vectors. And also they develop research to understand the composition, the community of the microbiota of the mosquitoes and related to parasites and viruses. The Human Virology Laboratory is dedicated to development and tests, but also to clinical research and epidemiology in connection with the epidemiology unity. We have another department dedicated to viruses, parasitology, and the epidemiology, of course, that you heard this morning Claude giving a talk on his work, but he's also developing other research related to modeling and the climatic, environmental, and the human determinant of vector-borne in connection with the Vectopol and also with the laboratory lab. We have in French Guiana also reference laboratories for supporting the public health. These reference laboratory related to influenza, arboviruses, hantaviruses, but also another laboratory reference center for malaria, which is also WHO collaborating center for surveillance and anti-malaria drug resistance. Then we have also services laboratory, 2 service laboratories, one dedicated to medical biology laboratory for patients that came and made the analysis, and the other one is the environmental and health laboratory for water and chemical analysis. Finally, we have, as I mentioned before, a teaching program related to the university in Guyana, and we receive students from French Guiana, but also from other universities in France to doing their PhD and also postdoc. And it's a very, very good connection with the university. This is a picture with my friend Luis Carlos. I am very frustrated that we didn't get the picture outside. And this is why, you know, it's a nice souvenir of friends from here, Sao Paulo, with you and with the president of the university. Thank you.
Antoine.
C'est bon? Voilà, c'est quoi ça?
Okay, I'll try to make it short if we want to have some time for discussion. So the Pasteur Institute of Guadeloupe is nearly as old as the one in French Guiana. It was created in 1948. It's one of the smallest of the network, and we have service activities that hardly are sufficient for their own activities, so they don't finance at all the research. Sorry, up. No, okay, as you can see, nearly all staff, no, more than one half of the staff is in the service activities, but only 55 to 60% of the income. And we have melted two units. We had recently two units, research units, one on mycobacteria and the other one on environmental health, and we have melted the both to create one unit, pathogens transmission, reservoir, and diversity, with two labs. One lab on entomology with at the head Anubis, and the other one, the laboratory of microbial ecosystems. And I am at the head of this one. So as an axis of research, we have three axes: vector control, with some study on the surveillance of resistance to insecticides, but the major part of the entomology lab is alternative control methods, with studies on pheromones and the use of pheromones to control and to better understand how Aedes aegypti, because we are working on Aedes aegypti, how we can control Aedes aegypti with very specific molecules. And the other alternative control methods is a study of the microbiota of Aedes aegypti, and to see if some bacteria can modify the competency of the vector. And the other lab, we have two axes: the pathogen from the environment, and these pathogens are rather specific of French Guiana or emerging. French living amoeba, and especially Naegleria fowleri, are present in hot water, and they have a death occurred in a child 10 years ago. So we are working to now better understand the virulence factor of Naegleria fowleri. Strongylus cantonensis is emerging there. We have some cases occurring each year, and it was not present before the 21st century. And so we are now trying to better understand how this parasite was introduced in Guadeloupe. And the other one is Klebsiella pneumoniae. We have some cases of abscess due to Klebsiella pneumoniae hypervirulent. And so we are trying to understand how the patients are contaminated. And concerning antimicrobial resistance, we are studying the diffusion of resistant bacteria and resistant genes through the environment. And we have made some study in work plant, and now we are studying the exchange of bacteria and genes between different biotopes, and especially between humans, domestic animals, and wild animals. And for this, we have some platform. Of course, a level 3 microbial safety laboratory for mycobacteria and also for entomology. And we have also molecular biology, proteomics also with the Madi-Tov and NanoLC. And especially we have bioinformatics facilities with the help of Montevideo. And we have now 3 bioinformaticians, and we are the leader in Guadeloupe on these topics. It's very important for us. And we have a chemical lab, which is not so frequent in the network. And this chemical lab is rather well equipped. And it's not using its equipment now for research, but it will occur especially for pheromone in the near future. So what the training axis we want to develop is clearly the development of the Guadeloupean bioinformatic capacities, because it's very important for us and for Guadeloupe. And we have already organized some metagenomics training with Carlos, who is here. And we have also a discussion group led by the Pasteur Institute, and the Master in Bioinformatics should probably be implemented next year or in the very near future. And in entomology, there is a need in the Caribbean, but we think that the platform for entomology training should be French Guiana for us.
Thank you.
So main partners, of course, we have made a lot of collaboration, especially since the arrival of Anubis, especially with Instituto Pasteur in Cuba. And we have only 3 projects in America, with Pasteur Institute of America. Okay, so as you can see, the small size of our institute limits ambitious projects. And as you know, monthly R&D increases. So I think the network should be used as a lever to raise money. And for this, my conviction is that we should implement ambitious public health programs with, inside these programs, fundamental, applied, and transnational research project. And for this, we must use complementarity of the network teams instead of duplicating the same activities in different countries. Thank you.
Merci Antoine. Thank you, Antoine. And Carlos now from Montevideo.
So good afternoon. I would like to introduce the Institut Pasteur Montevideo. We are a very novel institute. We are arriving to our 12, 13 years of life. And as you can see, so at the very beginning we were very few ones, and right now we are like almost 300 persons working at the institute. So the Institut Pasteur is a private foundation that was founded in 2006, at the end of 2006, almost 2007, in an agreement that was signed between the French and Uruguayan governments and the National University. Universidad de la República is the name in Spanish. Our mission was, first of all, to produce curiosity-driven science of international level in the biomedical field. Second, to train highly qualified scientists and related human resources. And right now we have the opportunity to join the IPIN or RIIP task force that we are going to discuss later on. Finally, the last mission of our institute was to articulate research discoveries and their applications, contributing to something that is very difficult in Uruguay, to make Uruguay an innovative country with an economy based on knowledge, okay? So in our region, our countries do not have the view that we can be a society supported on science and based on knowledge, okay? So that is our next goal. So just for you to see at a glance, we have different research groups. Half of them are being paid from our own institute, and half of them are being paid by other institutions that are in Uruguay or abroad. For example, Pasteur Paris is paying one of our main researchers. We have a recently launched G4 program. Right now we have 2 groups which are directed by Gregorio Iraola, that you already know him, and Maria Eugenia Francia. They started last year, and we are going to fulfill the program with 4 groups running all the time. So this is like from the personal point of view, we have 268 personnel right now, 220 are scientists, and last year there were 21 theses defended in 2018. Half of them were master theses and half of them were PhD theses. If we are talking about our scientific priorities for the next 5 years, we have decided that we are going to be working on 4 axes: molecular mechanisms of pathogenesis, human and animal diseases. Of course, not all of them. In the human area, we are working mainly on cardiovascular and cancer diseases, also a little bit on some infection diseases. And in animal diseases, we are focusing on zoonotic diseases. We are starting working on biomarker diagnosis and also rational drug design and development, immune response and vaccine development for animal diseases, where we have 2 examples already going, and system microbiology and antimicrobial resistance is the last part of our work. When we were launched in 2006, one of the main ideas of the Pasteur Institute was to bring important technological units and core facility to the regions. So we have different technological units like recombinant proteins, proteomics, mass spectrometry, crystallography, so on and so on. And we are trying to always to renew and update one of our main equipments every 2 years. So the last equipment that we acquired in the last 5 years are the ones that I'm showing in the slide. The second mission that the Institute has is to train people from our country and from abroad, and we are giving around 10 courses per year, receiving around 200 students per year, half of them from the region, half of them from Uruguay, and we are reaching a plateau with 70 invited professors per year to help us with the courses. And the grants, the money is coming from the ICGB, the United Nations University, our own network, also some funds are from Uruguay. How are we doing from a scientific point of view? So when we started in 2006, of course, we were not publishing a lot. Right now, we think that we reach a nice situation where we are publishing almost 90 papers per year. We don't want to increase the number of papers. What we want to do is to improve the quality of our papers, trying to have several principal investigators working together. Right now, our citations per paper, average citation per paper is 24, with an impact factor of around 4.6. That is pretty good for our region. The last point, and I'm going to spend a few minutes talking about this, is that in 2014, the previous director, Luis Barbator, was able to build an incubator and an innovation space in our institution with the idea of pursuing our 3rd foundation mission. So right now we have an important space for innovation, and as you well know, the venture capitals are soaring right now in Latin America, and they were moving from United States to Europe, towards Asia, and right now they are coming to Latin America. Latin America is a non-exploited right now region for venture capitals. 16% of the money is being allocated right now outside the information and communication area. So we think that for life science technologies, we have a spot right there to develop. So what is our next goal for the next 5 years is to develop an Institut Pasteur Montevideo Latam BioVenture incubator. The idea is to consolidate a private fund of between $30 to $50 million to support the development of between 30 to 40 startups in 5 to 8 years. I'm not going to go into details, but this is going to be our real challenge project for the period. And of course, we are going to do this as we do everything in our institution, and that is doing international calls to bring to our country talent scientists willing to develop their discoveries. When we founded the institution, the Institut Pasteur, all the researchers were hired by international calls. They were working abroad, they were working at the United States, in Europe, whatever. So, and they came back to Uruguay because of the opportunity the Institut Pasteur was giving to them. So the idea for this field is actually the same, is to, if we allocate the money, then we are going to perform international calls, and we believe that the network, the IP network, is going to be very important for that. So finally, I'm just ending. I want to thanks the International Scientific Committee, because we could not have achieved what we did without the help and their important messages and advice that they gave to us in the last 12 years. In the picture, you can see Samuel Goldenberg from Brazil, Cecilia Fernandez, Alberto Kornblit, Cecilia from Uruguay, Argentina, Pedro Alzari from Pasteur Paris, our former director, Luis Barbeito, Felix Rey from Pasteur Paris, Sabino, who everybody knows, Adrian Kreiner from Uruguay, but working in New York, and Ricardo Arlich. So, thank you very much.
Thank you. Paola? No. Donc, Savino. Savino for Fiocruz. Yes. Yes.
Very good. No. Wonderful. Impressive. Bravo. Great institute. Very excellent. Very good.
So good afternoon. Actually, I was just asked to present a little bit of Fiocruz, and I'll try to do that in less than 10 minutes. And one part of present actions actually will be presented by Milton Moraes in a few minutes in the next session. Where do you intend to find your money, your 50 million? We already talked with private funds, and they are already in Uruguay? In Uruguay, Argentina, and Brazil. Okay. Yeah, so we are trying to capture that. Not here. No, it's not this.
Yeah, but we are depending on Paris and here. C'est bon, on t'a soulevé les signes.
So, as you know, the Oswaldo Cruz Foundation was built in 1900. So, next year we'll be celebrating the 120th anniversary of Fiocruz. And sorry, let me do that. So what I'm going to do is to present very briefly some aspects that I think it's worthwhile that can be used in the next discussion on how we could use tools of the different institutions in terms of doing something together. So first of all, I think it's important to say to everybody that Fiocruz is a public institution belonging to the Ministry of Health. So different from other institutions, since ever, Fiocruz is a public institution. We are employees of the Ministry of Health in Brazil. Although being a foundation, it's a public foundation. And it is a nation perspective since it was founded by Oswaldo Cruz in 1900, with different campaigns in different parts of Brazil.
And based on the experience that Oswaldo Cruz had in Pasteur, so production of vaccine was also something very important. Here you have the people putting in the vials of yellow fever vaccines in boxes to be expedited. So it is a complex system in which you do research, education, with innovation and production, surveillance with reference centers, health and medical care, environment. So we took, since many years ago, the concept of One Health to work on and tackle problems of human health. We also have a strong part of information and communication, in unity for quality control, and also doing history of science and health. And of course, to do these kinds of things, it's absolutely necessary to have a strategic plan and management of this plan. So this is Fiocruz in a very brief cartoon. So we are 12,000 workers, almost half of it being employees, public employees. We have more than 1,500 PhD employees, and in terms of research, almost 30 areas of research. Again, this goes from social sciences to genomics in different areas. Let's say, for example, from violence to transcription, with more than 1,500 research projects going on, with almost 40 MSc PhD programs nationwide. I'm going to show you in a moment that Fiocruz is a nationwide institution, and so we have thousands of students coming from the institution. In terms of production, sort of 80 million doses of vaccines, 4.5 million doses reagents for diagnosis, etc., etc., etc. So we do research, education, and production, all this to the Brazilian society, actually. And as I told you before, so Fiocruz is physically implanted in different states of the country. Don't forget that Brazil is a federative republic. And I'd like to point out some aspects that I think can be useful for the discussion afterwards, is that this Fiocruz Brasilia in the capital has more and more a quite strategic position in terms of dealing with the Congress. And I think this is one of the important things that we should look for in terms of planning in medium terms and long-term strategies for the regions. We cannot forget, and we must act with the parliaments. In this scenario of Brazil, of course, the Amazon region is completely different in terms of epidemiology, environment, etc., from the south. And more and more different units in different parts of the country are being more specified so that we can have in the future a more complementarity between the different research units throughout the country. And of course, the international cooperation exists since ever, and reference centers as well. I'm not going to go into details, but we have WHO collaborating centers, other reference centers either in the national level or the PAHO level. With this kind of strategy for education, we are devising not only PhD, MSc programs, but we also have a high school, actually, specifically for formation of people with techniques in the biomedical field. We don't have university, we don't have undergraduate formation, except for those people that come to do research, those students, undergraduate students that come to do research in the research units that we have throughout the country. So there are a number of priorities, and these priorities are more and more being structured in the form of translational programs in which you can have from basic science to development of products. And when I say products, I'm not only saying, let's say, a pill or a test or something, but also products that can be used directly to the society. Such a kind of soft innovation, use it for education and communication directed to the society. These are reference centers, the themes of reference centers, both in infectious diseases, but also in maternal and infant care. The lactation and human milk banks is something very important that has been actually exported throughout the world already. Different countries in Africa incorporated based on technological transfer from Fiocruz to develop milk banks in their countries, and this is really quite important for these countries. I will not go to this part because Milton is going to talk about, but in any case, innovation is something that is part of Fiocruz, actually part of Fiocruz DNA, with the innovation with tradition, as we say. So looking for the past, looking at the past, but looking for the future. And Milton is going to explain the recent Innova Fiocruz, which is a large and ambitious program for intramural financing and developing of research. So Lisa has already talked about. So productions of vaccines. Fiocruz is very important in terms of producing vaccines for the Brazilian society, but also for other countries. And we are now developing intramurally some vaccines, particularly one vaccine for schistosomiasis, which has been so far completely developed at Fiocruz. Also, Biomanguinhos, which is one of the two units for production. Biomanguinhos produces vaccines, but also diagnostic tests for a number of infectious diseases. And biopharmaceuticals that we have already entered the era of biopharmaceuticals. So I'll go ahead. Drugs, by the unit of producing drugs for Manguinhos. Fiocruz is accredited by the government to do quality control for many things for the Ministry of Health. And information communication. And here I'd like to say that there is some strategies that can be applied for the whole region, as for example, using a TV channel for health that we have at Fiocruz that can be used to everybody, by everybody, and also the fact that we have a structure for editing books and videos, not only in Portuguese, by the way. And this is done in the context of looking for the past and for the future at the same time. And so far, we are one of the most important institutions doing history of science and health. So this is it, 8 minutes, so I think we can do that this is one of the most important things that we have, science and health for all. And I would say, I would finish by looking for the society rather than ourselves. Thank you.
Paola.
So I think that I will try to be very quick. First of all, I would like to thank you very much to give us the opportunity to give this some words. We are not from the RIIP, and we are pretending to be on the RIIP. And our platform, scientific platform, is not inaugurated yet, so it's very easy to talk about it. So we are... Ah, se foi copiar lá dentro? Ups. Thank you, Paulo. So it's very, very easy. The scientific platform Pasteur USP, we are going to meet, to visit tomorrow. So you see, we are not functional yet. We are moving. Since 1 month ago, we are buying equipments, receiving equipments, putting people inside, trying to move some books, etc., etc. It is in the building, a beautiful building called Innova USP. That is the new innovation center from USP. We are one of the 4 units of the Innova USP, and the executive coordinators of this platform are Luis Carlos Souza Ferreira, that you know that is the boss also of the ICB, and myself. And we are very in phase doing our best for that platform, at least start being functional in the next few weeks. So the inauguration tomorrow, we have the students of the platform that are supposed to come into the labs of the platform are going to make you to visit, because we are open everything, and you see the labs, how they are, with or without all the equipment, but at least we are very proud. We have been working very hardly to have you there, seeing what we achieved. So you see in the right part of the slide, the 3 buildings which compose Innova USP, and you have the 3rd and 4th floor is our platform. So actually we have been basing our strategy, scientific strategy, on the agreement that was found the 31st of August 2016 for thinking that the Pasteur Institute in Brazil cannot have just one site. So the first site would be that platform in São Paulo, and another possible site would be in Ceará, in Fortaleza, in Eusébio, sorry. It is a new unit of a few crews. So we are starting now with this platform in São Paulo, trying to work with emerging diseases. As Paulo said this morning, we have emerging diseases, emerging viruses, and also some parasites that can commit the brain. And we are going to study a little bit of migration of vectors with our partners from Guyana, and do our best with the people that we choose, especially by a scientific committee, to study the strategy, capitalizing the resources offered here in South America and our colleagues from the state of São Paulo. So the most specific initiatives that we are pretending are to increase and make very high standing laboratory infrastructures, following strict biosafety and quality issues, perform joint research programs between us that are going to be there, and also the non-resident of the platform. That means laboratories that are outside the USP or in the Institute of Biomedical Sciences or in the other state or internationally. We wanted to do trilateral and multilateral courses, innovative courses, and promote innovation. That's why we are in the center of Innova USP. What we would like is to perform as a cellule d'intervention d'urgence, I have no idea how to say that, an intervention cellule to react very quickly to new epidemics. And for that, we have been we have chosen some scientists. So I have here the name of the scientists that are going to join us there. Neurological disease modeling is going to be performed by the team of Patrícia Beltrão Braga. Computational system biology by Elder Nakaya. Tropical epidemiology, an office is going to be there. Who is going to be there is Eduardo Massado from the Faculty of Medicine School. My group, biosafety management that is going to be done by Pedro Teixeira from Fiocruz, that is going to try to apply all the safety strategies that he learned in Fiocruz during his career and also in Pasteur, a year that he spent there, and also apply this first approach in other unities of Fiocruz, for instance, Ribeirão Preto and also Eusébio in Ceará. So just to have an idea, we wanted to establish and standardize procedures in terms of hygiene and safety of the laboratories, to translate and adapt some French rules to the Brazilian reality, to prepare a series of courses that are mandatory for professionals that wanted to be there. So this is Pedro Teixeira. Paulo Zanotto, that talked this morning, is with Edson Durigon, are going to study applied virology, focusing on reverse genetics and viral erotics with viral isolates from relevant outbreaks, and with emphasis on emerging viruses. We are going to do a collaborative job, my group and his group, their groups, studying 2D and 3D systems, in vitro and in vivo, to study isolators of virus that are going to be genetically modified. And this study with Patricia, for instance, in neuroinvasiveness, neuropathogenesis, and with Jean-Pierre Perron, also the mechanisms, immunological mechanisms involved in these diseases, and for sure, discovery of any virus, biobanks, etc. We have Eduardo Massad, that is one of the best mathematical modelists that we have here, computational and statistical methods. System biology, I have already said that. So Patricia is going to do neurological disease modeling. As you all know, Patricia can do a modeling of mini brains that we are going to study with diverse diseases, for instance, autism, influenza, dengue, and Zika. We are going to study with Jean-Pierre Schatzman-Perron, that is the first G4 of the platform, study of the neuroimmunological mechanisms involved in neuroimmune system damage caused by viruses. My group are going to show to offer a little bit of expertise in 2D and 3D imaging strategies for viruses. And as I said, integrated biology is going to be performed by the group of Elder Nakaya. So, the ability to innovate is critical for economic, social, and environmental development. So what we are going to profit from another group that's going to be located in this platform, which is IRIS, that will attract the resources from the private initiatives and the international research funding circuit to make our products real products. Tomorrow we are going to see, we have to offer very high standing conditions of P3 labs. We have a P3 lab, 4 P3 labs, one for maintaining animals, another one for a platform of imaging, one just to use virus diseases, another one for bacteria and parasites. And we have 4 P2, and all the other labs for BSL2 labs, and all the other labs are going to work in partnership, so we are not going to multiply resources. And lastly, we have a platform it's not very big, but it's a platform of 60 square meters that is going to be dedicated to bioinformatics and system biology. So I hope that we are going to offer something good for the state, for the country, and for the region. Thank you very much.
Thank you, Paula. Now it's time for the discussion. You have some questions about the priority of the region. The roundtable is about the priorities and question of proposal for the region. We have some presentation of the main Pasteur Institute in the region. Perhaps some other center of research would like to have some question and some interaction with Institut Pasteur. Yeah. No. It's a time for the discussion and proposal for different orientation.
I have to sort out something with Luis. Luis, can you go there? We have the TV there in the platform and trying to make some movies. However, we don't have everything functioning, so we need a scientist there that can go on. And you know everything about, so you just talk about.
Maybe I should just leave a message here. I will not talk about the University of São Paulo. You are in the University of São Paulo. You know that this is a university of big numbers. Altogether, we have a budget of almost $2 billion per year. We have more than 96,000 students, graduate and undergraduate students, and something like 6,000 faculties, 13,000 employees. But I guess that the most important thing here today that we are celebrating, besides the building, the labs, the bricks and equipment, which is definitely important for research, is to celebrate the union of the people who made everything possible. And particularly, in my opinion, it's very, very important that today, as we are doing the last almost 5 years, to build links among people that trust in each other and wish to work together. Not for ourselves, but mainly for the students and the young people that will be the next generations. And this is my hope and my expectation that today, as well in the coming days, we'll keep this ideal in mind, that we have the responsibility to build together a better future for our students, for our population, and for the people who want to have people like us solving problems and ensure that the knowledge continues to grow and will go to the next generation. So thank you very much. I will have to leave because, as Paula said, there are people waiting for me to have a record there of the lab space. Yes, and that's the message, and I want to leave for them too. Thank you very much. Enjoy the day, and tomorrow I will be back because I will have a meeting here in a few minutes, okay? So I will return soon. Thank you very much.
What do you think we do now? Do we want to discuss, Caitlin, about the priorities, what we do on Friday?
I
or
give
what?
Savino.
Or we leave it to the brainstorm of tomorrow, Friday morning?
No.
Huh?
We still have time for discussion. At least for beginning to make proposals. One of the things that I think is important for any action, either in a single institute or in conjoint with all the institutes of Pôle Amérique and other institutions of the region, is if we're going to draw a project or an action or anything, I think it's absolutely necessary that we have as a background for reasoning the 2030 agenda for the sustainable development goals. In the field of health, several of these objectives for 2030 deals with health directly, as for example number 3, or indirectly when there are climate, there are different aspects, poverty, that are fundamental in terms of understanding and acting upon health problems. So I think that this one of the things that we should take into account when designing a strategy for either a research project or a political action in different countries, or in the for example, the association, the Latin American Association of Academy of Sciences, that can also be a tool for that. But in any case, in any fora that we can discuss this, or projects that we can write. I think that it should be quite clear that we are allied with these sustainable developmental goals.
Yeah, I have also something to say. It's my third meeting of the America region, and I have 3 projects with American IP non-emerged from the American meeting. You know, each time we write a project, but we don't have any money to do it. So it collapsed. It collapsed. So main point is to have a project able to raise money. If we don't have this, it's no use writing anything. The other point for me is we must have a leader, a good leader, and dedicated to this. After that, it's open.
If I can say 2 words and then I pass to Caitlin, I think that this is the way, this is why we decided to do these kind of sessions, very informal, where we present our institutes, we say what we can do. We invited the people from different states, different countries of South America that could also explain and present their work around various emerging resistance of antibiotics and everything. And I think that now we have all the tools necessary to be scientists and create projects. And I think that Antoine is right, is not only to just talk, jogar conversa fora, just talk and not to do anything. We have a space tomorrow to be together and try to discuss how, I don't know if it's tomorrow, I think it's tomorrow morning or Friday morning, how can we work together? What is the way to be together and to explore our ideas? And we have also rooms that are available for in the afternoon where we can sit. The guys and I think Caitlin are going to explain something about that. I don't know if now or on Friday, about the different 3 identified subjects that we found that could be porteurs, I mean, that could be explored for new letter of intentions. Caitlin. Onde é que está o microfone? Não tem microfone?
Yeah, he's coming.
Vem aqui, Kate. Não, ele vai ler.
Yeah, and as you said, Paula, this is the first time for the regional meeting. We invited people from Chile, from Argentina, from Colombia, which is very important for them also to know what we are doing in our network.
Because when we talk about the RIIP, we say that we are regional, we have to be interested in regional problems, but the region is not only the guys that are in the RIIP, in the network of Pasteur Institute, but all the other institutions that are in countries that are suffering from the same public health problems. So that's why we are together and we are exploring this new way of doing a regional meeting. Sorry, Caitlin.
So the idea of the presentations was also, it's a whole, the presentation of this morning and then the presentation of the different institutes were on subjects that we told before with the core of the directors of the Americas that could be important, which is antimicrobial resistance and emerging viruses. What you saw now is also the presentation of what technically is possible in the different institutes. And this is going to have a link also with what are the presentations you are going to follow. Because we need also to think, and Antoine already touched that issue, that we have different platforms within the institute, different know-how, and I think it's also important on the research questions that are between institutes or at the level of the Americas, how can we share these platforms? What would be a possibility on imaging? What are the possibilities in one institute that can be shared? The vector pole of Guyana, everything which has been set up on AMR and on vector control in Guadeloupe. All the possibilities and with innovation in Uruguay, This is also a new development that some of the other institutes also in other parts of the world want to take, and which they have a link to with what's happening in Paris. And then, of course, the huge, the huge experience of Fiocruz in all these as well, in your experience in teaching and in production. And I think all that is linked, and it's not enough used. So in a project or in an action that we want to set up, it's important to take these different points to keep them in mind. And what we would ask to the people who have been invited from other countries on these issues is, for example, how do you see what you are doing in your country? How do you see an interaction with the institutes which have been presented now and with the actions that you have seen this morning and that you will hear on in more technical aspects later on? How do you see the interaction of the institutes where you come from or from your laboratory with these different persons? And there will be room for direct questions and meetings with the director or with scientists on Friday as well, in the afternoon. I don't know if you all will be there, but in the meantime, you know all the different scientists who are here, the directors who are here. Just go and talk to them. We're happy to interact with other institutions in the region. We have to, and it's a first way of getting into action with you. Somebody else?
Yeah, maybe it could be important to find a common axe, a common topic between all the institute, and I think one clearly emerging in my mind anyway, it's arboviruses transmitted by Aedes aegypti. I think it's one of the topic shows by Chile, Colombia, Brazil, French Guiana, Guadeloupe. I don't know for Montevideo if you have an accent, but anyway, your platform will be very useful.
So as you may have seen in my presentation, we do not have, as part of our mission, public health, because Uruguay in that sense is not suffering as other countries in Latin America, because we are more to the south, we are a small country, we have one natural separation with Argentina, so actually tropical diseases are not a big problem in Uruguay till now. I don't know what is going to happen in the next future. Right now our main causes of death are cardiovascular diseases and cancer in the in adults. In the newborns are also congenital diseases and other stuff. So infectious diseases are not a big problem. Of course, antimicrobial resistance is starting to become a problem, and we have to have a global plan for our country, but it's a little bit different than other countries.
However, I think, Carlos, you have also one very, very important thing. You have a very nice platform. Most of you are chemists, and you have a vast experience on that. And I think when we are thinking here in the platform, for instance, the discovery of a new products, when you talk about this, the vast experience that you have, and the startups that we are going to incubate, we can think also on one possibility to take one group that is doing a very nice thing, and you incubate there in Montevideo, just giving a little seed money in order the person can get some things, show if there is possibility or not to go on. This is one other thing that we can think.
Actually, that's the main idea. Our idea is to follow the model of flagship venture capitals. That is what they are doing. They are just giving a little bit of seed money to proof of concept. If the scientists go ahead and they show that the proof of concept is okay, then they can start developing the project. And of course, we are a small country with a limited number of scientists. If we want to pursue our goal, we need to work with other scientists from the region. And we know in Argentina there is a huge problem for scientists to get a job. The same is happening right now in Uruguay and in Brazil. So I think that we need, as a region, to develop that area just to improve our economies.
Identifying talents.
Yeah, of course. And allow us to dream that we can also be countries based on knowledge. And that's very important.
Question? Yes.
Here. So I'm from Chile, as you well know. So in fact, we're having the same problems as everybody's having. One of the, I would say, the public problem number one, lack of funding, lack of positions for scientists. We do have a minister, but actually that ministry was created this year with 0 additional funding. So you understand that that means nothing thus far. So I think we're having problems. From the scientific point of view, we do have strong groups in diverse areas that I know they're collaborating with Pasteur. Felix Rey is a strong collaborator from our people, for some scientists in Chile, as well as Montevideo for everything that has to do with protein synthesis. I would say the main problem we have is that we have a very small scientific community. Therefore, we lack platforms. So all our research is based on using platforms that are abroad. So we highly depend on collaborative efforts, and we are keen to collaborate with everybody. So we collaborate with France. I have 3 collaborators in France with grants. We were part of this USDEP, that was a big collaborative in the 6th call. We have money from NIH because we collaborate with US. If we don't collaborate, we die. So collaboration is a main thing. What is interesting, however, is that when I look at the platform you're building, I envision, as you say, that one of the biggest problems will be getting new and fresh funding to keep it alive. And I know that Europe has a lot of funding, as US, if you can win them, because they're very limited, but they're a lot of money, but they're very limited in number. So it's highly competitive. And I think the only way to do it is to join efforts. And to do science in Europe is even though you don't believe it, or in United States, it's much cheaper than to do science in South America, because we have to pay 2.5 taxes for transport. So everything is 2.5, at least in Chile, higher. So maybe that's something that should be considered. I love the idea. I would love that to be in Chile. But I don't think if we would be able to sustain it.
I know. So I kind of agree with some of the things that you have said, and in other side, I kind of disagree, because even when I realize that it is very important to have technological platforms and to have joint efforts to do better science together, and that's very important, I also think that one of our main problems in the region is our cultural problems. So we never tried to go further, and I think that's one big problem. I cannot imagine the reasons why we have 3 Nobel Prizes in Argentina and nobody was able in Latin America, outside Cuba and outside the vaccines that Fiocruz produced, to produce a compound that is being sold in the pharmacies for the people. And that's, why is that? And I think we need to think about that. And also, let me finish with one concept. We do not have enough money to support our science, and that is true. But also, if you go and see the countries that are investing the most amount of money. For example, Israel is adding 4.3 points of the gross incomes in science, but 86% of that money is coming from the private sector, and 50% is coming from foreign capitals. And we are not allowing in our region to the private sector to play part of the game that they need to play to improve our own science.
Right. I don't want to answer that question. I don't have the answer for Uruguay, but I think I do have the answer for Chile, because we did produce our own vaccines. That was before the military government.
Yeah.
And it was the deal of the military government with the US to actually shut down our production plants. So we became buyers.
But we're novel compounds or generic compounds. But in Uruguay, we are producing a lot of compounds. And actually, we have one of the biggest pharmaceutical companies in Latin America. But they are producing generics. They are not investing in research and development. And that's a huge problem. And actually, in Uruguay, that company that makes $2 billion per year, it's in a tax-free area. Is giving nothing to Uruguay, and we cannot do research and development in that with that company.
I wanted to say just some numbers. I am a French also, so I am now invited because I'm here. This first year that I moved here, after 34 years in France. And I was invited to many different sessions in the Club Santé and other things that France is entertaining here. The French cooperation is entertaining by the consulate. And I knew some very important numbers. For example, there is 600 French companies in Brazil. And I was in one of these French club, the Club Santé. And Sanofi and lots of other companies that are dealing with health, they don't have any thing going on with the academy. And you know, it is very important because there is a contradiction, because there are many things going on in Europe, R&D, research and development. And here it's very difficult to find university, because most of the research is being done in the academy. And there is very few people from the academy that is paid or funded by companies. It's a problem that we are not looking for them. We are not selling our work. Unfortunately, we are scientists, but we have passed in France also a period where we had to sell our stories and the things that we were performing in the lab to attract these people to give us money. I think that is difficult, but we have to learn how to do it. And if we can help, in Pasteur, we have here our director for innovation and industry partnership that is going to tell us how she's contributed and direction contributing to the network of a Pasteur Institute. So let's see if we can have some ideas coming from that. Isabel. Yes?
Thank you. If I can do a comment, and especially what Carlos is saying, it's not only in Uruguay that the big pharma, especially because they have the biggest money, are not investing in research. It's everywhere in the world. And this is why this type of discussion between different countries is so important, is you have the science and you continue to do the discovery, because without the fundamental research, there would be no invention and innovation. And then the second point is to know where they are putting the money. And Paola is completely right. In Latin Americas, you have mostly sales team. You don't have research, and they don't do R&D collaboration. But they are investing more and more now in everything that is linked to hospital. For example, I was with the new head of the business unit hospital of Pfizer, and there they have a lot of money to help them sell more, and then you have to see if it goes with your topic of research. But there are very fundamental research, especially on the topic that were presented this morning, like the epidemiology of Zika or the dengue and other stuff, or AMR, where you can find the money. So the point I wanted to make is, by starting with the science, then trying to know who has the money in the industry is a key element to move forward.
Thank you. It's time to conclude and to close this session. We have a meeting on Friday to finish the discussion. And to summarize, I have understood that you want to discuss about vector-borne diseases and emergency on Friday, yes? And Aedes aegypti? Okay.
Okay.
And now it's a coffee break. I
didn't
invite you to the coffee break.
I didn't get my picture. The picture. Where is the picture? I didn't get my picture. I need the picture. I need the meeting picture.
Tomorrow, before the coffee break in the morning, everybody stays here. Either we take a picture or we don't have coffee, right?
Tomorrow it's raining. Tomorrow it's raining.
Well, we are going to do the picture here. Here. You can have picture here.
We can go and take a picture now. No?
No, now the photograph is not here.
It's not. If you change the... I know it's...
Quel labo. So I would like to call here Thierry Lang, that comes from the direction of teaching of Pasteur Institute. And Thierry Lang is going to explain how the mechanisms that he is trying to approach now, how it is started in the RIIP. Thank you, Thierry, for that. You are going to talk alone, you and Carlos Battiani, or you?
Yes, we are going to.
You are going to share your? Okay. You know that you have only 20 minutes, both of you.
Lang. This one. And for the pointer, yes. This one? Yes. No, but for the light. Yes, the light. But I don't see anything.
Someone has a pointer? Someone has a pointer? No? So Thierry, show the versatility of the Pasteurien.
Good afternoon, everybody. First of all, I would like to thank the organizer for inviting us, Carlo and I, to present the teaching activity carried out Institut Pasteur International Network, and to talk also about the new project that we are currently investigating. On the Pasteur courses labeling project we are working on. Since the first course the first course of microbiology created 130 years ago. What is that? This one? Oh. Institut Pasteur has played a major role in the teaching scientific, and education became scientific progress and health. And we can say now knowledge transfer is still one of the Institut Pasteur course mission. Institut Pasteur has played is organizing actually 40 courses a year. 50% of courses are in English. The duration is from 1 to 9 weeks courses, and taught by 700 foreigners and French speakers, and initial education, master, and continuing for professional are offered for 900 students and 60 nationalities a year. The main point of these courses, we mix theoretical and practical courses. Out of the 40 courses, 20 courses are given on public health topics, such as medical mycology, tuberculosis, and vaccinology, and 40 courses are in the specialist master in public health, Pasteur CNAM. I would like now to emphasize the partnerships that we have developed with the French university, but also with other European universities, such as the University of Alicante. It means that all the students which are registered in Paris, at Institut Pasteur in Paris, are registered at partner university. And all the people who are following the Pasteur courses can validate European Credit Transfer Accumulation System, CTS, or doctoral school modules, and get university diploma. But I think it's very interesting to think now the university of the future, it will be maybe the Europe University, and a first pilot call dedicated to European University was launched in 2018, and 54 submitted project was submitted from 114 European universities, 24 countries, on research, training, and education. And very soon, last week, 17 projects were selected, and one of these projects was supervised by Sorbonne University, with which we have a partnership. We can see now the link between Pasteur courses and Sorbonne, and the link with the university in Europe. And now I would like to focus my talk on IP prize course offering in the IP Institut Pasteur network. And for that, we are working with the Pasteur International Network Association in order to identify the education needs, identify our global education trends, in order to establish an education plan for 5 years. And it means that all courses, some courses are given alternatively in IP Paris, Institut Pasteur in Paris, and also in the Institut Pasteur network. Since 2014, massive online courses which cover the main topics of biological science and public health are greatly increased. And currently we have 18 MOOCs, and which are presently diffused, and 4 MOOCs are recorded. These MOOCs are diffused on 3 platforms: the FUN platform, which is a French platform; Coursera, which is an American platform; Zwentang, which is a Chinese platform, but we have also established a partnership with the Université Virtuelle du Sénégal for the diffusion of MOOCs in Africa. And the slide shows you the massive online courses which are diffused in America, and here you have the the percentage of American registration in different MOOCs. This percentage can reach near 20% for the MOOC resistant to antimicrobial agents, and this percentage increase regularly, and we would like to increase this diffusion of MOOCs in America. And recently we have decided to create a specific diploma for the people who are following the massive online courses. And for instance, we create online diploma of infectious disease epidemiology of the Institut Pasteur, And for that, the students must validate MOOCs and succeed the certification of 4 out of these 5 MOOCs. And this project was made with the partnership of UNESCO, was sponsored with AMSUD, which is a pharmaceutical group, up to 300,000 euros per 3 years. And the next step now is to ask for the MOOCs diploma. We decided that the people who are following Pasteur courses of the International Pasteur International Network, we decided to offer an easy access to local region or regional university, and also the possibility to acquire skills in the field of infectious disease to get a diploma officially attesting their skills. It's the reason why we think it's very important to move on from unstructured to structured Pasteur International courses offering. And the next step is to create a Pasteur label for international education in order to adapt it to public health challenge, and which will certify quality of a common requirement for the courses taught by or in the Institut Pasteur International Network. And this project, I would like to show you all the people involved in this project from Institut Pasteur in Montevideo. Carlos who will introduce you this project. Spencer Schott from Seoul, Eshmi Louzir from Tunis, Fernando Alzana from Shanghai, Agnès Anthony from Roma, and all the staff from the Paris Institut Pasteur, from the Department of International Affairs, Jennifer Hurley, Kusai Raji, Mariana Lucaurani, Hans Hagen, Juliette Hardy, from the Education Department, Monica and myself, and also Kathleen Victoire from Pasteur International Network Association. Hi, Carlos, please. Thank you very much.
Terry, do you have the pointer?
No.
No.
When you were discussing there.
No, the pointer.
We decided, yeah, the pointer is over.
Ah, okay.
I thought that I was going to joke with you, but no. Let's see if you know how to do PowerPoint.
So, first of all, I would like to thank a lot both Thierry and Victoire and Kathleen, because actually they were the ones who did the job, who put all the ideas together, and they are inviting us to take or not their ideas and their work. So the main idea of this talk is trying to see if we want to go from unstructured to structured Pasteur International courses, and in the long run also if we want to have like an open university, and if we are willing to have the possibility to have our own titles coming from like a Pasteur university. So you are going to see along the presentation that the RIIP, the Réseau of International Institut Pasteur, or the previously called IPIN, International Pasteur, Institut Pasteur International Network, is going to be now called the PAINA, Pasteur International Network Association. And the difference, I was having like a long talk with Kathleen, the difference between RIIP, IPIN, and PAINA is just that in the PAINA, right now we have legal rights, okay? And each of our institutions or institutes are paying to be part of the RIIP, And now we have the opportunity to have our rights and to vote and to make our own decisions. So what I'm going to present is an idea that was around for the last 2 years or 3 years, and I think that we need to discuss and to take a decision. So right now, today, there are several uncoordinated individual courses that, for example, the Association of French Universities may have courses for PhD or Magister in Science students, and also other local universities or other research centers. Pasteur courses are almost the same. They are unstructured, they are from 1 week to 3 months. It depends on who is the one in charge, where the course is being held, so far and so on. The idea is that we can move from here, from absolutely unstructured courses to a more organized structure, thinking that we have to prepare our professionals and scientists in a better way. As I said before, at the end of the way, perhaps we can be talking about a Pasteur Open University and having our scientists and professionals from all of our countries having like a common curricula to be trained in a good way. So the idea is to develop courses that are going to be well recognized worldwide, as for example are the EMBO courses or other trademarks that are already in the market. As Thierry said before, Pasteur International label, Pasteur International course, will certify the quality and a common requirement for the courses taught by or in the RIIP, okay? And of course, this is essential for our scientists to be trained as we need. So education to research from research, open at the international level, high standard, both in the theoretical and practical education, intensive education, one course may be going from 1 week till 2 months, fundamental, advanced, and professional education. And I really like the concept that was probably 100 years ago suggested by Louis Pasteur, that another way to transfer technology is to transfer knowledge. And when we are teaching, what we are doing is a very important thing for our society, that is knowledge transfer. So what is a PEAK course, or what are we talking about when we are talking about the PEAK label? So it's a course that is going to be held within one of the international, the PINA network. It's going to be a course that has a clear presentation of objectives. It's going to be conference and practical with a quality ensured. Of course, each of the courses are going to have a clear evaluation process, a student selection process stated and well defined. The program of the course is going to be also defined, and the examination procedure is already known. We are going to be providing supporting material, and finally, we are going to have all the courses in a web page. So what are the advantages of having these international Pasteur courses? First of all, to assure the quality, and I think this is very, very important. So if one of our students are going to take part of the course, we know that they are going to be given a high quality course that is going to follow some rules anywhere where the course should matter. On the second place, I think it's very important to strengthen our own relationships. And we all know that the best way to collaborate is to have students going and traveling from one point to another one. To better promote the courses at local and regional university, and here we have the opportunity to use the already existed European credit transfer system And this is also very important for the students because all of the courses they are going to be taking part of are going to be creditable. Finally, it's going to be a way to consolidate our network, and it's going to be an opportunity to develop a new institutional and financial partnership with other institutions, like the Wellcome Trust, the Open University, and others. So how is it going to work? So when one of our researchers in one of our institutions are willing to develop a course, they are going to present the course to an education committee for labeling. The idea is that the education committee, that is going to be integrated by 3 members of the network, 1 member of the university, or member of the Association of French University, another member of the PINA, 1 member of the Education Department of Pasteur Paris, and 1 member of the International Affairs Department. So this committee is not only going to be labeling the course or giving the label, but it's also going to advise, and this is going to be like a going on and off proposal. Finally, the accreditation will be part and help for what I already talked about, the European Committee certification, and it's going to be part of the doctoral school module and university diploma. It depends on which kind of courses we are talking. Let's see about the timeline, when did this started and when we want to start with the idea. So during the last General Assembly, the idea was presented, I don't remember, I think it was by Spencer Short. And the presentation was voted by all of us in November 2018. Then during the spring in Europe, March, April, the peak level specifications were already presented. Right now, there is a presentation of what will be the board of direction of the PINA. The education committee is going to be created, and partner universities are going to be held. So the idea is that for 2020, we are going to be ready. We are going to be asking for courses to be presented, and hope some of them are going to be labeled. So along the road, the dream is to have not only Pasteur International courses as a brand name, as an important name for the students, but also a Pasteur Open University. So here is what you can see as a different step that we should go on. So from unstructured, different kind of courses, going to an open university where the master and PhD curricula, and this is important to note that this is just for PhD, the open university strategy, and then we can go perhaps a little bit further, arriving to a Pasteur Open University. So if we look at what is happening right now with the Open University, well, it's difficult to see on the screen, but then you have the Open University, you have a central hub that may be one of our research centers, where you have a senior research degree coordinator, you have associate research centers as a network, and each of them have a coordinator for research degrees. So the idea is to work as a real network. So I think that was it, and of course we are open for questions, and probably Kathleen and Thierry are going to be more or in best shape to answer. So I think we have time for questions.
Yeah. We have time for questions, 10 minutes. We started a little bit earlier because Luis was leaving, so we didn't open the ceremony, the session saying things about that. And I think that we are here with Thierry and Carlos, they would like to have your input on that. I have one question to start. For instance, if there are people in the network or abroad that are not in the network that have a very nice course, they can present the course to the committee for the directors, and you are going to analyze if it is a good quality course, if it is implicating different scientists, and you are going to have an agreement. And for that, the diploma is going to be from which university? For example, we are at the University of São Paulo, and if we here decided to do something, that's just an example.
Yeah.
How are you going to deal with that?
Let me try an answer, and then we will ask Catherine and Thierry. I think that right now the idea is that the courses needs to be held on one of the Institut Pasteur, part of the network. So right now the peak, I don't think is open for other institutions. But okay, so I did.
But, okay, it's not open for the other institutes, but what is the answer then? If there is, for example, a Pasteur Institute in Guadeloupe that proposes a course, what happens?
What happens?
With the diploma?
Okay, so who is going to give the diploma?
Yes.
Okay, so, yeah. Yeah, no, so probably it will depend a little bit on with which university we are working with. At the end, the idea is that the diploma is going to be part of the Pasteur, okay? So the Pasteur is going to give the diploma.
Yeah, but Pasteur is unable to give diplomas until today, right?
Yeah.
Yes.
Pasteur Paris side, there is a risk there that actually the Paris universities say we are not happy with that, and so all the grants we give to PhD students, we don't give them anymore. And so I don't know whether this would apply to, say, University of São Paulo or this kind of thing. So I think it's something that is very critical. We have to think whether we really want to go up to the Pasteur Open University, or we want to stop at a stage that, so it's quite political.
Yeah, so right there.
Why don't you
I agree, in the network it's different, but it's likely that this kind of issues may arise with other universities.
In our case, for example, we can do that in association with our university. Yeah.
So, Carlos, really nice the idea to hear as well. The question is maybe concerning the MOOCs, the massive online courses, and knowing that, for example, in Fiocruz, we do have the Open University of Health, or the Ministry of Health, of the system, right, of the universal health system, and we have 1 million students and thousands of courses. And it's for some courses, for example, we have Zika and chikungunya courses that was translated in Spanish and English. And some of those courses could be, we could exchange platforms. That would be very nice if we think that maybe one of the courses that we have in the what we call UNASUS could be offered in the Pasteur platform and vice versa. I guess there was some thought of, I don't know if someone here actually participated in these discussions a few years ago, but we didn't launch it. I guess this is a very good opportunity maybe to get back and do it again.
Yeah. Go ahead.
Yes, I mean for the MOOC, it's the reason why we have done a diploma specific for the MOOC, and the people who are following the MOOC can apply for a specific diploma. But I think the MOOC can supplement the presential courses on it. And in this project, we have done a specific project for the MOOC, but only for the presential courses, and just the MOOC can supplement the presential courses.
We have time for one more question. Savino.
Actually, it is rather a comment than a question. I mean, this discussion happened in the last meeting of the RIIP. And already it was discussed that if you're not agreed officially by the Ministry of Education of the country, you cannot just, put diplomas out of your institution because they're worth nothing from an official point of view. And this is a problem for the students rather than the institution. This is why, for example, in the context of the Mercosur, we have just made a PhD program for the whole 4 countries of the Mercosur, but the diploma will be issued by Fiocruz, which is agreed by the Ministry of Education, officially agreed to issue diplomas for PhD. Then I think that this is a very formal, necessary point to overcome. If you don't have that, I mean, you should stop because it will be with no end. It will be a dead end, actually, and this is not good for anybody, particularly the students.
No, I agree with that, but remember that I mentioned that it was in the long run, the idea of the open university. So what I think, for example, we all know that our biggest university in South America are already joined under the Grupo de Montevideo, Asociación de Universidades Grupo Montevideo, and most of us have already signed an agreement with the University of Bologna, where we can do PhD courses and have everything validated. And actually Brazil sent a lot of students to Uruguay to do courses that are being validated by both countries. So I don't think that is going to be a matter in the long run if the quality of the courses and the curricula is good enough. I think there's a lot of ways to solve that problem.
Well, I don't think so, really, because it's quite difficult to have official things from Ministry of Education in any country.
No, I know, but we are not...
This is under evaluation, etc., etc.
Yeah,
and this is, sorry, just to finish. And this thing is for each country. I mean, even if you have the agreement of Bologna, Bologna has nothing to do with Brazil or Uruguay. I mean, it must be issued in each country in which the PhD is being done.
Yes and no, because there are a lot of universities in Brazil that, under the umbrella of the agreement with Bologna, are giving titles, PhD titles, with different universities in South America under the Group of Montevideo. So, and on the other hand, I think that the master degree and the PhD degree, if it is quality enough, so it's not a professional title where the Ministry of Health of the Ministry of each country is going to have a concern. What we need is to have good scientists. And I think it's a very good idea from my point of view.
Okay. Thank you very much, Carlos. Thank you very much, Thierry. So.
Now we'll continue going to another subject that is also very important, not only for universities and schools and faculties, but also research institutes, that is innovation. Innovation in the mean that the knowledge that we generate in a science institute will go to companies. And that bridge between private companies and public universities or research centers is something that is very important today. And I'm very pleased to announce Dr. Isabel Buckle, who is the Executive Vice President of Technology Transfer and Industrial Partnership of Institut Pasteur, Paris. She has been here with us before. She had a chance to have a glance of what we're doing here. But today we're going to listen to her and understand what Pasteur Institute is doing in that direction. Thank you very much, Isabel.
Thank you very much for this nice introduction. Disagreeing with Paola Minoprio, which is always a challenge, because she opened this session saying that you are going to hear how Institut Pasteur Paris can help you. And for innovation, I've been extensively in Fiocruz and AUSP, and I could talk during 2 hours about what I learned from you, because what you are doing for innovation is tremendous. And therefore here, I'm going just to review how we are continuing to improve at Pasteur Paris the innovation aspect. And second, some time ago, we launched a global business development team, and we achieved the phase 1, and we are going to launch with the international network the phase 2. So I will spend the last part of my talk talking about that. And thank you for the introduction, because you use innovation in the right sense. Most of the researchers confuse invention and innovation. And one of the basic rules that you have so well understood at USP and Fiocruz is that we need the basic research, because the outstanding invention are coming from basic research, but some of them need to be helped to be transformed in a product. And at Fiocruz, you have the production facilities, you have it in Butantan, but we don't have it in Pasteur Institute, and therefore we need to transfer that. The second point is my hat for industrial partnership, because we need this collaboration, we need also the financing for all the projects, and the previous round table showed that we need this. So briefly, I will, like my colleague, show that technology transfer is part of a mission dedicated and stated by Louis Pasteur. One of the big difference now with the new management is that the technology transfer is part of the executive committee, which is a small committee around our new general director, Professor Stuart Cole. And the objective is really to develop the application of research and to increase industrial partnership. So one of the key challenges that my colleagues from the tech transfer here knows very well is that we are moving when there is an idea about vaccine or therapeutic or diagnostic, but on the basic side. And then there is a lot of experimental activities that are done and performed with often a lot of money spent by the scientists to do mouse model, even non-human primate model, and then it's a black hole. Why? Because often the key parts that are needed to go forward into a product are missing. And sometimes some experiments are done that are interesting, but not really necessary to transfer to industry and development and to an industrial proof of concept. So therefore, we are trying, it's the objective, but we are ambitious, to close this Death Valley by having a very early identification of pure fundamental research and talking with the scientists who are never talking to us to see if they don't have an application that can be interesting. It can be a buffer. It's not necessarily the big next vaccine of the work, but a good buffer coming from industry. I can tell you that it brings a lot of money to the academic partner who developed this buffer. We have also developed a new activity to work really with researchers to develop the different steps towards a real proof of concept, academic one, but that is ready to be transferred to a partner. And we are helping to find the necessary funding. Necessary funding is something I will come back because I think it's key for the international network. So this is the new organization where we have developed new incentive for the inventors. We have put together, with the help of Christophe Denfer here, an advisory committee on innovation that include pure scientists, but also people coming from the research, but industrial research. We have new rules for a scientist to work with industry in terms of framework, how many percentage he is able to work, paid by Institut Pasteur Paris, to create a startup or to work with an industrial partner. And this is something we include in our master research agreement with industrial partners to promote, for example, scientific exchange between industry and academy. We have set up a new process for the startup creation, and I know that it's particularly a topic for my colleague Celeste and for Fiocruz at the moment. And we have developed education program because, as most of you know, 90% of the startup fail because of educational training, because of the governance, and not because of the science. In order to fulfill that, 2 new activities in my department have been set up, and this is, there was a pointer. It's that, and I press here. No. Il est où? Okay, I see that. So, what is important, and I'm here to discuss further, it's, yes. Anyway, it's the pink one. The pink one is a new innovation office, and of course, we are part of the network, so we are sharing also these competencies with our people, but in USP and Fiocruz, you have this type of competencies. We have this new service dedicated to fundamental researcher, but we have these new people who are managing the innovation development, and they share in common something that they are all coming from industrial research. So you have somebody who joined in April, who spent 25 years in therapeutic industry, leading many groups for in research. Somebody in diagnostic who joined in March. We have somebody in vaccine that some of you knows already, Christian Gerk, who spent many years in Novartis vaccine. And we have Elodie, who is more dedicated to new technologies. Why this group? I give you 2 examples. You all know that there was a great diagnostic tools for that point of care that were developed by the CIBU and that applied for Ebola, Zika, and everything. The test is great. And it's not a criticism about the test. But many of you like the enzyme that is used, which is RT-LAMP. RT-LAMP to be used in Brazil, there is no problem because you can use that. RT-LAMP to be used in the rest of the world is a problem because there is a patent held by a Japanese company, and this Japanese company has only 8 sub-licenses to develop diagnostic tests. So for us in Pasteur and my team, it's an easy job. When the test is ready in the lab, we have only 8 phone calls to give to ask, are you interested to take the license? If the scientists would have chosen another thermostable enzyme, there are hundreds of company startups allowed to develop diagnostic tests. But that, it's not the job of a scientist to know it, it's our job. So this is one example. We have another example with a great potential product in therapeutic, and there is currently a non-primate test being done. And the study that has been developed has been developed with only scientific, pure scientific parameters for the protocol. If you put that in the hand of a professional of therapeutic, a researcher, it would have become what we called in industry a pivotal study, and here all our professionals of vaccines know it. It would have been less experiment, less expensive, quicker, and all the information needed to start the development of the vaccine. So for you in Brazil, it's obvious, but for the rest of the international network, it's not obvious. And this is why there is this department. Funding is important. Creation of the startup is important, and I'm here to discuss how we have put this process not only to have the business side, but you have also the educational side, because a good scientist can or cannot be a good CEO of a startup. And education. You had a lot about education. We have developed at Pasteur Paris a lot of educational program from undergraduate to permanent scientist for this technology transfer knowledge, and this is listed and I can develop further. What about the RIIP? International network. The international network, the phase 1 is implemented, and we are moving to phase 2. Phase 1, we have hired at Pasteur Paris somebody called Antonio Borderia, that some of you know already, who is the coordinator and the point of contact with Pasteur Paris for all activities related to the network. He has regular meeting with Kathleen and the team, and his role is really to dispatch the needs and to find the best expertise to develop projects. The idea is really to have this global BD team that was launched by some of you. So it was Institut Pasteur Montevideo, it was Institut Pasteur Korea, Institut Pasteur Shanghai, and Institut Pasteur Tunisia. And together we put a mission statement, and the idea was really to improve the technology transfer and to develop the critical mass we have, which is unique in the world, by sharing tools, training staff, making on-site and online expertise, combining technologies and scientific offers, and developing opportunities. We presented to the international network several times this BD Alliance team. Phase 1 is done. Many tools have been shared and templates. Many training have been done everywhere. The last one was in Pasteur Montevideo, where there was a course on creation of a startup some weeks ago, and Carlos can develop how it was done, but I think it was a great success for South Americas, and making on-site and online expertise, like Institut Pasteur of Iran asked us to make an audit of their facilities. But once again, it's not only from Pasteur Paris to the rest of the world, it can be also from Fiocruz or USP to the rest of the world, part of the international network. Phase 2 is something that is key, and we just exchanged with Antoine Talarmin about that, is to use this critical mass for project collaboration, how to map it, how to think big together, to develop opportunities between teams, and combine these technologies and scientific offers. Oops. The idea of this international course is a good example. You had in the previous roundtable, okay, we can put together nice scientific programs, and then who finances it? The international companies don't put money in research anymore, but they have a lot of money in other places. Most of the research part, and of course there are exceptions, is put in startup companies with other venture capitalists or with academic centers. But the big industries have money in other parts. They have money in human resource. And in human resource, training is key. So to find money for a brand of training with the international network is for me something, and for my colleague for innovation, something easy to sell, because they need experts, and they are looking for experts within academia. There are money in everything that is the medical affairs and clinical of the industry, and they have a lot of money. So having the network to follow up on phase 4 for their dengue vaccine, for example, or putting money to have research collaboration to put more argument for some of their products that are already in the pipeline is something that is interesting for them. So it's something that I will be very happy to leverage and discuss more. And again, I feel really like a small person compared to the experience that we have here, especially in Brazil, when it comes to innovation that has been implemented for years and years and years in this country. So this is just a picture of the LATAM workshop on PhD to entrepreneur. This is what I told you. And this is a team that has been hired in Pasteur. Thank you.
Advice, although you were saying that you are just starting, you did a lot so far, and I think it's changing a little bit the way Pasteur network is thinking about how to handle such important issue. Just to give an announcement, I'm reserving some time tomorrow afternoon in another building to have a chance to have you and maybe Thierry Long to talk with people here from Fiocruz and our university, to have an opportunity to talk about this very important subject, education and innovation, in a more informal way with you. So anyone who would like to discuss more with Isabel questions of innovation should be here on Friday, third floor in Alcani. That's International Affair Unit. It's a building close to the Pasteur Institute.
It is going to be in the Pasteur Institute office.
Okay. You can give this.
It's going to be in the Pasteur Institute office, room 9, third floor, Alcani. So you have a table there to discuss with people from the university. You and people that are interested in innovation and teaching can come there. The provost for teaching and innovation is going to meet you there. Thank you very much, Isabel. It's me. Okay, now we are going to change a bit. We are going to pass, unfortunately, the Vice President for Innovation and Production of Fiocruz cannot join us. He was called in Brasilia for very quickly, and he could not come. And he asked Milton Moraes, that I talked to this morning, to give the talk for him. And Milton is also part of the committee innovation of Fiocruz, and he's going to talk about the Fiocruz Intramuro Innova program to accelerate science and technology for health. Thank you very much, Milton, to be here and talking like this, preparing very quickly your talk.
Thank you, Paula. Well, hello again. It's a pleasure to be here, and well, it's indeed. Let me just do this. Yeah. Well, actually, it's a great pleasure to be here and part of this innovation roundtable and discuss some of the new initiatives that we have in Fiocruz that we are just discussing for the past maybe 3 years, and we actually selected the projects I mentioned briefly earlier this morning, and I will try to present, but I had a very easier job now, since Savino had a really nice presentation earlier and introduced most of part of the things that I was planning to introduce to you about FieldCruise, so I'm just skipping all of that, and I will concentrate on the innovation program. And just after Isabel, it's also very easy to discuss what innovation is and what is the idea that we are planning to introduce in FieldCruise. So let me just start here. Okay, this is the innovation program at FieldCruise. And basically what we had is that we got funding from the Minister of Health, from the Secretary of Science and Technology in Health, and then we started to select, well, we have an open call, and in 4 different areas. I'm going to show which areas are that, but it's important to mention, and this is the first slide that I presented earlier today, is that we had 35 million reais to this program. So actually, the governance of this is associated with 2 vice presidencies. It's also very important to mention that. We have the vice presidency of production and innovation in health, and the vice presidency of research and biological collections. And 2 of these calls that are the more, let's say, fundamental. How does that work? All right. Is that okay? I'm gonna use the pointer here. Yeah. No, that's okay. So basically, what we have is that these 2 calls here that we call knowledge and knowledge young talents are associated with vice presidency of research, and these 2 calls, that is ideas and products, are associated with vice presidency of production and innovation in health. So basically, what is the, well, Isabel just mentioned that actually we need basic science, we need applied science to develop new products and to have innovation. So this is the idea. We need to actually foster good ideas and good science in FieldCruise, and for that we have this call that is knowledge, which is basically trying to fill gaps that could improve better understanding of very fundamental questions in health. And obviously, we selected a group of young talents that are mainly researchers that are, actually get into the public service in FieldCruise for in the past 7 years. And then we selected them to actually also provide a specific call and some money for that. And well, and the other 2 calls, one is what we call products, and products is actually a very specific type of a call that's basically we have a very mature project, which means that we already have the proof of concept. So we have preclinical work on a vaccine or a diagnostic kit, for example. And well, on the other hand, we have the very basic and fundamental issue that could be highly disruptive, which is the we call ideas. So basically, the ideas is when we have, it's just like, let's say, the grand challenges from Gates Foundation, for example. We have a good idea, and but we don't have proof of concept yet, but we're gonna, just fund for a year or 2 and see how it goes. And whenever a good result is obtained, we're gonna go ahead and keep funding that. So this is I just said. So it's important to mention that we had submissions for Fiocruz community. The ideas, for example, that is this call here. We had a part of the submissions that the principal investigator did not have the PhD degree. It's also important that we are also fostering new ideas coming from the community without the need to have, formal and trained education. It's also really nice. And we also try to grant it a more balanced gender in our call. So we allowed that women with children could apply with more 2 years, one year per children, and during for this young talents call, which is also very interesting. So the submissions were more balanced in this specific call. So what are the highlights? And well, this is in Portuguese, but I guess all of you will see that concerning the grand areas and fields, we have health here, right? But we have environment, we have inflammation, and we have obviously omics, we have surveillance, different areas of specific subjects like immunology, parasitology. We have also information, communication, it's very important. And what about the technologies? Well, we have apps. It's very important. So digital health is one of the big projects that we have. We have a lot of, again, computational work, and we have also novel technologies, DNA editing technologies such as CRISPR-Cas9, for example. So if I could combine all of this in access, I could say that one big area, obviously it's not limited to that, it's just to provide you a glimpse of what we got in this calls. We have digital health with apps, games, e-learning, big data, and geolocation, for example, as main areas in different research projects that we are funding. And on the other hand, we have new technologies for health. That is basically 3D printers, next-generation vaccines based on DNA technologies, and diagnostic tests, CRISPR-Cas9, and new omics, also for surveillance. So we are, all of that new, knowledge will be dedicated to better solutions in treatment, surveillance, and diagnosis. Not only that, but only to provide what we got in most of the cases, right? And we are dedicated to what? Obviously, our tradition is neglected diseases, but it's also important to mention that we have now projects in cardiovascular diseases, neurodegenerative diseases, and also cancer. So we are also doing the epidemiological transition, which is very important to mention that we are going in this direction as well. So, and the other thing is that obviously we have this legal framework in Brazilian since 2015, and it allowed us also in Fiocruz to provide a new policy and new regulations towards this legal framework in Brazil. So we now have this novel policy, and we are trying to work better towards what comes next. So the idea is that we have the innovation program, which is actually how we are funding science in different areas, and what actually in how we try to gear the projects towards better results, and obviously understanding what are the needs from the Minister of Health and what are the needs from the society, right? So we have this portfolio associated with great demands that we have in the Minister of Health. But the point is, obviously, Fiocruz, as I mentioned earlier today, and Savino also mentioned, we have to plants and producing biologicals, producing vaccines, producing diagnostic kits, producing. So most of these ideas, generally, and the research, were funneled towards these development centers and institutes. But now we still need to provide a little bit more of that, but sometimes we don't have enough investments to do so. So we are trying to have this, what we call the regulatory areas, trying to promote, for example, let's go, to promote scientific and technological entrepreneurship, provision of technical services, facilities, which means, and trying to doing some internationalization for research development and innovation. So let me skip this one. This one is in English. Basically, we have two main axes in this, what we call the program to support entrepreneurship and Fiocruz, and it's being now discussed in our higher leadership council that we have in Fiocruz. And basically, the idea is that we have the possibility in Fiocruz to have partnerships for development, to provide services, to share infrastructure, and also funding, and also associated with the development of spin-offs and startups, either in pre-incubation and incubation levels. And the idea is that actually we can have in the future either spin-offs and startups in an incubation in our institution. And the other thing is also, Isabel mentioned, it's very important, is that we have to change the culture for scientists and for the community in general in Fiocruz concerning what is entrepreneurship and how we do that. And so we have to improve the dialogue with the society. We have to increase the number of workshops and courses in this and training. I was talking with Luis earlier today, and he was sharing with me a very nice and successful experience here in USP, and I think that we're gonna follow that and try to organize also courses in this direction. So the general idea is that we could, with these new strategies, try to improve the awareness of the scientists in our institution concerning what is important to protect, what is important and how we're going to do it, and understanding a little better the ways of how they should pursue this path. So, and this is my last slide. Basically, so the great, putting all of that together, the innovation program, is that we have now the calls, and we have the projects, and so we are funding good science. We are trying to follow that up, changing a little bit the way that we are evaluating the follow-up of those projects. And we expect by that in the end, we have products that could be developed by one of our institutes associated with development and innovation, such as Biomanguinhos and Farmanguinhos, which is the units that produce pharmaceuticals and vaccines, but also that we could have, angel investments to startups and spin-offs in our own institution. That's it. Thank you very much.
Thank you very much, Milton. As well as with Pasteur Institute, I'm very glad that Fiocruz is now giving a special emphasis to innovation in-house. And from the side of USP, I'm really anxious to share experience and do things together. That is very important for all of us. We have time for one question, because before the next presentation, someone would like to address a question to Milton? Or at the end of the session, we'll have also some time for final questions. If not, okay, thank you. Let's move, and now I'm very also glad to introduce you, Dr. Luis Catalani. He's from USP, and he's the present director of Innova USP, which is also an initiative of the University of São Paulo towards improving innovation in our environment, and that's a building that we will going to visit tomorrow, and a Pasteur platform is also insert in this initiative. So thank you very much, Luis, and I'm very thankful that you could come here to talk with us.
Thank you. Good afternoon, everyone. Thank you for giving me this opportunity to present Inova USP. And by the people that were before me, I can see that was such a right decision to bring Pasteur and scientific platform Pasteur USP to Inova USP, as I will try to present you now. And especially what was presented by Isabel, I think Pasteur USP is the right partner for us to be at the Inova USP, as I'll try to convince you. Well, let's talk a little bit about innovation at USP. USP, as you probably heard or read in your booklet, is an 85 years old university. It was formed from 7 schools in the beginning. From those, 6 were professional schools, and most of them devoted to technological professions. So we do applied science from the very beginning. We are very good at that. Actually, to give you some numbers, nowadays is very impacting to talk about unicorns. So Brazil has today 6 unicorns, and eventually this year this number will rise. But today we have 6 unicorns. From these 6 unicorns, 4 were bred in this university. Another number, from these 6 unicorns, they are owned by 15 CEOs. From these 15 CEOs, 10 of them were graduated from this university. So we are really not only a very basic science university, but also innovation type of university. And where and why Inova? Inova is a very new initiative. It's like 1 to 2 years old only. Pasteur was here before, but Pasteur was here actually before Inova was born. And we have, as every other university in Brazil has, an agency, a tech transfer type of office. In our, do we have a pointer? No. We have an agency, USP Innovation Agency, which is a tech transfer office, an IP office, as every other university in Brazil must have. And, does it work?
Yeah, the last one, this one. Last one.
Oh, great, okay. So that's AMSUD is our tech transfer office. And what in the last years we decided to do is to divide innovation activities in the university into twin brothers. And Inova USP was born as a center of innovation to try to organize the activities of innovation and give to AMSUD only the tasks dealing with the law and the organization of other issues in innovation. So the 3 partners here, we have, you met this morning Professor Silvio Canuto. He is our pro-rector of research. Together with Inova USP and AMSUD are the main actors in innovation in the University of São Paulo. Our ecosystem, though, is much bigger. I can tell you, for instance, that we have 5 incubators in this university. As you probably read in this booklet, we have many campuses, 6 campuses. We are 48 units, schools, 4 hospitals, 4 museums, and so on. So we are really a very comprehensive type of university. We have areas of all kinds somewhere in one of these 6 campuses represented in this university. So these incubators, 2 of them are in this city, and then Supera is in Ribeirão Preto, Exalc is in Piracicaba, and Unictec is in Pirassununga. We also have some think tanks within the university, in the Innovation Observatory, for instance, and then the Center for Entrepreneurship and the Center for Technological Policy and Management. So those are groups that try to think innovation and organize innovation within the university. We also have some other activities like EMBRAPIS. I'm sorry, there is a misspelling there. EMBRAPIS are unities that are federal core innovation centers. We have in the whole Brazil, do you remember the number, Luis? It's like 15 EMBRAPIS or something like that. 30? And University of São Paulo has 4 of these. And then we have 11 CEPIDs, which are the similar state core research and innovation centers. Then we have 2 other innovation centers here in the area of health and on the hospital. So what is our strategic vision here? Is to make Inova USP a hub for innovation for the partners within the university and also for the partners outside of the university, that we can have one place to find and to connect people that wants to do some science with the university. Okay, so we are not only a place, we're not only an address, we are an idea that connects the whole university, all the 48 institutes. Of course, we as an individual that is above all this institute, the idea is to use Inova USP as a multidisciplinary type of approach for every problem. So if we have, of course, approaches that are monodisciplinary, Inova USP will take this partner and will lead to whoever is the right partner to develop the innovation needed for that problem. However, what we are looking at is to a more broader, more multidisciplinary type of approach. We are trying to arrange a more innovative type of a model. I think who was before me, it was Milton. Is your name Milton? You're talking about these new laws in Brazil that allows a more effective type of partnership with industries, with private enterprises in all kinds. And so we need to modify, of course, for you, people from different countries, maybe this is not important, but in Brazil, this is a very, very special moment that we are living now. So Inova USP is out leading the transformation of all the process within the universities to bring this to a more effective type of integration with our partners. We also are set to mobilize teams from any of this part of the university. So we have 42 different schools plus some other institutes. So we want to be the place to give access to all of this infrastructure and all of this expertise within the University of São Paulo. So, of course, for this, we need a very efficient and transparent type of management. We are ready for giving priority for startups. Remember, we are university. Of course, we want to provide new knowledge, provide partnership with industry, with private organizations, but we are an education facility. So the one major goal of Innova USP is to give the opportunity for our students in every level, undergraduate, graduate level, postdoc level, opportunity to be an entrepreneur, to learn to get in contact with all this type of activity. So we will be ready for tutoring any type of students that would come in contact with us. So then, of course, you know that Pasteur USP is there. Who else is there at Innova USP? Let's just give you a tour. We have 4 major initiatives at the moment in Innova USP. Innova USP was born out of these 4 partners. Besides Pasteur USP, we have Patio, IRIS, and S2B. Patio is a group of scientists. They are devoted to digital technology. We have people dealing with gaming technology. I mean, serious gaming is not regular game, but serious game, user experience, industry 4.0, and artificial intelligence and machine learning. And this S2B, which is Synthetic and Systems Biology Center, this is a group of researchers that came, they were originated in the bioenergy effort from this country. You know that Brazil has a very long story of using biomass to transform into bioenergy. And this group started that, but today they are more open to different crops and use of biomass to develop new chemicals for industry, for new chemical inputs for industry, and so forth. So the next and the last one is IRIS. This is a very specific group of people. Their task, their challenge is to really dig major problems, more challenging problems, more disruptive type of problems that can be organized by Innova USP. So we receive from, especially from major corporations, inputs of very hard problems to take on, and they try to organize the way to attack these problems. We also have in-house an undergrad course. It's called Molecular Science course. This is a 25-year-old course in this university. It takes freshmen from any course into one single class, and they take science as a major, but this is a very special type of people because they come from every unity, every different courses here to join into this multidisciplinary type of course. And so those are the guys that probably will be more involved in all the activities of Innova USP. We also have a makerspace projected that. It's, we hope to be a very advanced type of a prototyping shop and laboratory in order to support designing and building prototypes during the innovation process within the house. We also have a co-working space called Cosmos. This is run together with the USP Center for Entrepreneurship. That's where we hope to put our students, our postdocs, and give a tutoring into let's say the language of innovation and entrepreneurships. And last, I guess that's the last one, is the Computational System Biology Laboratory, which is run by Professor Elder Nakaya. He's here, he's reading. But is a lab that is devoted to development and use of computational tools and to support not only Pasteur, but also S2B in other groups in the university. And then we have also a Center for Artificial Intelligence and Machine Learning. This is part of the PATU, the other digital groups, but they are organized into a Center for Artificial Intelligence, and this is a big thing now in this stage. And there is a lot of investment into artificial intelligence, not only here, but in the whole world. So those are the actors of today. We hope to be able to bring more of these activities, all of them in the area of innovation. And I guess Pasteur USP has a very special place in this environment. So that's my last. Thank you.
Thank you very much, Lick Catalani. And we have time for a couple of questions. Anyone would like to address a question to Professor Catalani? Okay.
To what extent your buildings are made to allow these different groups to interact? Because I think one of the issues in innovation is also to break the silos. And so you have this number of groups, but the question is, you know, how did you design your buildings to ensure that people will interact?
Yeah, okay. I have maybe two different questions, two different answers for you. Innova USP today has those groups there. But as I said in the beginning, Innova USP is much more than that. So I want to build a place in the university. We want to build a place in the university where we can bring people from every other school and interact with the people there. So not only the problems that are run from these people that I would like to be able to run initiatives or organize initiatives that goes much beyond the scope of the Innova USP. So I'm sure we will have an easy access to all of these people inside the house and ways to make them interact with each other. We have done this already, although we are still not fully organized for that, but they are already done. But that's not the idea of Innova USP. Innova USP is much beyond what is inside this place, what is inside this building. You see, this is a very nice building. I used to say it's a very sexy body, but has a very high soul. It's much interesting than whatever is inside. It's much beyond that.
How do you structure that course in order to fill all the needs?
Okay, that's a very interesting question and very interesting story. This course was put together 25, 27 years ago. Luis, help me here. I think it's 27 years, I guess.
Yeah.
So this was intended for a group of students that wants to be to get a more strong scientific background. So it was organized in molecules. That's why it's called molecular science. So it was represented by biomedical sciences, biology, mathematics, physics institute, chemistry, pharmacy, I guess, medicine. So all of these, there were courses from all of these schools put together to give a major in science, which means a very strong in scientific background, but not direct to any professional, right? So they had a problem at the beginning actually to get a bachelor, because you cannot get in Brazil, you cannot get a bachelor in science. You have to have a bachelor in something. So most of them come back to their original schools and finish their course to get another bachelor that can be used for professions, for instance. And as the story goes by, this course were on, and we could see that people from, students from the most different areas came together. Students from philosophy, students from social science, students from education, and as this course was put together, we had to accept them all. And actually, this was very interesting because you create a very distinct type of people. And you can see the story of all of them. They became scientists of very different pedigree. It is very interesting. Some of them came back to humanities, but now with this very distinct background, they do all kinds of different things. I have in my institute, I'm a chemist. I have in my institute, one of these students became my professor in my institute. He does astrobiology. I guess that's the only astrobiologists I know in Brazil. And so I found this a very successful experiment. And what we are doing now is taking this course and giving a new input in direction to innovation and more of applied science. And they are receiving this very strong. They are interacting all these people. I think most of these activities in Nova USP have already received these students to get. So that's probably a second step in the evolution of this course. It's very interesting.
Okay. Thank you very much, Alik, for this interesting presentation of Innova. So, okay, we just now go to the last speaker of the day. It's our colleague from Pasteur Paris, Christophe Denfer, which is the Director of Technology and Scientific Program in Paris. And he is going to talk about accelerating research through access to technological platforms. Thank you very much, Christophe, for being here with us and to share these initial steps of our scientific platform.
I found it. So thanks, Paola. As you saw, I was anxious to speak, especially because Paola told me that I should not speak too long. So at Institut Pasteur, I'm in charge of two things. I'm in charge of all the core facilities, but I'm also in charge of incentive programs that we have to promote new ideas, new talents. I won't talk about this, but if time is available at the end of the talk, I can answer a few questions about our incentive programs. So I wanted to start this talk by this quote from Sidney Brenner. So you know that Sidney Brenner is a Nobel Prize winner. And he quoted that progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. And clearly, we all in our laboratories rely on a number of techniques and technologies and on accessing these technologies. And in the past, let's say 20 years, these technologies have been more and more provided by core facilities. So what I'd like to do today is briefly tell you about how we have organized our core facilities and technology offer at Institut Pasteur in Paris. And then I'd like to, in my last 2 slides, discuss how we could improve technology networking within the Institut Pasteur International Network. Because my impression, having stepped into this position about a year ago, is that we are lacking coordination in terms of technology within the network. So, at Institut Pasteur in Paris, our core facilities are located in 2 centers. A center that is hosting technological platforms, and a center that is hosting core facilities for animal research. And we have 2 types of core facilities. We have units for technology and services, and we have technology platforms. The main differences being the size. The unit for technology and services are larger, and they also develop more technology research than do the technology platforms. And so in total, well, so the missions of these 2 centers are actually to provide resources to the research group that we have at Institut Pasteur. So they have to structure and organize the facilities, provide upgrades of instruments. They have to provide advanced technology and animal resources for biological research. And most importantly, they have to provide services at the highest level. And this comes with speed, and all scientists are really anxious to get their results from the core facility as fast as possible. And clearly, that's an issue in core facilities, and quality. And they have to respond to user input and satisfy them. We see that working in a core facility is different from working in a research lab, and this is something that we have to take into consideration when we deal with human resources aspects. So our core facilities also have a mission of research. They have to develop or provide the next generation of equipment and methods. They have to interact with other institutes of technology, engineering schools, industry partners, and they have to collaborate with our research units to ensure the development of new methodologies. They have to ensure technology watch and support innovation. And finally, they have a mission of dissemination, dissemination in the form of training of our scientists in technologies, but also networking with other infrastructures and core facilities. And I think this is one of the aspects that we would like to develop at the level of the Institut Pasteur International Network. So currently, these centers, they employ 209 people. This is about 10% of the people who do actual research on Pasteur campus. And most of these people are paid by Institut Pasteur. And this is actually indicating that for Institut Pasteur, these core facilities are central to our research. And so they provide services in all types of technologies you may think of: genomics, proteomics, flow cytometry, imaging from light microscopy to cryo-electron microscopy, protein biology, so nanobody discovery, protein production, molecular biophysics, and structural biology. And we have screening platforms and a facility for microfluidics and organ-on-chips. And on the side of animal research, we have a facility for genome engineering and parasite production. So, I have this list of new technologies that have been implemented in the last 2 years. I won't go into the detail of that. We have provided long-read sequencing, new imaging modalities, including light-chain imaging and cryo-EM. We have a number of single-cell pipelines that have been developed, And we have developed also organ-on-chips, and I will come to that, and methods to develop new animal models using CRISPR-Cas9. And these developments are really reliant on access to funding from Institut Pasteur, but also other funding sources that we have in France. So I'd just like to give a few highlights of recent developments that we have done. One thing we would like to promote is actually a switch to autonomous usage of the technology, and this is something that we are doing in the field of sequencing. We've developed courses, online courses, so that people can actually train on how to use different apparatus that are necessary for sequencing, and people can then actually access a next generation sequencer independently, and this is to the benefit of the speed at which the projects are performed. Another thing that is important to us is actually to allow our scientists to explore biology at different resolution. So I mentioned light microscopy. We also have a lot of technologies in the field of structural biology, but in the recent years, you know that cryo-electron microscopy has started to provide a new perspective on in structural biology because it allows only through imaging to define the structure of proteins and assembly of proteins, but also it also allows to integrate these structures into the context of the cell. So in the past few years, we have started a nano-imaging initiative with the acquisition of 4 cryo-electron microscopes, including a Titan Cryos, that allows for single particle imaging and cell tomography. This is hosted in a new building at Institut Pasteur, and it's been permitted by a significant investment of the institute, but also through funding of the government. Finally, we have set up a new platform for biomaterials and microfluidics, because we think it's very important to allow our scientists to access these technologies. And in particular, we are promoting the development of organ-on-chips in collaboration with the company, because this will allow bridging research with cell cultures, with research with animals, and we think this is important. And we have developments around organoids, disease on chip, etc. So importantly, I'd like to mention that all these facilities are open to external scientists that are in France, but also members of the RIIP. And there are a number of collaborations that go through this facility within the international network. So it's part of our strategic plan to continue developing technology. I mentioned the nano-imaging initiative. We would like to strengthen or develop metabolomics, develop single-cell analysis pipelines. We have plans to set up a new building to study interaction between host, vector, and pathogens. And we would like to strengthen our screening capacities to develop new therapeutics and possibly new diagnostics. So for the next year, we will create a metabolomics core facility. We will create a facility for humanized mice, and we will reinforce our screening capabilities. So I mentioned that one of the missions of our core facilities is dissemination. And so indeed, they are developing online courses, technology seminars, technology workshops. They participate in a number of courses that were presented earlier. They interact with the Pasteur network, and they interact with a number of university teaching. And importantly, they host teaching courses that are related to core facility management or how to work in a core facility. So I'd like to finish by now that I have described this offer, try to see what is the status of networking in the field of technology at the level of the international network, and suggest how we could improve that. So obviously, I think you will all agree that access to technology is an essential component of our research programs. But what I think is that technology networking is poorly organized within the Institut Pasteur international network. So from our perspective, there is occasional use of our technology platforms by teams of the international networks and vice versa. This is obviously influenced by the nature of the technology. If you want to do live microscopy, you need to have a microscope near you. You cannot rely on a platform. But for proteomics or sequencing, there are opportunities for interactions. There is also an impact of legislation. Some samples cannot travel easily, especially with the Nagoya Protocol. What we note is that interaction between institutes of the network and technology platforms are promoted in the frame of our transversal research projects or other collaborative projects. So funding is key for these interactions. And this is something that we actually discussed in the previous session, that to collaborate, we need funding. And finally, there are some teaching actions. A course was organized with the Institut Pasteur of Athens on transgenic technologies. There is a quantitative immunology course that is organized in collaboration with IP Hong Kong. And finally, I think one of the difficulties we have in the network is that the needs are very varied. The needs of Institut Pasteur in Montevideo are totally different from the needs in Guadeloupe or in Guyana or in Dakar. And this is something that we have to address as a rule to see, what can we do? So some institutes are interested in technology benchmarking or training. Some institutes are interested in support for implementation. Some institutes are interested in accessing the technology. Other institutes do not have the resources to host highly cutting-edge technologies and would like to see the development of ad hoc technologies. So I think that we should think about an Institut Pasteur technology network. This could be based on the example of alliances for the development of core facilities. So I take the example of core technologies for life sciences, which was created by Spencer Short and is now led by Patrick England at Institut Pasteur, and bring together people that work in core facilities to try to exchange on developments, on a way to organize the facilities. The same is true for Core for Life, that bring together different institutions in Europe that have a large setup for core facilities. And I think the actions of this technology network could be on communicating on the Institut Pasteur International Network technology offer, on promoting the involvement of core facilities in externally funded grant applications, on sharing good practices for running core facilities, such as steering committees, charters, pricing, human resources, on supporting the establishment of new core facilities, on sharing methods, providing technology trainings, either through theoretical and practical courses, short-term visits, and helping with technology benchmarking, and finally promoting interactions with technology providers. So I think there was this action at the level of the international network around courses and having a structure courses at the level of the network. And I think we should also structure our technology access as another example of shared action at the level of the network. So I leave you here with that slide for discussion. Thank you for your attention, and be happy to answer questions.
We have time for 2 or 3 questions. Luis?
Well, impressive policy to establish such facilities. And I'm especially interested on the cryo-IM facility that you have already there. Just a question, it's already working, functioning, and is it accessible for partners like us at the University of São Paulo?
So,
that's a personal interest, because I'm having problems to find a good place to do it here in Brazil.
So it's not fully operational yet.
Yeah.
So 2 of the microscopes have been received, the Titan Cryos and the Glacios. We are hoping to, make them accessible to projects before the end of the year. And then it's been financed by funding from the French government, as well as funding from Pasteur. And it's open to external groups, French or non-French, with a selection process. And clearly, the idea is to allow single particle imaging and structural biology, but also develop cryotomography to really be able to image proteins in the context of the cell.
And how is the policy of access? I mean, scientists from Pasteur have it free access, or everybody has to pay different prices and so on?
So use of the microscopes will be
for any equipment, not
yeah.
only
So for any equipment, we ask people to pay. So it's not free. So external people, when they are academic, pay a slightly increased fee, and the non-academic groups pay an ever increased fee. I think it's important. So nevertheless, it should be clear that for academics, whether from Pasteur or from outside, it's significantly subsidized by the institute. And clearly, access to technology has to be subsidized, because there is a lot of investment in terms of equipment, and most of the labs could not afford to access this technology if it was not subsidized.
Just one question before. You wanted to make your question before?
No, go.
Yeah, it's just to know, for example, as you know, we are investing a lot on capacity building in the different Pasteur institutes in the RIIP. I would like to know if there is a possibility that, for instance, we apply to send someone there to be trained how to use these equipments, and if so, how long is the time to get all the formalities to get in France and for these people?
So that's one. So it's always one issue. I think that we can host. So for instance, to get someone at Pasteur in Paris, you need to apply 3 months in advance, so that the person is screened and blah blah. But most of the time there is no restriction, and so I think it's some paperwork. You've been working there, the administration there, but it works relatively easily, especially when there is no issue about salary being paid by Pasteur and so on. So most of the time there is no issue in hosting someone. I think when I mentioned this idea of an Institut Pasteur technology network, is also we need to find ways of funding this type of visits, so that someone could come to Paris for 1 month to learn how to use different type of equipments and come back or try an equipment. Yeah, we could. Yeah, we can use that. We can think of applying for European funding. That's not necessarily working for Brazil, but for other other institutes in the network, there is a potential to find funding at the level of Europe that could, help supporting this interaction and exchange between the different institutes, especially in the field of technology training.
Okay, thank you, Christophe, for very, very clear presentation. But you didn't mention any things about the bioinformatics department. Yeah, there's a lot of people work on that. Maybe you can...
Yeah, there are two things I didn't mention. One is, so we have a Fab Lab, so in addition to these platforms, which is also to, helping for the development of prototypes. And as part of the previous strategic plan, Institut Pasteur hired 40 bioinformaticians, which all together form a hub. And I think they can support all the research group in Paris for different bioinformatics projects. But I think the hub has also, especially with the help of Antonio Bordera, who Isabel mentioned, the hub has been very important in organizing teaching within the Institut Pasteur network in bioinformatics and supporting that. So I think this is not under my umbrella. This is why I didn't talk about it. But this is another component of our resource offer, which we think is essential to, accelerate research.
One more question, Marcelo.
So the question is very simple.
Thank you,
how
Paula.
far do you go into the...
Don't fight.
So the question is, how far do you go in the analysis? Because we can buy a service. It had happened to us. We got a service, and we got a whole bunch of numbers coming back. So then we had to go and get another service to get those numbers transformed into something that we could comprehend. So, I mean, especially with cryo microscopy, I mean, who's going to, does the service contemplate the image analysis?
So the way it's envisaged is that the facility is not going to do the image analysis, okay? So you have to do that, especially because, engaging into a cryo-EM project, especially getting images on the Titan cryos, you need to dedicate your energy onto that. You need to have, so then we have reinforced our expertise in image analysis, so we are hiring someone who is a specialist. We have an image analysis hub within our core facilities, and so these people can be there to help.
The only issue with that is that you only know if the data is good or the quality of the data once the analysis have been made.
So that's one of the things we do. So we are in the process of hiring a new group with specialists in image analysis, and one of the things they will develop is a system to quality control the images that are produced by the microscope, so that you can actually predict whether it's worth continuing the acquisition or stopping the acquisition. So that's something we have in mind.
Antoine.
Yeah, I wanted to congratulate you for the access. It's much easier than 5 years ago, and my scientists are very happy with that.
Okay,
I have two questions and maybe two problems. First, it's a small problem. We have to write a project each time we want to sequence something. Is it to differentiate you from commercial factories, or what is the interest for you to have a project?
I think it's so in terms of sequencing, we have fully reorganized our facility, and one of the ideas was to really streamline the process and ask people to indeed apply each time for a project. I can understand the limitation of that, but in terms of billing, for instance, it makes things a little bit easier.
Okay, and the second question, one scientist in my institute wanted to work on Naegleria florei with on mice.
Yeah, I remember
in the
that.
Petri, in the Petri facilities. But the problem is that they can inoculate the Naegleria, but she has to prepare in another lab, and this is a limitation for us because which lab to
So
go,
I think,
and it's
yeah.
not
So
very
I think
easy.
well, so that's something we are trying to slightly modify in the way the animal facility is organized. So what we realize is that so up to now, people. So you have zootechnicians to take care of the mice, and you have people in the labs who come to the mice and do the experiments, etc. And to do that, they need to get training, and getting training takes time. Okay. So for instance, if you want to do some surgery on animals, you need to take a lot of training. And so what we are trying to do at the moment is to have some of our zootechnicians be able to perform the experiments. So that would actually help. It doesn't help when it comes to growing the bugs. And so that you need to actually identify another lab on campus that would be the place to grow the bugs. I think we can hardly find, organize a core facility that would allow to grow any bugs, etc. But maybe this is something to discuss.
Okay, so I would like to thank you, Christophe, and thank you everybody that is that late to finish this marvelous day that we have had today with different speakers around emerging diseases. We had... No, you can't. Go on. You can go on. I said go on, no problem. What do you want? You can talk 5 minutes. Sinval, just 1 second. It's okay for tonight. Just a message for you. Okay, please say some words about your...
So I spoke only 15 minutes.
15 minutes? No, you have 5 minutes to do that.
Okay, 2 minutes. Institut Pasteur in Paris is actually organizing a number of incentive programs. So there are some incentive programs that are under my responsibility. They are called transversal research programs. There are some incentive programs that are under the joint responsibility of my division and the Department of International Affairs. They are called ASIP. And finally, in Isabel's department, they have a number of incentive programs that are aimed to support innovation and development. I won't talk about this because they are not open to the network yet. But I will talk about the PTR and ASIP. So transversal research programs, they are meant to bring together at least 3 teams from the institute, from the international network. This year, they were focused on the 3 strategic axes of the strategic plan. That is antimicrobial resistance, emerging infectious disease, and neuroscience. And we want them to be coordinated by a young scientist, not someone like me, and they are there to, really promote interaction between groups and promote new ideas, promote new talents. We have funded this year. We have funded 10 such projects, and each project is funded with 250,000 euros for 2 years, which allows to hire a postdoctoral fellow and cover for equipments. I'm talking French rates, not Brazilian rates. And so there is no obligation to have an institute of the network in these projects, but I think almost all, I think 9 out of 10 of the projects we have funded have an institute of the network. And so they are really important for promoting interaction between the different institutes of the network. So the ASIP are a much smaller project because they are funded with 50,000 euros for 2 years, and they are really there to promote interaction between the institutes of the network and allow to kick off an idea. So, you know, it's just a little money to try something, and if that works, then you can move to the transversal research program, or you can move to another funding agency. So I think they are really important. I always, François Romanex is not here, but I always, you know, push him to put more money into these, because I think they are very important for promoting new people, new ideas, new interactions, and promoting the network as well. And one thing we discussed during the evaluation of the ASIP would be to have the association of the Pasteur Institute to actually contribute to this funding, so to increase the volume of the funding and to increase the number of projects that can be funded. So that's a suggestion. I think these projects are funded by the Institut Pasteur in Paris. I think the network should contribute to them to increase their breadth and strength.
Thank you, Christopher. This was very nice. Thank you. Okay, so now we're going to close the session. So I just wanted to remind you that tomorrow we start the session by 9. The invited persons that are in the hotels closer to Hotel RIIP, the bus is going to leave by 8:15. Please be on time. It's going to be a very nice session, I think, that was organized by to celebrate our colleague from Fiocruz, the work of our colleague from Fiocruz, Ricardo Galler. So I think that you are going to like this. We have here Professor Akira Oma that is going to give an historical point of view about the yellow fever. I think it's important that you stay here with us. So unfortunately, you know, we were not able to invite everybody to the dinner at the residence of France because it is a limited number of people that can get there. So I just remember the guys that were invited that there is a bus here. I do not think, I don't know, I'm going to talk with you to know if we can go first to the hotel that is in the direction of the residence or not. So we are going to check with the chauffeur and check this, okay? So I wish you a very nice evening and see you tomorrow.